Frontiers in Science. Genes that map the body. What signals a monkey to grow a tail and a fish to grow fins? The answer is in their genes. What's in a gene? Humans and fish are about as different as one animal can be from another. Yet both organisms have a similar body pattern: front and back, top and bottom, left side and right side. The head is at one end and limbs extend from the body. Fins in a fish, arms and legs in a human. Inside are similar structures: brains, hearts, and stomachs, and cells that function in similar ways. DNA is the genetic material found in all living things. DNA determines how cells grow, develop, and function. Within the DNA are genes, segments of DNA, that determine whether a cell becomes a brain cell or a heart cell. Both a fish and a human start out life as a single cell. As the cell divides again and again, each organism grows into its familiar shape. Scientists are studying what it is that maps out the head-to-tail development that gets every part of a body in the right place. One group of genes, called Hox genes, are critical in the early development of an animal's body. These genes are found in the DNA of every animal, from humans to fruit flies. The position of Hox genes, from top to bottom along the DNA, matches up to the particular parts they control of an organism's body. How Hox genes work. Hox genes act like switches. A particular Hox gene turns on the development of a particular structure in an animal's body. One type of Hox gene switches on the development of structures in the head, for example, eyes and ears. Another switches on the development of limbs, the arms, legs, fins, or wings of an animal. The position of the genes within an animal's DNA matches to the parts of the body it controls. Hox genes at the top control development of parts of the head. Those toward the middle control development of the main part of the body and the limbs. How a limb develops. What happens if a Hox gene gets out of position? If the Hox gene that controls the development of legs in a fruit fly is placed in with the Hox genes that control development of the head, the fruit fly will grow legs from its head. The gene functions as it should. It's just that it's not doing its job in the right place. Another interesting thing about Hox genes is that they are active only for a certain period of time. They switch off when the part of the body they control has developed. Studies of the zebrafish have provided clues as to how this happens. A window on development. Zebrafish are tiny fish that hatch in about 3 days. Scientists can actually see through the fish egg to watch its body develop. Working with the Hox genes of zebrafish, one researcher studied the amount of time that the Hox gene that controls fin development was active. The gene turned on for a short period of time. The fin developed. Then the gene turned off. This research led scientists to think about the length of time the same Hox gene is active in other animals. It's possible that limbs are longer in larger animals because their Hox genes are active for a longer period of time. Researchers are excited because what they learn about the Hox genes of a simple animal can provide clues into the development of larger, more complex animals. Unanswered questions. There are many unanswered questions about the role Hox genes play in the development of body plans. Which Hox genes control which stages of development, and how long are the genes active? What is it that signals the genes to turn on and off? How can research on Hox genes be used by medical researchers to help them treat genetic diseases or disorders that affect how a body develops?
Chapter 1. The Cell. The Big Idea. All living things are made up of cells. Key Concepts. Section 1.1. The cell is the basic unit of living things. Learn why cells are important to the study of life. Section 1.2. Microscopes allow us to see inside the cell. Learn what microscopes have shown about the inner structure of cells. Section 1.3. Different cells perform various functions. Learn about different types of cells in both unicellular and multicellular organisms. 1.1. Key concept. The cell is the basic unit of living things. Before, you learned, living things have common characteristics. Living things have common needs. A theory is something that explains what is observed in nature. Now you will learn how living things are different from non-living things, how the microscope led to the discovery of cells. About the cell theory. Living things are different from non-living things. You know life when you see it. Perhaps your class takes a field trip to a local state park to collect water samples. You are surrounded by trees. There is a stream with rocks covered with moss and green algae. There are fish and frogs. There are birds and insects. You are surrounded by life. But how would you define it? One way to answer the question is to think about what makes a living thing different from a non-living thing. You might ask if a thing uses energy, or maybe you would observe it to see if it moves. You could investigate whether it consumes food and water. These are characteristics of living things or organisms. Any individual form of life that is capable of growing and reproducing is an organism. All organisms get water and other materials from the environment. Characteristics of life. Living things have these characteristics: organization, the ability to develop and grow, the ability to respond to the environment, the ability to reproduce. An organism's body must be organized in a way that enables it to meet its needs. Some organisms, like bacteria, are very simple. A more complex organism, such as the kingfisher shown in the photograph below, is organized so that different parts of its body perform different jobs, called functions. For example, a kingfisher has wings for flying, a heart for pumping blood, and eyes for seeing. Another characteristic of organisms is that they grow and, in most cases, develop into adult forms. Some organisms change a great deal in size and appearance throughout their lifetimes, whereas others grow and change very little. Organisms also respond to the world outside them. Think of how the pupils of your eyes get smaller in bright light. Finally, organisms can reproduce, producing new organisms that are similar to themselves. Check your reading. What 4 characteristics are common to all living things? Needs of life. Organisms cannot carry out the activities that characterize life without a few necessities: energy, materials, and living space. What does it mean to need energy? You know that if you want to run a race, you need energy. But did you know that your body also needs energy to sleep, or to breathe, or even to think? All organisms require a steady supply of energy to stay alive. Where does this energy come from, and how does an organism get it? The energy used by almost all forms of life on Earth comes from the sun. Some organisms, like plants and some bacteria, are able to capture this energy directly. Your body, like the bodies of other animals, uses food as a source of energy. The food animals eat comes from plants or from organisms that eat plants. Food also provides the materials necessary for growth and reproduction. These materials include substances such as carbon dioxide, nitrogen, oxygen, and water. Finally, all organisms need space to live and grow. If any one of these requirements is missing, an organism will die. All living things are made of cells. The cell is the smallest unit of a living thing. Some organisms are made of a single cell. These organisms are unicellular and usually too small for you to see directly. Pond water is full of tiny unicellular organisms. Most of the organisms you can see, such as a frog or a water lily, are made up of many cells. Organisms made up of many cells are called multicellular organisms. The needs and characteristics of a single cell in a unicellular organism are the same as those for any organism. Each of the tiny single-celled organisms found in a drop of pond water performs all the activities that characterize life. Multicellular organisms, like a frog or a water lily, have bodies that are more complex. Different parts of the body of a multicellular organism perform different functions. A water lily's roots hold it in the soil, and its leaves capture energy from the sun. A frog moves with its arms and legs and eats with its mouth. Multicellular organisms have different types of cells that make up their body parts and help the organisms meet their needs. Roots are made of root cells, which are different from leaf cells. Muscle cells have special parts that allow them to move. In a multicellular organism, many cells work together to carry out the basic activities of life. The microscope led to the discovery of cells. Most cells are microscopic, too small to see without the aid of a microscope. A microscope is an instrument which makes an object appear bigger than it is. It took the invention of this relatively simple tool to lead to the discovery of cells. In the 1660s, Robert Hooke began using microscopes to look at all sorts of materials. Anton van Leeuwenhoek took up similar work in the 1670s. They were among the first people to describe cells. Robert Hooke gave the cell its name. While looking at a sample of cork, a layer of bark taken from an oak tree, he saw a group of similarly shaped compartments that looked to him like tiny empty rooms, or cells. You can see from his drawing, shown at right, how well these cells fit Hooke's description. Hooke used a microscope that magnified objects 30 times. In other words, objects appeared 30 times larger than their actual size. The bark cells Hooke saw were actually dead cells, which is why they appeared empty. Anton van Leeuwenhoek was one of the first people to describe living cells. He looked at a drop of pond water under a microscope. Imagine his surprise when he saw that a drop of water was full of living things. Using lenses that could magnify an object almost 300 times, he observed tiny unicellular organisms like those shown on page 11. Check your reading. How did the invention of the microscope change the study of biology? You can understand how powerful a microscope is if you think of how big a penny would be if it were increased in size 30 times. It would be a little bigger than the tire of a 10-speed bicycle. Enlarged 300 times, that penny would be so big that you would need a tractor-trailer to move it. Magnify your best friend 30 times, supposing a height of 1 5/10 meters, or almost 5 feet, and your friend would appear to be 45 meters, 147 feet tall. That's almost the height of Niagara Falls. Change the magnification to 300 times, and your friend would appear to be 450 meters, 1,470 feet tall, taller than the Empire State Building. Cells come from other cells. The studies of Hooke and Leeuwenhoek made people ask if all living things have cells. People continued to observe samples taken from all sorts of living matter. They continued to find cells, although often these cells looked very different from one another. Still, it was clear that all living matter was made of cells. There was another important question scientists were trying to answer. Where do cells come from? The answer to this question was settled by the 1850s. People studying all types of living cells observed the same thing, that cells divide. One living cell divides into two living cells. Here, under the microscope, was evidence of where cells come from. Life comes from life. That is, one cell comes from another cell. Check your reading. What do scientists mean when they say that life comes from life? Your answer should include the word cells. The observations and evidence gathered over a long time by many scientists are summarized in the three concepts of the cell theory. 1. Every living thing is made of one or more cells. 2. Cells carry out the functions needed to support life. 3. Cells come only from other living cells. The cell theory is important to the study of biology. The three ideas on page 13 are so important that they are grouped together using the word theory. A scientific theory is a widely accepted explanation of things observed in nature. A theory must be supported by evidence, including experimental evidence and observations. A theory proves its value when it explains new discoveries and observations. Check your reading. What are two characteristics of a scientific theory? Theories are important for a number of reasons. Certainly, they satisfy scientists' desire to understand the natural world, and they serve as foundations for further research and study. Theories can also lead to research that has some practical benefit for society. Louis Pasteur. The work of the French scientist Louis Pasteur shows how an understanding of cell theory can have practical uses. Pasteur lived in the 1800s, when there was no mechanical refrigeration in homes. People were used to having food spoil, like milk going sour. During this time, many people died from diseases such as typhoid fever, tuberculosis, and diphtheria. Pasteur's work showed that microscopic organisms were involved both in the spoilage of food and in disease. Pasteur observed that milk that turned sour contained large numbers of tiny single-celled organisms called bacteria. He developed a process, now known as pasteurization, in which heat is used to kill the bacteria. Killing the bacteria keeps milk fresh longer. The fact that bacteria cause milk to sour or sicken made Pasteur wonder whether microscopic organisms could also be the cause of sickness in humans and animals. Bacteria and spontaneous generation. Using a microscope to study air, water, and soil, Pasteur found microorganisms everywhere. He found bacteria in the blood of animals, including people who were sick. Pasteur referred to the microorganisms he observed as germs. He realized that an understanding of germs might help prevent disease. Pasteur's work led to the first animal vaccinations for cholera and anthrax, and to a treatment for rabies in humans. At the time Pasteur was doing his research, there were scientists who thought that bacteria grew from non-living materials, an idea called spontaneous generation. Pasteur conducted a now famous series of experiments that disproved spontaneous generation and confirmed the cell theory. He showed that cells come only from other cells. Two of these experiments are shown above. Both began with a sealed flask containing boiled broth. In the first experiment, the flask remained sealed, while in the second experiment, the top of the flask was broken to expose the contents to air. Bacteria grew only in the second flask. 1.1 Review. Key concepts. 1. Name 4 characteristics of living things. 2. How did the microscope change human understanding of life? 3. Explain the 3 concepts that make up the cell theory. Critical thinking. 4. Analyze. Relate the characteristics of a scientific theory to the cell theory. 5. Compare and contrast. Draw a Venn diagram to compare and contrast multicellular and unicellular organisms. Challenge. 6. Synthesize. Explain how Pasteur's experiment supported the cell theory and disproved the theory of spontaneous generation. 1.2. Key concept. Microscopes allow us to see inside the cell. Before, you learned some organisms are unicellular and some are multicellular. A microscope is necessary to study most cells. The cell theory describes the cell as the fundamental unit of life. Now you will learn about different types of microscopes, about prokaryotic and eukaryotic cells, how plant and animal cells are similar and different. Think about how small are cells? Because cells are so small, describing them requires a very small unit of measure, the micrometer. A micrometer is 1/1,000,000th of a meter. Cells vary in size from about 1 micrometer, some bacteria, to 1,000 micrometers, some plant and animal cells. To get a sense of the sizes of cells, consider that it would take about 17,000 tiny bacterial cells lined up to reach across a dime. How many of these cells might fit on your fingertip? The microscope is an important tool. The invention of the light microscope led to the discovery of cells and to the development of cell theory. In light microscopes, lenses are used to bend light and make objects appear bigger than they are. Modern light microscopes can magnify objects up to 1,000 times. The light microscope is still used today to study cells. Over many years, scientists have found ways to make light microscopes more useful. Cell samples are treated with dyes to make structures in the cells easier to see. Scientists use video cameras and computer processing to observe the movement of cell parts and materials within cells. One important advantage of light microscopes is that scientists can observe living cells with them. Two other types of microscopes are important in the study of cells: the scanning electron microscope, SEM, and the transmission electron microscope, TEM, can produce images of objects as small as 2,000 micrometers. The light microscope can be used only for objects that are larger than 2/10 micrometers. Therefore, although a light microscope can be used to see many of the parts of a cell, only the SEM and TEM can be used for looking at the details of those parts. In both the SEM and the TEM, tiny particles called electrons, not light, are used to produce images. The advantage of these microscopes is that they can magnify objects up to 1,000,000 times. The disadvantage is that they cannot be used to study live specimens. Check your reading. Compare light microscopes with electron microscopes. What are the advantages and disadvantages of each? To be viewed with an SEM, a cell sample is coated in a heavy metal, such as gold. Then a beam of electrons is run back and forth over the surface of the cell. The electrons bounce off the coating and are read by a detector that produces a three-dimensional image of the surface. A cell viewed with a TEM is sliced extremely thin. Electrons pass through a section. Images produced by a TEM appear two-dimensional. Cells are diverse. Very early on, the people studying cells knew that cells have a great diversity of sizes and shapes. As microscopes were improved, scientists could see more and more details of cells. What they saw was that the inside of one cell can be very different from that of another cell. Every cell has a boundary that separates the inside from the outside. That boundary is the cell membrane, a protective covering that encloses the entire cell. Any material coming into or out of the cell must pass through the cell membrane. Contained inside the cell membrane is a gelatin-like fluid called cytoplasm. Most of the work of the cell is carried out in the cytoplasm. Scientists separate cells into two broad categories based on one key difference: the location of the genetic material cells need to reproduce and function. In a eukaryotic cell, the genetic material is in a structure called the nucleus, a structure enclosed by its own membrane. Scientists use the word organelle to describe any part of a cell that is enclosed by membrane. In a prokaryotic cell, there is no separate compartment for the genetic material. Instead, it is in the cytoplasm. There are no organelles. Most unicellular organisms are prokaryotic cells. Almost all multicellular organisms are eukaryotic. Plants and animals have eukaryotic cells. Plant and animal cells, like all eukaryotic cells, are divided into two main compartments. The nucleus, the largest organelle, is the compartment that stores all the instructions a cell needs to function. You will learn more about how cells use this information in Chapter 5. Surrounding the nucleus is the cytoplasm. The cell membrane is the boundary between the cytoplasm and the outside of the cell. Plant cells also have cell walls. A cell wall is a tough outer covering that lies just outside the cell membrane. The cell wall supports and protects the cell. Having a cell wall is one important way in which plant cells differ from animal cells. Structures that process information. The nucleus is often the largest organelle in a cell. It contains information a cell needs to function. Some of the information is translated by ribosomes, tiny structures located in the cytoplasm and the endoplasmic reticulum. Ribosomes use the information to build important molecules called proteins. Organelles that provide energy. No cell can stay alive without energy. Cells need energy to perform all the activities of life. Plants get their energy directly from the sun. Within plant cells are chloroplasts, organelles in which the energy from sunlight is used to make sugar. Plants use some of the sugar immediately to keep their cells functioning. The rest of the sugar is stored in the cells. Animal cells do not contain chloroplasts. As a result, animals are not able to use the energy of the sun directly. Instead, animals get their energy from food. Much of the food an animal uses for energy comes from the sugar that plant cells have stored. Animals can get this energy by eating plants or by eating animals that have eaten plants. Check your reading. How can a chloroplast, a structure found in plant cells but not in animal cells, provide energy for both plants and animals? Both plant cells and animal cells must be able to use energy to do work. The energy is made available by organelles found in all eukaryotic cells. Mitochondria are the organelles that use oxygen to get energy from processing food. Organelles that process and transport. You know that plant and animal cells get their energy from the sugars that the organisms make or consume. Sugars are also an important part of the starting materials that cells use to maintain themselves and grow. The job of making cell parts from the starting materials that enter a cell is divided among a number of structures in the cytoplasm. In the illustrations on page 22, you can see that the endoplasmic reticulum is a system of twisting and winding membranes. Some of the endoplasmic reticulum contains ribosomes, which manufacture proteins. The endoplasmic reticulum manufactures parts of the cell membrane. The endoplasmic reticulum is also part of the cellular transport system. Portions of endoplasmic reticulum break off to form small packages called vesicles. The vesicles transport processed materials to an organelle called the Golgi apparatus. The folded membranes of the Golgi apparatus make it look something like a stack of pancakes. The Golgi apparatus takes the materials manufactured by the endoplasmic reticulum and finishes processing them. Organelles for storage, recycling, and waste. Cells store water, sugar, and other materials, which they use to function. Cells must also store waste materials until they can be removed. Inside plant and fungus cells are sacs called vacuoles. Vacuoles are enclosed by a membrane and can hold water, waste, and other materials. Vacuoles function within the cell membrane to move materials either into or out of the cell. A plant cell has a large central vacuole in which water and other materials can be stored. Water in the vacuole provides support for smaller plants. Animal cells do not have central vacuoles. What animal cells do have are similar structures called lysosomes. Lysosomes are vesicles that contain chemicals that break down materials taken into the cell, as well as old cell parts. Remember that animals, unlike plants, take in food. Nutrients brought into the cell need to be broken down, as well as wastes contained. Check your reading. Compare and contrast lysosomes and central vacuoles. 1.2 Review. Key concepts. 1. What advantages and disadvantages does a light microscope have in comparison with an electron microscope? 2. What is the difference between a eukaryotic cell and a prokaryotic cell? 3. List 3 structures found in plant cells that are not in animal cells. Critical thinking. 4. Synthesize. What organelles can be said to act like an assembly line within a cell? Explain. 5. Compare and contrast. Make a Venn diagram comparing and contrasting plant and animal cells. Challenge. 6. Synthesize. Identify the type of microscope used to capture the image at the right and indicate whether the cell is a plant cell or an animal cell. How do you know? 1.3. Key concept. Different cells perform different functions. Before, you learned modern microscopes reveal details of cell structures. Some cells are prokaryotic, and some are eukaryotic. Plant and animal cells have similarities and differences. Now you will learn how organisms are classified into 3 domains, about specialization in multicellular organisms, how cells, tissues, and organs are organized. Organisms can be classified by their cell type. Look around you at this moment. The living organisms you see may number 10, 20, 100, or 1,000, depending on where you are. What you are not seeing, but what is also there, is a huge number of unicellular organisms. For example, there are at least 2 to 3 million bacteria living on each square centimeter of your skin. Most of the organisms alive on Earth today are made of a single cell. One of the most interesting scientific discoveries made recently had to do with a group of unicellular organisms. These organisms were found living where no one expected to find any life at all. Archaea and bacteria. In the early 1980s, scientists discovered unicellular organisms living in rather extreme environments. Some were living deep in the ocean, at thermal vents where there is extreme heat and little oxygen. Others were found in the salty waters of the Great Salt Lake and in the hot sulfur springs of Yellowstone Park. At first, these organisms were referred to as archaebacteria. The organisms were similar in appearance to bacteria. The prefix archae comes from a Greek word that means ancient. Many of these organisms live in environments that scientists think are like the environments of ancient Earth. It took a while for scientists to realize that these organisms that looked like bacteria were genetically very different from bacteria. Scientists decided to establish a separate category for them, a domain called archaea. A domain is a broad category of living things that is based on characteristics of their cells. Scientists have identified 3 domains. Bacteria are classified in the domain bacteria. A third domain includes organisms with eukaryotic cells. Organisms that belong to the domains bacteria and archaea are similar in some important ways. They are prokaryotes, which are unicellular organisms made of prokaryotic cells. Their cytoplasm contains ribosomes but no organelles, so the structure of a prokaryote is simple. Another feature of a prokaryote is a tough cell wall that protects the organism. Check your reading. Why did scientists decide to establish separate domains for archaea and bacteria? Eukarya. The third domain is the domain eukarya. Organisms in this domain have cells with a nucleus. This domain includes almost all the multicellular organisms on Earth: plants, animals, and fungi. It also includes many unicellular organisms called protists. The cells of unicellular eukaryotes are more complex in structure and larger than the cells of prokaryotes. Check your reading. How are eukaryotes different from prokaryotes? The paramecium is one of the most complex of all unicellular eukaryotes. Its body is lined with hair-like strands called cilia that allow it to move. It has dart-like structures that carry a substance used in healing and perhaps defense. Along the outside of the cell is a long oral groove lined with cilia that leads to a mouth pore. In addition to a nucleus, the cell of a paramecium has organelles that enable it to digest food and remove water and wastes. The paramecium has all it needs to live as a single cell. By comparison, in most multicellular eukaryotes, no individual cell can survive on its own. Cells in multicellular organisms specialize. Most multicellular organisms consist of many different types of cells that do different jobs. For example, most animals have blood cells, nerve cells, and muscle cells. The cells are specialized. Specialization of cells means that specific cells perform specific functions. This specialization is why a single cell from a multicellular organism cannot survive on its own. A blood cell can help you fight infection or deliver oxygen to your muscles, but it cannot cause your body to move as a muscle cell can. Plants have cells that function in photosynthesis and other cells that draw water from the soil, and still others that function mainly to support the plant's weight. Check your reading. What does it mean for a cell to be specialized? A multicellular organism is a community of cells. Cells in a multicellular organism are specialized. The ways in which the cells work together and interact depend on the organism. You can think of the cells of an organism as members of a community. The size and complexity of the community differ from organism to organism. A sponge is an animal that is fairly simple in its organization. It spends its life attached to the ocean floor, filtering food and other nutrients from the water. Like all animals, the sponge is organized at a cellular level. Different types of cells in its body perform different functions. For example, certain cells take in food, and other cells digest it. However, cells in a sponge are not very highly specialized. A piece broken from a living sponge will actually regenerate itself as new cells replace the lost ones. In more complex organisms, such as plants and animals, cells are not only specialized, but grouped together in tissues. A tissue is a group of similar cells that are organized to do a specific job. If you look at your hand, you will see the top layer of tissue in your skin. Humans have two layers of skin tissue, layered one on top of the other. Together, these skin tissues provide protection and support. Check your reading. In what way is a tissue an organization of cells? Different tissues working together to perform a particular function represent another level of organization, the organ. The eye is an organ that functions with the tarsiers brain to allow sight. A leaf is an organ that provides a plant with energy and materials. It has tissue that brings in water and nutrients, tissue that uses the sun's energy to make sugar, and tissue that moves sugar to other parts of the plant. Check your reading. What is the relationship between tissues and organs? Different organs and tissues working together form an organ system. An organism may have only a few organ systems. The organ systems of plants include roots, stems, and leaves. Other organisms have many organ systems. Humans have 11 major organ systems, made up of about 40 organs and over 200 types of tissue. The human nervous system, for example, includes the brain, the spinal cord, nerves, and sensory organs such as the ears and eyes. An organism itself represents the highest level of organization. It is at this level that we see all the characteristics we associate with life. If an organism is a complex organism, a human for example, it will consist of trillions of cells grouped into tissues, organs, and organ systems. However, a simple organism, like a sponge, meets its needs with a body made up of only a few types of specialized cells. Check your reading. What level of organization is an organism? What do we see at this level of organization? Scientists use models to study cells. Any drawing or photograph on a flat page is two-dimensional. In addition, diagrams of cells are often simplified to make them easier to understand. If you look at plant or animal cells under a microscope, you will notice some differences between real cells and the diagrams on page 22. In order to study cell structures and their functions, scientists use many types of models, including three-dimensional models. One of the most important discoveries in science involved the use of models. DNA is the genetic material common to all cells. You will read more about the structure and function of DNA later in this unit. In the early 1950s, scientists had a good idea what DNA was made up of. The problem was that they could not figure out how all the pieces of the molecule fit together. A scientist named Rosalind Franklin used X-rays to produce images of DNA. The X-ray provided an important clue as to the shape of the molecule. Two other scientists, James Watson and Francis Crick, were then able to put together a three-dimensional model of DNA and present it to the world in 1953. Today's scientists have many different tools for making models. The images at the left show a computer model of DNA along with Watson and Crick's famous model. 1.3. Review. Key concepts. 1. What are the three domains, and what type of cells do the organisms in each domain have? 2. Define specialization in your own words. 3. Describe the levels of organization in a tree. Critical thinking. 4. Synthesize. In what way does a specialized cell in a multicellular organism differ from the cell of a unicellular organism? 5. Compare and contrast. How is a model similar to the real object it represents? How is it different? Challenge. 6. Evaluate. The organism below is called Chlamydomonas. What domain does it belong to? And what do the internal structures tell you about it? Think science. Skill. Making comparisons. Cells and spacesuits. What do a spacesuit and a unicellular organism have in common? Both have to support life, and both can support life in difficult environments. What are some of the similarities and differences between the cell body of a unicellular organism and a spacesuit that supports an astronaut in outer space? Some features of spacesuits. Feature. Function. Strong outer material protects the astronaut from space particles. A special jet-propelled backpack helps the astronaut move in the weightlessness of space. Tanks of compressed air provide oxygen for the astronaut to breathe. Some features of cells. Feature. Function. Tail-like flagella on the outside of some cells help cells move. An outer membrane keeps harmful particles out. Tiny openings in a cell's membrane let oxygen move into the cell. Make comparisons. On your own. Match each cell feature with a similar spacesuit feature. What characteristics do the cell and spacesuit have in common? What is one key difference? As a group, use your comparisons to make a Venn diagram. Challenge. An analogy uses a familiar thing to help explain or describe something new. Come up with your own analogies to describe the cell or some of its organelles.
Chapter 2. How Cells Function. The Big Idea. All cells need energy and materials for life processes. Key concepts. Section 2.1. Chemical reactions take place inside cells. Learn why water and 4 types of large molecules are important for cell functions. Section 2.2. Cells capture and release energy. Learn about the process of photosynthesis and the 2 ways cells release energy. Section 2.3. Materials move across the cell's membranes. Learn about the different ways materials move through cells. 2.1. Key concept. Chemical reactions take place inside cells. Before you learned, all living things are made of cells. Cells need energy to sustain life. Plant and animal cells have similarities and differences. Now you will learn about the types of elements found in all cells, about the functions of large molecules in the cell, why water is important to the activities of the cell. All cells are made of the same elements. The microscope allowed people to observe the tiny cells that make up all living things. Even smaller, too small for a light microscope to show, is the matter that makes up the cell itself. All matter in the universe, living and non-living, can be broken down into basic substances called elements. About 100 different elements are found on Earth. Each element has its own set of properties and characteristics. For example, the characteristics of oxygen include that it is colorless, odorless, and on Earth it exists in the form of a gas. Of all the elements found on Earth, about 25 are essential for life. As you can see from the table, just 6 elements account for about 99% of the mass of the human body. But very little of this matter exists as pure elements. Instead, most is in the form of compounds, which are substances made up of 2 or more different elements. For example, water is a compound made of hydrogen and oxygen. The smallest unit of any element is called an atom. In a compound, atoms of 2 or more elements are joined together by chemical bonds. Most compounds in cells are made up of atoms bonded together in molecules. For example, a molecule of water is made up of 1 atom of oxygen bonded to 2 atoms of hydrogen. Most activities that take place within cells involve atoms and molecules interacting. In this process, called a chemical reaction, bonds between atoms are broken, and new bonds form to make different molecules. Energy is needed to break bonds between atoms, and energy is released when new bonds form. Cells use chemical energy for life activities. Large molecules support cell function. In living things, there are 4 main types of large molecules. 1. Carbohydrates. 2. Lipids. 3. Proteins. And 4. Nucleic acids. Thousands of these molecules work together in a cell. The 4 types of molecules in all living things share one important characteristic. They all contain carbon atoms. These large molecules are made up of smaller parts, called subunits. Carbohydrates. Carbohydrates provide the cell with energy. Simple carbohydrates are sugars made from atoms of carbon, oxygen, and hydrogen. Inside cells, sugar molecules are broken down. This process provides usable energy for the cell. Simple sugar molecules can also be linked into long chains to form more complex carbohydrates, such as starch, cellulose, and glycogen. Starch and cellulose are complex carbohydrates made by plant cells. When a plant cell makes more sugar than it can use, extra sugar molecules are stored in long chains called starch. Plants also make cellulose, which is the material that makes up the cell wall. Animals get their energy by eating plants or other animals that eat plants. Lipids. Lipids are the fats, oils, and waxes found in living things. Like carbohydrates, simple lipids are made of atoms of carbon, oxygen, and hydrogen, and can be used by cells for energy and for making structures. However, the atoms in all lipids are arranged differently from the atoms in carbohydrates. Many common lipids consist of a molecule called glycerol, bonded to long chains of carbon and hydrogen atoms, called fatty acids. This structure gives lipids unique properties. One extremely important property of lipids is that they cannot mix with water. Check your reading. How do cells use carbohydrates and lipids? Proteins. Proteins are made of smaller molecules called amino acids. Amino acids contain the elements carbon, oxygen, hydrogen, nitrogen, and sometimes sulfur. In proteins, amino acids are linked together into long chains that fold into three-dimensional shapes. The structure and function of a protein is determined by the type, number, and order of the amino acids in it. Your body gets amino acids from protein in food, such as meat, eggs, cheese, and some beans. After taking in amino acids, your cells use them to build proteins needed for proper cell functioning. Some amino acids can be made by the body, but others must be taken in from an outside food source. There are many types of proteins. Enzymes are proteins that control chemical reactions in the cells. Other proteins support the growth and repair of living matter. The action of proteins in your muscles allows you to move. Some of the proteins in your blood fight infections. Another protein in your blood delivers oxygen to all the cells in your body. Proteins are also important parts of cell membranes. Some proteins in the cell membrane transport materials into and out of the cell. Nucleic acids. Nucleic acids are the molecules that hold the instructions for the maintenance, growth, and reproduction of a cell. There are two types of nucleic acids, DNA and RNA. Both DNA and RNA are made from carbon, oxygen, hydrogen, nitrogen, and phosphorus. The subunits of nucleic acids are called nucleotides. DNA provides the information used by the cell for making proteins a cell needs. This information takes the form of a code contained in the specific order of different nucleotides in the DNA. The pattern of nucleotides in DNA is then coded into RNA, which delivers the information into the cytoplasm. Other RNA molecules in the cytoplasm produce the proteins. Check your reading. What is the function of DNA and RNA? About 2/3 of every cell is water. All of the chemical reactions inside the cell take place in water. Water is also in the environment outside the cell. For example, water inside cells makes up about 46% of your body's mass, and water outside the cells in body fluids accounts for another 23%. A water molecule consists of 2 atoms of hydrogen bonded to 1 atom of oxygen. Because of its structure, a water molecule has a slight positive charge near the hydrogen atoms and a slight negative charge near the oxygen atom. Molecules that have slightly charged ends are said to be polar. Like a magnet, the ends of a polar molecule attract opposite charges and repel charges that are the same. Because water is a polar molecule, many substances dissolve in water. However, not all materials dissolve in water. If you have ever shaken a bottle of salad dressing, you've probably observed that oil and water don't mix. Most lipids do not dissolve in water. A special type of lipid is the major molecule that makes up cell membranes. These special lipid molecules have 2 parts, a water-loving head and 2 water-hating tails. In other words, the head of the lipid molecule is polar, while the tails are nonpolar. Why is it important that cell membranes contain lipids? Remember that cell membranes function as boundaries. That is, they separate the inside of a cell from the outside. Most of the material inside and outside the cell is water. As you can see in the diagram above, the water-hating tails in the lipids repel the water, while the head clings to water. 2.1 Review. Key concepts. 1. Explain how just a few elements can make up all living things. 2. What functions do proteins, carbohydrates, lipids, and nucleic acids perform? 3. What does it mean to describe water molecules as being polar? Critical thinking. 4. Compare and contrast. How are carbohydrates and lipids similar? How are they different? 5. Draw conclusions. What do the major types of molecules that make up living things have in common? Challenge. 6. Model. Some people have compared the nucleic acids DNA and RNA to a blueprint for life. How are DNA and RNA like blueprints? How are they different? Science on the job. Textile designer. Natural dyes and cells. Where does the blue in your blue jeans come from? How about the red, yellow, green, or pink in your favorite wool or cotton sweater? Most fabrics are colored with dyes made up in labs, but some designers prefer to use natural dyes and natural cloth. All textile designers must understand the science of dyes and fibers to produce the colors they want. Fibers. Natural fibers come either from plants or animals. Wool is an animal fiber. Silk, too, is made up of animal cells. Cotton, linen, and rayon are fibers made from plants. Plant fibers have thick cell walls, made mostly of cellulose. Animal fibers, on the other hand, contain mainly proteins. Dyes. Most natural dyes come from plants, but sometimes insects are used too. The indigo plant is used for most blue, including the original blue jeans. Insects are most often used to make reds. All dyes are made of molecules: carbon, oxygen, hydrogen, and other atoms. The molecules of the dye bind to the molecules of the fibers, adding the dye's color to the fiber. Color fixers. A mordant is a chemical compound that combines with dye as a color fixer, or color keeper. The mordant must strengthen the bonds between natural dye molecules and fibers. A stronger bond means the color is less likely to fade or wash out of the fibers. Most mordants are liquid solutions containing metals, such as chromium. Explore. 1. Experiment. Design an experiment using onion skins, beets, or blackberries to color white wool and white cotton. The procedure should include chopping the plant and heating it with water to make the dye. Be sure that your experiment procedure includes only one variable. Your experiment should start with a question, such as, how do the dyes differ? Or, which dye works best? 2. Challenge. Using different mordants with the same dye can give different colors. For example, dandelion leaf dye gives yellow-green, gray-green, tan, or gold with different mordants. Explain why this happens. 2.2. Key concept. Cells capture and release energy. Before you learned, the cell is the basic unit of all living things. Plant cells and animal cells have similarities and differences. Plants and animals need energy and materials. Now you will learn why cells need energy, how energy is captured and stored, how plants and animals get energy. Think about, what do these cells have in common? Both muscle cells and plant cells need energy to live. Your muscle cells need energy to help you move and perform other functions. Even though plant cells don't move in the same way that muscles move, they still need energy. How do human muscle cells and plant cells get energy? All cells need energy. To stay alive, cells need a constant supply of energy. Animal cells get energy from food, while plant cells get energy from sunlight. All cells use chemical energy. Chemical energy is the energy stored in the bonds between atoms of every molecule. To stay alive, cells must be able to release the chemical energy in the bonds. A major energy source for most cells is stored in a sugar molecule called glucose. When you need energy, Cells release chemical energy from glucose. You need food energy to run, walk, and even during sleep. Your cells use energy from food to carry out all their activities. Think about muscle cells. When you run, muscle cells release chemical energy from glucose to move your legs. The more you run, the more glucose your muscle cells need. You eat food to restore the glucose supply in muscles. But how do plant cells get more glucose? Plants transform the energy in sunlight into the chemical energy in glucose. Some cells capture light energy. The source of energy for almost all organisms ultimately comes from sunlight. Plants change the energy in sunlight into a form of energy their cells can use, the chemical energy in glucose. All animals benefit from the ability of plants to convert sunlight to food energy. Animals either eat plants or they eat other animals that have eaten plants. Photosynthesis is a process that plant cells use to change the energy from sunlight into chemical energy. Photosynthesis takes place in plant cells that have chloroplasts. Chloroplasts contain chlorophyll, a light-absorbing pigment or colored substance that traps the energy in sunlight. The process of photosynthesis involves a series of chemical steps or reactions. The illustration on the next page shows an overview of how photosynthesis changes starting materials into new products. 1. The starting materials of photosynthesis are carbon dioxide and water. The plant takes in carbon dioxide from the air and water from the soil. 2. The process takes place when carbon dioxide and water enter the plant's chloroplasts. Chlorophyll captures energy from sunlight, which is used to change carbon dioxide and water into new products. 3. The products of photosynthesis are oxygen and sugars such as glucose. The plant releases most of the oxygen to the air as a waste product and keeps the glucose for its energy needs. Check your reading. Summarize photosynthesis. Remember that a summary includes only the most important information. Plants do not immediately use all of the glucose they make. Some of the glucose molecules are linked together to build large carbohydrates called starch. Plants can store starch and later break it back down into glucose or other sugars when they need energy. Sugars and starches supply food for animals that eat plants. All cells release energy. All cells must have energy to function. Glucose and other sugars are cell food. They are the power source for cell activities in almost all living things. When glucose is stored as glycogen or taken in as starch, it must be broken down into individual sugar molecules before cells are able to use it. Chemical energy is stored in the bonds of sugars. When a sugar molecule is broken down, a usable form of energy is released for the cell's life functions. Cells can release energy in two basic processes: cellular respiration and fermentation. Cellular respiration requires oxygen, but fermentation does not. In addition, cellular respiration releases much more usable energy than does fermentation. Check your reading. What is released when a sugar molecule is broken down? Cellular respiration. In cellular respiration, cells use oxygen to release energy stored in sugars such as glucose. In fact, most of the energy used by the cells in your body is provided by cellular respiration. Just as photosynthesis occurs in organelles called chloroplasts, cellular respiration takes place in organelles called mitochondria. Remember that mitochondria are in both plant cells and animal cells, so both kinds of cells release energy through cellular respiration. Like photosynthesis, cellular respiration is a process that changes starting materials into new products. 1. The starting materials of cellular respiration are sugars, such as glucose, and oxygen. 2. The process begins when glucose in the cytoplasm is broken down into smaller molecules. This releases a small amount of energy. These molecules then move into the mitochondria. At the same time, oxygen enters the cell and travels into the mitochondria. As the smaller molecules are broken down even further, hydrogen is released in a way that allows cells to capture energy in a usable form. The hydrogen combines with oxygen to make water. 3. The products are energy, carbon dioxide, and water. Some of the energy released during cellular respiration is transferred to other molecules, which then carry the energy where it is needed for the activities of the cell. The rest of the energy is released as heat. Carbon dioxide formed during cellular respiration is released by the cell. Check your reading. What are the 3 products of cellular respiration? You may find it interesting to compare cellular respiration with photosynthesis. The diagram above highlights the cycle that occurs between photosynthesis and cellular respiration. Notice that the starting materials of one process are also the products of the other process. This cycle does not necessarily occur in the same cell or even in the same organism. Fermentation. Fermentation is a process by which cells release energy without oxygen. Recall that in cellular respiration, the cell first breaks glucose into smaller molecules. This releases a small amount of energy. Without oxygen, cellular respiration cannot continue. In eukaryotic cells, instead of entering the mitochondria, these smaller molecules stay in the cytoplasm, where fermentation occurs. There are 2 types of fermentation: alcoholic fermentation and lactic acid fermentation. Both types of fermentation break sugars down to small molecules. In the absence of oxygen, different reactions occur that produce either alcohol and carbon dioxide or lactic acid. In both cases, a small amount of energy is released. Check your reading. Use a Venn diagram to compare and contrast fermentation and cellular respiration. The production of many foods that people eat every day involve either alcoholic fermentation or lactic acid fermentation. 3 important foods are bread, yogurt, and cheese. Bread is often made by mixing flour, milk, and sugar with a microorganism you know as yeast. Yeast runs out of oxygen and uses fermentation to convert the sugar into alcohol and carbon dioxide. Bubbles of carbon dioxide gas forming inside the dough cause it to rise. When the dough is baked, the small amount of alcohol evaporates, the yeast is killed, and the carbon dioxide bubbles give the bread a light, spongy structure. Some bacteria release energy through lactic acid fermentation. These bacteria convert the sugar found in milk into lactic acid and are used to make yogurt, cheese, and sourdough bread. Lactic acid changes the acidity of a bread mixture to give it a slightly sour flavor. In yogurt and cheese, the buildup of lactic acid causes the milk to partially solidify, producing the creamy texture of yogurt. If fermentation continues for a long time, the milk eventually turns into cheese. Energy and exercise. Your muscle cells, like some organisms, are able to release energy by both cellular respiration and fermentation. While you are at rest, your muscle cells use specialized molecules to store both energy and oxygen. During hard or prolonged exercise, your muscle cells may use up all their stores of energy and oxygen. Then your muscle cells rely on fermentation to break down sugars. There is much less energy available to cells that use fermentation, which is why you cannot continue to run rapidly for long distances. When your cells use fermentation to release energy, one of the waste products is lactic acid, which can cause a burning sensation in your muscles. When you stop after this type of exercise, your muscles continue to hurt, and you continue to breathe hard for many minutes. During this time, your muscles are playing catch-up. They use the oxygen brought into your blood by your heavy breathing to finish breaking down the byproducts of fermentation. As the lactic acid is converted into carbon dioxide and water, the burning sensation in your muscles goes away. Your muscles build back up their stores of energy and oxygen until the next time they are needed. 2.2 Review. Key concepts. 1. Which form of energy is especially important for living things? Why? 2. How is photosynthesis important to life on Earth? 3. What starting materials do cells need for cellular respiration? Critical thinking. 4. Compare and contrast. How are photosynthesis and cellular respiration similar? How are they different? 5. Predict. Suppose that in a lab, you could remove all the oxygen from a terrarium. What would happen to the plants? Why? Challenge. 6. Synthesize. In everyday language, the word respiration refers to breathing. How is breathing related to cellular respiration? Hint: the air we breathe out contains more carbon dioxide than the air we breathe in. 2.3. Key concept: materials move across the cell's membranes. Before you learned, all cells have an outer covering called the cell membrane. Cells need starting materials for life-sustaining processes. Cells need to get rid of waste products. Now you will learn how materials move into and out of the cell through the cell membrane, how energy is involved in transporting some materials into and out of cells, how surface area affects transport in cells. Some materials move by diffusion. When you walk toward the shampoo section in a store, you can probably smell a fragrance even before you get close. The process by which the scent spreads through the air is an example of diffusion. Diffusion is the process by which molecules spread out or move from areas where there are many of them to areas where there are fewer of them. Diffusion occurs because the molecules in gases, liquids, and even solids are in constant motion in all directions. This random movement of molecules tends to spread molecules out until they are evenly distributed. But diffusion does more than just spread a scent around a room. Cells use diffusion to carry out important life functions. Diffusion helps cells maintain conditions necessary for life. For example, the oxygen needed for respiration enters cells by diffusion. Similarly, the carbon dioxide produced by respiration leaves cells by diffusion. Concentration. Diffusion occurs naturally as particles move from an area of higher concentration to an area of lower concentration. The concentration of a substance is the number of particles of that substance in a specific volume. For example, if you dissolved 9 grams of sugar in 1 liter of water, the concentration of the sugar solution would be 9 grams per liter. When there is a difference of concentration of a substance between two areas, diffusion occurs. Generally, the greater the difference in concentration between two areas, the more rapidly diffusion occurs. As the difference of concentration decreases, diffusion slows down. The number of particles moving to one area is balanced by the number moving in the other direction. Particles are still moving in all directions, but these movements do not change the concentrations. Check your reading. Summarize what happens during diffusion. Remember, a summary includes only the most important information. Diffusion in cells. Diffusion is one way by which materials move in and out of cells. Small molecules, such as oxygen, can pass through tiny gaps in the cell membrane by diffusion. For example, consider the conditions that result from photosynthesis in a leaf cell. Photosynthesis produces oxygen inside the cell. The concentration of oxygen molecules becomes higher inside the cell than outside. Oxygen molecules move out of the cell by diffusion. In a plant cell, some of the oxygen produced by photosynthesis is used in cellular respiration. The remaining oxygen diffuses out of the cell. Much of it escapes to the air. Some of it diffuses to other cells where there is a lower concentration of oxygen. This process of diffusion continues from one cell to the next. Diffusion is a form of passive transport. In passive transport, materials move without using the cell's energy. Cells benefit from passive transport because some materials can move through various cell membranes without any input of energy. Whether or not a substance can diffuse across a cell membrane depends on how well the substance dissolves in the lipids that make up the cell membrane. A special form of passive transport allows polar substances, such as glucose, salts, and amino acids, to pass through cell membranes. All cells need the food energy supplied by glucose, yet glucose is produced in just some plant cells. Polar substances move into the cell through protein channels, or openings in their membranes, that are specific for each substance. This type of diffusion is still passive transport because it uses no energy. Check your reading. What is passive transport? Your answers should mention energy. Osmosis. You have read about the importance of water. Water molecules move through cell membranes by diffusion. The diffusion of water through a membrane is given a special name, osmosis. If the concentration of water is higher outside a cell than inside, water moves into the cell. If the concentration of water is lower outside a cell, water moves out of the cell. You can easily observe the effect of osmosis on plants. If you forget to water a plant, it wilts. Why? The soil dries out, and the plant's roots have no water to absorb. As a result, water leaves the plant cells by osmosis, and they shrink. If you water the plant, water becomes available to enter the shrunken cells by osmosis. The leaves will return to normal as water moves into the cells. Some transport requires energy. Not all materials that move in and out of a cell can do so by diffusion. For cells to carry out life functions, materials must often move from areas of low concentration into areas of high concentration. This process of moving materials against a concentration requires energy. Active transport. Active transport is the process of using energy to move materials through a membrane. This process is different from diffusion and other types of passive transport, which do not require energy. Check your reading. How is active transport different from passive transport? Cells use active transport to perform important life functions, including the removal of excess salt from the body. Consider the example of active transport in marine iguanas shown below. These lizards swim and feed in the salty ocean. As a result, they soak up a lot of salt. Too much salt would seriously damage the iguana's cells, so the cells must get rid of the excess. The solution to the marine iguana's salt problem is found in two small glands above its eyes. Cells in these glands remove excess salt from the blood by active transport. Even when cells in these glands have a higher concentration of salt than that of the blood, the cells use chemical energy to continue taking salt out of the blood. The gland forms a droplet of salt, which the iguana easily blows out through its nostrils. You may not be able to blow salt out of your nostrils, but your kidneys help to keep healthy salt levels in your body. Kidneys filter wastes from your blood by active transport. Cells in the kidneys remove excess salt from the blood. Endocytosis. Cells also need to move materials that are too large to go through the cell membrane or a protein channel. As the diagram below illustrates, endocytosis occurs when a large bit of material is captured within a pocket of the membrane. This pocket breaks off and forms a package that moves into the cell. Cells in your body can use endocytosis to fight bacteria and viruses by absorbing them. 1. As a particle approaches, the cell membrane folds inward, creating a pocket. 2. The particle moves into the pocket, and the membrane closes around it, forming a package. 3. The package breaks away from the cell membrane, bringing the particle into the cell. Exocytosis. When a cell needs to get rid of large materials, the process of endocytosis is reversed. In exocytosis, a membrane within the cell encloses the material that needs to be removed. This package moves to the cell membrane, joins with it, and the material is expelled. Cells often use exocytosis to flush out waste materials, or to expel proteins or hormones made by the cell. 1. A membrane-enclosed package carries materials from inside the cell to the cell membrane. 2. The membrane of the package attaches to the cell membrane, and the two membranes merge. 3. The materials are pushed out of the cell as the membrane of the package becomes part of the cell membrane. Cell size affects transport. Most cells are very small. In fact, most cells are too small to be seen without a microscope. The average cell in your body is about 50 micrometers, 0.05 of a millimeter, in diameter. Most of the cells on this planet are bacteria, which are only 3 to 5 micrometers in diameter. How can something as important as a cell be so tiny? Actually, if cells were not so small, they could never do their jobs. Everything the cell needs or has to get rid of has to go through the cell membrane. The amount of cell membrane limits the ability of cells to either get substances from the outside or transport waste and other materials to the outside. This ability is related to surface area. The relationship between surface area and volume controls cell size. As a cell gets larger, its volume increases faster than its surface area if a cell maintains the same shape. Why does this matter? As the cell gets bigger, there comes a time when its surface area is not large enough to allow resources to travel to all parts of the cell. So the cell stops growing. Bird eggs and frog eggs are much larger than typical cells, but they have a storehouse of food and also rapidly divide to give rise to multicellular embryos. In fact, this multicellular embryo is a good illustration of another way cells get around the surface area to volume problem. They divide. The ratio of surface area to volume in newly divided cells is much higher, giving more surface area for exchanging materials with the outside of cells. A cell's shape also affects its surface area. For example, some single-celled organisms are thin and flat, providing increased surface area. Other cells, such as nerve cells and muscle cells, are long and skinny, which also gives them a higher ratio of surface area to volume. 2.3 Review. Key concepts. 1. How are the processes of diffusion and osmosis alike? 2. What is the difference between active and passive transport? Use the term energy in your answer. 3. How does the surface area of a cell limit the growth of the cell? Critical thinking. 4. Apply. If you put a bouquet of carnations in water, through what process does the water enter the stems? 5. Predict. If a marine iguana were to spend a few days in a freshwater tank, would it continue to blow salt droplets from its nostrils? Why or why not? Challenge. 6. Predict. Freshwater protozoa, which are unicellular organisms, have a greater concentration of salt inside them than does the surrounding water. Does water diffuse into or out of the protozoa?
Chapter 3. Cell Division. The Big Idea. Organisms grow, reproduce, and maintain themselves through cell division. Key concepts. Section 3.1. Cell division occurs in all organisms. Learn about the functions of cell division. Section 3.2. Cell division is part of the cell cycle. Learn about the cell cycle and the process of mitosis. Section 3.3. Both sexual and asexual reproduction involves cell division. Learn how sexual reproduction compares with asexual reproduction. 3.1. Key concept. Cell division occurs in all organisms. Before, you learned cells come from other cells. Cells take in and release energy and materials. In a multicellular organism, some cells specialize. Now you will learn how genetic material is organized in cells. About the functions of cell division in multicellular organisms. Cell division is involved in many functions. Cell division occurs in all organisms, but performs different functions. Unicellular organisms reproduce through cell division. In multicellular organisms, cell division is involved in growth, development, and repair, as well as in reproduction. You're probably bigger this year than you were last year. One characteristic of all living things is that they grow. Your body is made up of cells. Although cells themselves grow, most growth in multicellular organisms occurs when cells dividing produce new cells. In this chapter, you will read about cell division in eukaryotic cells. The genetic material of eukaryotic cells is organized in chromosomes. The genetic material of a cell contains information needed for the cell's growth and other activities. When a cell divides into two new cells, each new cell receives a full set of genetic material. The genetic material in cells is contained in DNA molecules. DNA. The genetic material in cells is DNA, deoxyribonucleic acid. DNA is a chemical that contains information for an organism's growth and functions. You read in Chapter 1 that James Watson and Francis Crick worked with other scientists to build a model of DNA in 1953. They showed that DNA is made of two strands of molecules joined in a structure that resembles a twisted ladder or a double helix. You will learn more about DNA later in the unit. Check your reading. What is DNA? Chromosomes. In a eukaryotic cell, most of the cell's DNA is in the nucleus. During most of the cell's life cycle, DNA exists as a mass of loose strands. While the DNA is spread throughout the nucleus, the cell performs the functions needed for survival. During this time, the DNA is duplicated or copied. DNA is wrapped around proteins like thread around a spool and compacted into structures called chromosomes. Before division, the chromosomes compact more and become visible under a light microscope. During division, a duplicated chromosome can be seen as two identical structures called chromatids that are held together by a centromere. Within each species of organism, the number of chromosomes is constant. For example, humans have 46 chromosomes. Fruit flies, however, have 8 chromosomes, and corn plants have 20. Check your reading. Describe the relationship between DNA and chromosomes. Cell division is involved in growth, development, and repair. Multicellular organisms vary greatly in size and complexity. You may not think that you have much in common with an ant or an oak tree. Actually, you share many characteristics with these organisms. One of the most important characteristics is that both you and they are made of trillions of cells. But like most organisms, you and they started out as single cells. In multicellular organisms, cell division is essential for 3 major functions: growth, development, and repair. Through cell division, a single cell becomes 2 cells. Those 2 cells divide into 4, and the 4 cells divide into 8, and so on. A multicellular organism grows because cell division increases the number of cells in it. As the organism develops and its cells divide, many of the cells become specialized, and most of them continue to divide. Even when growth and development appear to stop, cell division is still occurring. When an organism ages or is injured, the worn-out or damaged cells need to be replaced by new cells formed when healthy cells divide. For example, the cells that make up the lining of your throat have a short lifespan, 2 to 3 days. Living throat cells are constantly dividing and replacing the cells that have died. Growth. In general, a large organism does not have larger cells than a small organism. It simply has many more cells than the small organism. When you were small, your body contained fewer cells than it has now. By the time you reach adulthood, your body will be made up of about 100 trillion cells. Individual cells grow in size, but there are limits to the size that cells can reach. As you learned in Chapter 2, cells need a high ratio of surface area to volume in order to function. As a cell grows, that ratio decreases. When the cell divides into two smaller cells, the ratio of surface area to volume for each cell increases. Scientists are still searching for answers about how cell size is related to the control of cell division. Some scientists think that there is no single factor that controls cell division. Instead, they think that many cell processes added together control when a cell divides. Check your reading. Describe how the number of cells in a multicellular organism changes as the organism grows. Development. Although multicellular organisms begin as single cells, they grow into larger organisms through cell division. However, cell division alone does not allow organisms to develop. If cell division were the only process occurring in cells, all multicellular organisms would end up as spheres of identical cells. But during development, cells become specialized to perform particular functions. These cells may take on shapes or structures that help them to perform their functions. Some cells might become layered skin cells, while others might become long, thin nerve cells. These cells still have the same set of genetic material as all the other cells in an organism's body. But as the organism develops, they specialize. Check your reading. Give 2 examples of specialized cells from the paragraph above. Repair. You may have cut yourself at one time or another. Perhaps you have even broken a bone in your arm or leg. The body repairs injuries like these by means of cell division. For example, when your skin is cut, skin cells on either side of the cut make new cells to heal the wound. You can see the process of healing in the diagram below. 1. Cells in the lower layer begin to divide quickly and move into the break. 2. New cells begin to fill the area as cells continue to divide. 3. Cells stop their rapid dividing once the break is filled. As cells age and die, they need to be replaced. In the human body, which is made up of about 200 different types of cells, cells are replaced at different rates. Your skin cells wear out quickly, so they need to be replaced often. Every minute or so, your skin loses about 40,000 cells, which are replaced with new ones. In contrast, most of the cells in your brain live a long time and do not divide very often. Check your reading. What role does cell division play in healing the body? 3.1. Review. Key concepts. 1. Why is cell division important? 2. How is genetic material organized in eukaryotic cells? 3. Explain how cell division is involved in the growth, development, and repair of an organism. Critical thinking. 4. Summarize. Explain how DNA compacts before a eukaryotic cell divides. 5. Infer. Why do you think that injuries to the skin generally heal faster than injuries to the brain? Challenge. 6. Apply. Describe the stages of development in a multicellular organism that is familiar to you. Connecting sciences. Life science and physical science. Chemical dyes show nerve growth. For years, the medical community has agreed that nerve tissue, once damaged, does not repair itself. In fact, the opinion was that new nerve cells didn't grow in adults at all. However, a surprising discovery has shown that new nerve cells do grow in the mature brains of both monkeys and people. The discovery. The discovery involves a chemical known as bromodioxyuridine, BRDU, which can be used to detect new cancer cells. 1. BRDU highlights the DNA of cells that are reproducing, such as cancer cells. 2. BRDU also makes it possible to count the new cells that are being created, because they stand out as well. 3. The cells that have been highlighted with BRDU can be seen under a microscope when they are illuminated with a special light. When scientists used this technique to examine certain areas in the brains of monkeys and of adult humans who had died of cancer, they found that new nerve cells had grown in the brains of each. Thus, the chemical properties of BRDU allowed scientists to discover new nerve cells growing in places where scientists had previously never expected to see them. Hope for the future. If new nerve cells grow in these tissues, it may be possible to stimulate growth in damaged nerve tissue such as that in the spinal cord. If researchers discover how new growth in nerve cells is stimulated, there may be new hope for people who have nervous systems damaged by accidents or by diseases such as Parkinson's disease. On your own. 1. Synthesize. How could you use chemicals, such as small dots with a pen, in an experiment to show how your fingernails grow? 2. Challenge. What are some possible effects of being able to grow new nerve cells? 3.2. Key concept. Cell division is part of the cell cycle. Before, you learned cells come from other cells through cell division. A cell must have a full set of genetic material to function. Cell division enables multicellular organisms to develop, grow, and repair themselves. Now you will learn about two main stages in the cell cycle, about the changes that occur in cells before mitosis, about the events that take place during mitosis. Think about, what is a cycle? Many things in your everyday life are cycles. A cycle is any activity or set of events that regularly repeats. Cycles can be short, like the sequence of events that make your heart beat, or they can be very long, like the turning of our galaxy. One example of a cycle is shown at the right. The photographs show a tree during 4 seasons in a northern climate. How are these seasons a cycle? The cell cycle includes interphase and cell division. All living things live, grow, reproduce, and die in a process called a life cycle. The life cycle of a tree, for example, begins with a seed. Under the right conditions, the seed begins to grow. It produces a very small plant, which may grow over many years into a towering tree. When it is mature, the tree makes its own seeds, and the cycle begins again. Cells have a life cycle too, called the cell cycle. The cell cycle is the normal sequence of development and division of a cell. The cell cycle consists of 2 main phases, one in which the cell carries out its functions, called interphase, and one in which the cell divides, which can include mitosis and cytokinesis. All cells divide, but only eukaryotes undergo mitosis. Each phase in the cell cycle requires a certain period of time, from hours to days or years, depending on the type of cell. Interphase. Interphase is the part of the cell cycle during which a cell is not dividing. Much activity takes place in this phase of the cell's life. During interphase, the cell grows to about twice the size it was when it was first produced. The cell also engages in normal life activities, such as transporting materials in and transporting wastes out. Also, cellular respiration occurs, which provides the energy the cell needs. Changes that occur during interphase prepare a cell for division. Before a cell can divide, it duplicates its DNA exactly. Correct copying of the DNA is very important. It ensures that after cell division, each new cell gets a complete set of DNA. Check your reading. What cell processes occur during interphase? Cell division phase. Mitosis is the part of the cell cycle during which the nucleus divides. Prokaryotes do not undergo mitosis because they have no nucleus. In most cells, mitosis is the shortest period in the life cycle. The function of mitosis is to move the DNA and other material in the parent cell into position for cell division. When the cell divides, each new cell gets a full set of DNA and other cell structures. Cytokinesis is the division of the parent cell's cytoplasm. Cytokinesis occurs immediately after mitosis. As a result of mitosis and cytokinesis, the original or parent cell splits into two genetically identical daughter cells. In this case, the term daughter cell does not imply gender. It is a term scientists use to refer to these new cells. Each daughter cell receives a complete set of DNA from the parent cell. Cell division produces two genetically identical cells. Recall that many cells in your body are continually dividing into new cells. The new cells help your body grow, develop, repair itself, and replace worn-out parts. Though your body cells divide at different rates, the same process, mitosis, divides their genetic material. Cell division produces daughter cells that are genetically identical to each other, as well as to their parent cell, which no longer exists. Being genetically identical to their parent cell helps the new cells function properly. A skin cell, for example, divides and produces skin cells genetically identical to it. Check your reading. How are daughter cells like the parent cell? Steps of mitosis. The process of mitosis is essential in evenly dividing the genetic material between the daughter cells. Although mitosis is a continuous process, scientists divide the events of mitosis into 4 phases. 1. Chromosomes form. During prophase, the DNA in the nucleus of a cell condenses and becomes visible under a light microscope. Each chromosome consists of 2 identical chromatids held together by a centromere. The membrane around the nucleus disappears. 2. Chromosomes line up. The chromosomes line up in the middle of the cell. This stage is called metaphase. 3. Chromosomes separate. During the stage called anaphase, the chromatids split, resulting in 2 separate identical chromosomes. These chromosomes are pulled to opposite sides of the cell. 4. Nuclei form. A new nuclear membrane forms around each group of chromosomes during telophase. The chromosomes return to their thread-like form. Mitosis is finished, and the cell's genetic material has been divided. Following telophase, the parent cell's cytoplasm is divided to complete the parent cell's division into 2 entirely separate daughter cells. Division of the cytoplasm. Cytokinesis, or the division of the parent cell's cytoplasm, immediately follows mitosis in eukaryotic cells. Cytokinesis differs slightly in animal cells and plant cells. During cytokinesis in an animal cell, a fiber ring forms in the center of the dividing cell. The fiber ring contracts, pulling the cell membrane inward. Eventually, the cell is pinched into 2 daughter cells. In a plant cell, the cell wall prevents the cell membrane from being pulled inward. A structure called a cell plate grows between the 2 new nuclei. The cell plate develops into a membrane and eventually becomes part of the cell wall of each of the new cells. Check your reading. How does cytokinesis differ in plant cells and animal cells? The 2 daughter cells are now completely separated. Each is surrounded by a cell membrane. Each daughter cell has some of its parent cell's cytoplasm. Though daughter cells are genetically identical to their parent cell, they are smaller. After division, cells may enter a period of growth, during which they take in the resources they need to increase the amount of their cytoplasm and to grow to full size. When cells are fully grown, they are about the same size as the parent cell was before division. Check your reading. What happens to cells after cytokinesis? 3.2. Review. Key concepts. 1. What are the 2 main parts of the cell cycle? 2. Describe the state of a cell about to start mitosis. 3. How is the genetic material in 2 daughter cells similar to the genetic material in a parent cell? Critical thinking. 4. Sequence. Describe in order the steps that occur during mitosis. 5. Compare and contrast. How is cytokinesis in plant cells similar to cytokinesis in animal cells? How is it different? Challenge. 6. Infer. You know that mitosis does not happen in prokaryotes. Do you think cytokinesis happens in prokaryotes? Explain your answer. 3.3. Key concept. Both sexual and asexual reproduction involve cell division. Before, you learned cells go through a cycle of growth and division. Mitosis produces two genetically identical cells. Now you will learn about cell division and asexual reproduction. How sexual reproduction and asexual reproduction compare. Think about, how does cell division affect single-celled organisms? In multicellular organisms, cell division functions in growth, repair, and development. But in unicellular organisms, each cell is itself an organism. Unicellular organisms, like this paramecium, also undergo cell division. What are some possible results of cell division in unicellular organisms? How might they compare with the results of cell division in multicellular organisms? Asexual reproduction involves one parent. Mitosis and cytokinesis are the processes by which eukaryotic cells divide. In multicellular organisms, the daughter cells that result are separate cells, but do not live independent lives. For example, new skin cells are part of skin tissue and cannot live independently. In multicellular organisms, mitosis and cytokinesis are not considered methods by which an organism reproduces. Most unicellular organisms and a few multicellular organisms use cell division to reproduce in a process called asexual reproduction. In asexual reproduction, one organism produces one or more new organisms that are identical to itself and that live independently of it. The organism that produces the new organism or organisms is the parent. Each new organism is an offspring. The offspring produced by asexual reproduction are genetically identical to their parents. Cell division in unicellular organisms. Cell division and reproduction are the same thing in all single-celled organisms. However, the process of cell division in prokaryotes and in single-celled eukaryotes differs. Binary fission is the form of asexual reproduction occurring in prokaryotes. Binary fission occurs when the parent organism splits in two, producing two completely independent daughter cells. Genetically, the daughter cells are exactly like the parent cell. Since all prokaryotic organisms are single-celled, cell division and reproduction by binary fission are the same process for them. In single-celled eukaryotic organisms, however, reproduction by cell division involves mitosis and cytokinesis. The unicellular organism undergoes mitosis, duplicating and separating its chromosomes. Then its cytoplasm is divided through cytokinesis. The result is two separate, independent, and genetically identical offspring. Examples of single-celled eukaryotic organisms that reproduce by cell division include algae, some yeasts, and protozoans such as paramecium. Budding. Both unicellular and multicellular organisms can reproduce by budding. Budding is a process in which an organism develops tiny buds on its body. Each bud forms from the parent's cells, so the bud's genetic material is the same as the parent's. The bud grows until it forms a complete or nearly complete new organism that is genetically identical to the parent. In some budding organisms, buds can form from any part of the body. In other organisms, buds can be produced only by specialized cells in particular parts of the body. A new organism produced by budding may remain attached to its parent. Most often, when a bud reaches a certain size, it breaks free of the parent and becomes a separate, independent organism. Some yeast and single-celled organisms reproduce asexually by budding. But budding is most notable in multicellular organisms. Hydras are freshwater animals that are famous for reproducing by budding. Among plants, the kalanchoe produces tiny buds from the tips of its leaves. Each kalanchoe bud that lands on a suitable growing surface will develop into a mature kalanchoe plant that is genetically identical to the parent plant. Check your reading. How is budding different in unicellular and multicellular organisms? Regeneration. In certain multicellular organisms, specialized cells at the site of a wound or lost limb are able to become different types of tissues. The process of new tissue growth at these sites is called regeneration. Although one function of regeneration is the regrowth of damaged or missing body parts, in some organisms, asexual reproduction is another function of regeneration. Regeneration can be observed in many animals called starfish. If a starfish is cut in half, each half can regenerate its missing body parts from its own cells. The result is two complete, independent, and genetically identical starfish. Sometimes a starfish will drop off one of its limbs. The animal will eventually form a new limb. In these cases, regeneration is considered a form of asexual reproduction. The growth of plants from cuttings is also a kind of asexual reproduction through regeneration. Cells near a cut made in a plant's stem begin to produce the missing part of the plant. Once the missing part is grown, the cutting can be planted in soil. The cutting will grow into a new independent plant that is genetically identical to the plant from which the cutting was taken. Check your reading. Describe the process of regeneration in starfish. Asexual reproduction and health. You have probably had the following experience. In the morning, you feel fine. By afternoon, you have a strange feeling that something is not quite right, but you are well enough to function normally. You may even continue to feel well at dinner, and you eat heartily. Then later that evening, it hits you. You're sick. That tickle in your throat has become a sore throat, requiring a visit to the doctor and antibiotics. How did you get so sick so fast? You could have picked up bacteria in school that morning. Perhaps another student coughed, spreading the bacteria that causes strep throat. A population of bacteria, like populations of other organisms that reproduce asexually through binary fission, increases in number geometrically. 2 cells become 4, which become 8, which become 16, and so on. The reason you get sick so fast is that for many bacteria, the generation time is very short. Generation time is the time it takes for one generation to produce offspring, the next generation. In fact, some types of bacteria can produce a new generation of cells in less than 30 minutes. In about an hour, the number of bacteria can increase to 4 times the starting number. Although all offspring are genetically identical, the rare genetic random change does occasionally occur during cell division. The rapid reproduction rate makes it more likely that some offspring will have a random genetic change that may be beneficial. Sexual reproduction involves 2 parent organisms. Reproduction of multicellular organisms often involves sexual reproduction as well as asexual cell division. The table shows some differences between asexual and sexual reproduction. If you grow a plant from a cutting, the new plant will be identical to the parent. However, plants that grow from seeds contain genetic material from 2 parents. Plants growing from seeds and animals growing from eggs are examples of organisms that reproduce through sexual reproduction. Cell division is part of both sexual and asexual reproduction. The process of mitosis produces cells identical to the parent cells. The diversity of life on Earth is in part possible because of the combining of genetic materials from 2 parents in sexual reproduction. In the next chapter, you will read about cell processes involved in sexual reproduction. Check your reading. List 2 major differences between asexual and sexual reproduction. 3.3 Review. Key concepts. 1. How does binary fission relate to cell division? 2. What is a bud, and where does it form on an organism that reproduces asexually? 3. Compare sexual and asexual reproduction. Critical thinking. 4. Predict. Do you think prokaryotes undergo regeneration? Why or why not? 5. Compare and contrast. How is binary fission in prokaryotic organisms similar to and different from mitosis and cytokinesis in single-celled eukaryotic organisms? Challenge. 6. Synthesize. Some bacteria can exchange pieces of genetic material with one another through a process called conjugation. What effect might this exchange have on the offspring of the bacteria that underwent conjugation?
Chapter 4. Patterns of Heredity. The Big Idea. In sexual reproduction, genes are passed from parents to offspring in predictable patterns. Key concepts. Section 4.1. Living things inherit traits in patterns. Learn about traits and how living things inherit traits from their parents. Section 4.2. Patterns of heredity can be predicted. Learn how math can be used to predict patterns of heredity. Section 4.3. Meiosis is a special form of cell division. Learn about the process of meiosis. 4.1. Key concept. Living things inherit traits in patterns. Before you learned, life comes from life. Cells contain chromosomes. Some organisms reproduce with asexual reproduction. Now you will learn how genes for traits are passed from parent to offspring. About discoveries made by Gregor Mendel. About dominant and recessive alleles. Think about what characteristics might be inherited. Make a list of characteristics you can observe about the girl in the photograph to the right. Perhaps your list includes the fact that she has pale skin or that she can read. Some of these characteristics are qualities or abilities learned or acquired from the environment around her. However, some of the characteristics were probably inherited from her parents. Of the characteristics on your list, which do you think were inherited and which do you think were acquired? Parents and offspring are similar. You are an individual who has a unique combination of characteristics. These characteristics are also known as traits. Many of your traits may resemble those your parents have, including your hair color, eye color, and blood type. These characteristics are called inherited traits. Some traits are acquired, not inherited. An acquired trait is developed during your life. Learned behaviors are one type of acquired trait. For example, your ability to read and write is an acquired trait, a skill you learned. You were not born knowing how to ride a bike, and if you have children, they will not be born knowing how to do it either. They will have to learn the skill just as you did. Some acquired traits are not learned, but result from interaction with the environment. Skin color, for example, has both an inherited component and an environmental one. The skin color of many light-skinned people darkens when they are exposed to the sun. Check your reading. How are inherited traits and acquired traits different? Give one example of each. In this chapter, you will learn about inheritance that happens through sexual reproduction. During sexual reproduction, a cell containing genetic information from the mother and a cell containing genetic information from the father combine into a completely new cell, which becomes the offspring. You will learn more about the mechanics of sexual reproduction in section 4.3. Genes are on chromosome pairs. Inherited traits are controlled by the structures, materials, and processes you learned about in chapters 1 and 2. In turn, these structures, materials, and processes are coded for by genes. A gene is a unit of heredity that occupies a specific location on a chromosome and codes for a particular product. Heredity is a passing of genes from parents to offspring. Individuals inherit their genes from their parents. The genes code for the expression of traits. It is important to understand that an organism does not inherit traits themselves from its parents. It inherits the genes that code for the traits it has. Most traits are not coded for by just one gene. Some characteristics are affected by many genes in complicated ways. We have much to learn about which genes might affect which characteristics. In most eukaryotes, cells contain pairs of chromosomes, with one chromosome of each pair coming from each of two parents. The chromosomes in a pair are called homologs. They resemble each other, having the same size and shape, and carrying genetic information for particular traits. On each homolog are sites where specific genes are located. Let us say, for example, that the gene that determines whether or not a plant is tall is located at place A on a pair of homologs. Though both homologs have the gene for height at site A, the genes may not be identical. They may be variations instead. The various forms of the same gene are called alleles. Thus, the homolog from one parent might have an allele for regular height at site A, while the gene from the other parent might have an allele for short height at site A. The alleles on a pair of homologs may or may not be different. Though any one plant can have only two alleles of a gene, there can be many alleles for a particular gene within a population. Check your reading. What are alleles? Each species has a characteristic number of chromosomes. Chimpanzees have 24 pairs of chromosomes, for a total of 48 chromosomes. Fruit flies have 4 pairs of chromosomes, or 8 in all. Humans have 23 pairs, for a total of 46 chromosomes. Scientists refer to chromosomes by their number. Human chromosomes are numbered 1 through 22. The 23rd pair are the sex chromosomes. In humans, the sex chromosomes are called the X chromosome and the Y chromosome. A human female has 2 X chromosomes, while a human male has 1 X chromosome and 1 Y chromosome. In addition to determining the sex of an offspring, the X and Y chromosomes contain important genes, just as the other numbered chromosomes do. Gregor Mendel made some important discoveries about heredity. The first major experiments investigating heredity were performed by a monk named Gregor Mendel, who lived in Austria during the mid-1800s. Before Mendel became a monk, he attended university and received training in science and mathematics. This training served him well when he began investigating the inheritance of traits among the pea plants in the monastery's garden. Mendel took very detailed notes, carefully recording all the data from his many experiments. He worked with 7 different traits: plant height, flower and pod position, seed shape, seed color, pod shape, pod color, and flower color. He studied each trait separately, always starting with plants that were true breeding for that one particular trait. A true breeding plant is one that will always produce offspring with a particular trait when allowed to self-pollinate. One example. In his experiments with plant height, Mendel took 2 sets of plants, one true breeding for plants of regular height, and the other true breeding for plants of short or dwarf height. 1. Instead of letting the plants self-pollinate as they do naturally, he deliberately paired as parents one plant from each set. Mendel called the plants that resulted from this cross the first generation. All of the plants from this first generation were of regular height. The dwarf height trait seemed to have disappeared entirely. 2. Mendel then let the first generation plants self-pollinate. He called the offspring that resulted from this self-pollination the second generation. About 3/4 of the second-generation plants were of regular height, but about 1/4 were of dwarf height. So the trait that seemed to disappear in the first generation reappeared in the second generation. Mendel's experiments with other traits showed similar patterns. Check your reading. Summarize the patterns shown in Mendel's experiments with plant height. Mendel's conclusions. Mendel drew upon his knowledge of mathematics while analyzing his data in order to suggest a hypothesis that would explain the patterns he observed. Mendel realized that each plant must have 2 factors for each possible trait, 1 factor from each parent. Some traits, such as dwarf height, could be masked. Dwarf height could be seen in the plant only if both the plant's factors were for dwarf height. All of the plants in the first generation had 1 dwarf factor and 1 regular factor. A plant with 1 dwarf height factor and 1 regular height factor would be of regular height, because the regular height factor masks the dwarf height factor. Later experiments allowed Mendel to draw a number of other conclusions about how these factors are distributed. Since the mid-1800s, Mendel's experiments and conclusions have been the basis for most of the scientific thought about heredity. Those things he called factors are what we now call genes and alleles. Check your reading. How many factors or genes does each plant have for each possible trait? Alleles interact to produce traits. The pea plant traits Gregor Mendel chose to study were all controlled by single genes, and each of the genes was on a different chromosome. As you learned earlier, most traits are not controlled by only 1 gene. However, simple examples, such as Mendel's peas, do help us better understand heredity. Phenotype and genotype. What color eyes do you have? The eye color you see when you look in the mirror is your phenotype. An organism's phenotype describes the actual characteristics that can be observed. Your height, the size of your feet, the presence or absence of a fold in your eyelids, all are observable traits and are part of your phenotype. By contrast, the genes that control the development of eye folds are part of your genotype. Genotype is the name for the genes an organism has. Your genotype is not always obvious from your phenotype. If you have eye folds, your genotype definitely contains at least 1 eye fold producing allele. But it may also have 1 allele for no eye folds. Sometimes your genes contain information that is not expressed in your phenotype. Check your reading. Which term describes characteristics that can be observed? Dominant and recessive alleles. The eye fold gene, which controls the development of folds in the eyelids, has 2 alleles, eye folds and no eye folds. If you have even 1 copy of the allele for eye folds, you will have eye folds. This happens because the allele that codes for eye folds is dominant. A dominant allele is one that is expressed in the phenotype even if only 1 copy is present in the genotype. That is, even if the other allele is an alternative form. Suppose your genotype contains a no eye folds allele. The no eye folds allele is recessive. A recessive allele is one that is expressed in the phenotype only when 2 copies of it are present on the homologs. If 1 chromosome in the pair contains a dominant allele and the other contains a recessive allele, the dominant allele will be expressed in your phenotype. If you do not have eye folds, it is because you got 2 no eye folds genes, 1 from each parent. Check your reading. Under what conditions is a recessive allele expressed in an offspring's phenotype? Hair color is determined by multiple genes, can be affected by the environment, and sometimes changes over time. However, in some cases, it has a dominant recessive pattern similar to that of the eye fold gene. As in the family shown at right, parents who both have brown hair can have a blonde child. The allele for brown hair is dominant, So if both parents have alleles for both brown hair and blonde hair, the brown hair allele is more likely to be expressed. Their child, however, could have two blonde hair alleles, one from each parent, and therefore have blonde hair instead of brown. 4.1 Review. Key concepts. 1. Explain the difference between acquired and inherited traits. 2. Describe the conclusions that Mendel drew from his experiments with pea plants. 3. What type of alleles are expressed only if two identical copies exist on the homologs of the offspring? Critical thinking. 4. Compare and contrast. What is the difference between a genotype and a phenotype? 5. Analyze. Explain why a person with an allele for a particular trait may not have a phenotype that shows the trait. Challenge. 6. Apply. In guinea pigs, the allele for black fur is dominant over the allele for brown fur. If you had two parent guinea pigs, each with brown fur, what color fur might the offspring have and why? 4.2 Key concept. Patterns of heredity can be predicted. Before you learned, genes are passed from parents to offspring. Offspring inherit genes in predictable patterns. Now you will learn how Punnett squares can be used to predict patterns of heredity. How ratios and probability can be used to predict patterns of heredity. Punnett squares show possible outcomes for inheritance. Mendel noticed that traits are inherited in patterns. One tool for understanding the patterns of heredity is a graphic called a Punnett square. A Punnett square illustrates how the parents' alleles might combine in offspring. Each parent has two alleles for a particular gene. An offspring receives one allele from each parent. A Punnett square shows how the parents' alleles may be passed on to potential offspring. The Punnett square on page 111 shows how alleles for pea plant height would be distributed among offspring in Mendel's first-generation cross. The dominant allele is regular height, and the recessive allele is dwarf height. The top of the Punnett square shows one parent's alleles for this trait, two dominant regular alleles. The side of the Punnett square shows the other parent's alleles for this trait, two recessive dwarf alleles. Each box in the Punnett square shows a way the alleles from each parent would combine in potential offspring. You can see that each potential offspring would have the same genotype, one dominant and one recessive allele. The phenotype of each offspring would show the dominant allele, in this case, regular height. Check your reading. What is a Punnett square? Ratios and percentages can express the probability of outcomes. The Punnett square on page 111 for the first generation of pea plants shows that all potential offspring would be of regular height, because they all have one dominant allele. You can say that 100% of the offspring will be of regular height. Or you could say that the ratio of regular height offspring to total offspring is 4 to 4. A ratio compares or shows the relationship between two quantities. A ratio is usually written 4:4 and read as 4 to 4. This can be interpreted as 4 out of 4. The Punnett square shows that 4 out of 4 offspring will express the dominant gene for regular height. Punnett squares and the ratios they show express probability. Probability is the likelihood or chance of a specific outcome in relation to the total number of possible outcomes. The ratios derived from a Punnett square tell you the probability that any one offspring will get certain genes and express a certain trait. Another way of expressing probability is as a percentage. A percentage is a ratio that compares a number to 100. That is, it states the number of times a particular outcome might happen out of 100 chances. Check your reading. What are two ways that you can express a probability? Look at the guinea pig Punnett square on page 113. This cross is between two parents, each with one dominant allele, black, and one recessive allele, brown, for the trait fur color. In this cross, only 1 in 4, ratio 1:4, offspring gets two dominant alleles. That is, there is a 1 in 4 chance that an offspring from this cross will have two dominant alleles for black fur. The likelihood that the offspring will get one dominant and one recessive allele is 2:4. 2 out of every 4 offspring would have this genotype. Like the one offspring with two dominant alleles, the two offspring with the genotype capital B, lowercase b, will have black fur. This makes a total of 3 offspring, 3:4, with the phenotype black fur. Only 1 out of 4 offspring of this cross will have the genotype and phenotype brown fur. When one parent has two dominant alleles and the other has two recessive alleles, there is a 100% chance that an offspring will have the dominant phenotype. The pea plant example on page 111 shows this pattern. All of the offspring are of regular height. When both parents have one dominant and one recessive allele, there is a 75% chance that an offspring will have the dominant phenotype. The guinea pig example on page 113 shows this pattern. Chances are that more offspring will have black fur than brown fur. Check your reading. What is the probability that an offspring from the pea plant cross on page 111 will be of dwarf height? In humans, females have 2 X chromosomes, and males have an X and a Y chromosome. The Punnett square on page 115 shows the possible sexes of human offspring. Unlike the guinea pig Punnett square, this one shows only 2 possible outcomes, XX and XY. The diagram also shows how to find the percentage chance that a potential offspring will be female. It is important to realize that Punnett squares and probability do not guarantee the outcome of a genetic cross. They indicate the probability of different outcomes. While there is a 75% chance that an offspring will have black fur, according to the Punnett square on page 113, you cannot know with any certainty what color fur a particular offspring will actually have. Actual experimental results may not match predicted outcomes. Check your reading. Can a Punnett square tell you the specific outcome of a genetic cross? Why or why not? 4.2 Review. Key concepts. 1. Explain how Punnett squares predict the outcomes of a genetic cross. 2. How are ratios and percentages related? 3. How can you find a percentage chance from a Punnett square? Critical thinking. 4. Predict. Mendel studied the colors of flowers in his experiments with pea plants. Let capital P stand for purple and lowercase p stand for white. Purple is dominant. Make a Punnett square for a cross between 2 capital P lowercase p plants. Find the percentage chance for each outcome. Challenge. 5. Apply. In pea plants, the allele for smooth peas is dominant over the allele for wrinkled peas. Create a Punnett square and calculate the probability that 2 smooth pea plants will have an offspring with wrinkled peas if each parent has 1 smooth and 1 wrinkled allele. 4.3 Key concept. Meiosis is a special form of cell division. Before you learned, mitosis produces 2 genetically identical cells. In sexual reproduction, offspring inherit traits from both parents. Genetic traits are inherited in predictable patterns. Now you will learn why meiosis is necessary for sexual reproduction, how cells and chromosomes divide during meiosis, how meiosis differs from mitosis. Meiosis is necessary for sexual reproduction. In section 4.1, you learned that 2 cells combine during the process of sexual reproduction. One of the cells contains genetic information from the mother. The other contains genetic information from the father. The two cells combine into a completely new cell, which becomes the offspring. Check your reading. How does the genetic material of offspring produced by sexual reproduction compare with the genetic material of the parents? Most human cells, which can be referred to as body cells, contain 46 chromosomes, the full number of chromosomes that is normal for a human being. Any cell that contains the full number of chromosomes, 2 sets, for a species, is a 2N cell, also called a diploid cell. The 2N cells for a fruit fly, for example, contain 8 chromosomes. Think about what would happen if 2 body cells were to combine. The resulting cell would have twice the normal number of chromosomes. Reproductive cells, called gametes, differ from body cells. Gametes are cells that contain half the usual number of chromosomes, 1 chromosome from each pair. Gametes are 1N cells, also called haploid cells. Human gametes contain 23 unpaired chromosomes. The gametes of a fruit fly contain 4 unpaired chromosomes. Gametes are found only in the reproductive organs of plants and animals. An egg is a gamete that forms in the reproductive organs of a female. A gamete that forms in the reproductive organs of a male is a sperm. Check your reading. What is a gamete? During sexual reproduction, 2 gametes combine to become a 2N cell that can grow into a new offspring. Fertilization is the process that takes place when a sperm and an egg combine to form one new cell. The diagram below shows what happens to the chromosomes in gametes during fertilization. In humans, an egg cell with 23 chromosomes joins a sperm cell with 23 chromosomes to form a new 2N cell with 46 chromosomes. You know that body cells divide by the process called mitosis. Mitosis produces 2 daughter cells, each containing exact copies of the chromosomes in the parent cell. Each daughter cell formed by mitosis is a standard diploid, 2N cell. But to produce gametes, which are haploid, a different kind of cell division is necessary. Meiosis is a special kind of cell division that produces haploid, 1N cells. During meiosis, a single cell goes through 2 cell divisions, meiosis I and meiosis II. Meiosis takes place only in the reproductive tissues of an organism. Cells divide twice during meiosis. Before meiosis begins, the chromosomes of the parent cell are copied. A cell that is ready to divide contains 2 copies of each chromosome pair, twice as many chromosomes as usual. So to end up with cells that have half the usual number of chromosomes, there must be 2 divisions. Remember that the 2 chromosomes in a pair are called homologs. At the beginning of meiosis I, the cell has 2 copies of each homolog attached together. During meiosis I, the homologs separate. The starting cell divides into 2 cells. One cell contains the 2 copies of one homolog of each pair, while the other cell contains the 2 copies of the other homolog of each pair. Then, during meiosis II, each of the 2 cells is divided, producing 4 haploid cells. Each haploid cell has 1 unpaired set of chromosomes. Meiosis I. As you can see in the diagram on page 121, there are 4 steps in meiosis I: prophase I, metaphase I, anaphase I, and telophase I. Included in telophase I is a cytokinesis, the division of the cytoplasm. The diagram shows what would happen during meiosis I in a species that has 4 chromosomes in its 2N body cells. 1. Prophase I. Chromosomes pair up with their partners. There are 2 sets of each of the chromosome pairs in the parent cell. The members of each chromosome pair are attached together in sets of doubled homologs. 2. Metaphase I. Each set of chromosome pairs lines up along the center of the cell. 3. Anaphase I. The 2 copies of one homolog are pulled apart from the 2 copies of the other homolog. This dividing of the homologs is the most significant step of meiosis I. 4. Telophase I and cytokinesis. A new cell membrane forms at the center of the cell, dividing the parent cell into two daughter cells. Check your reading. What happens to the parent cell during telophase I? Meiosis II. During meiosis I, two daughter cells are formed. The chromosomes of these two cells are not copied before meiosis II begins. Both of these cells divide during meiosis II to produce a total of 4 daughter cells. The 4 steps in meiosis II, shown on page 121, are prophase II, metaphase II, anaphase II, and telophase II with cytokinesis. 5. Prophase II. In each daughter cell, there are 2 copies of each of N chromosomes. The copies are attached together. 6. Metaphase II. The chromosomes line up along each cell's center. 7. Anaphase II. The 2 attached copies of each chromosome separate and are pulled to opposite poles in each cell. 8. Telophase II and cytokinesis. A new cell membrane forms in the center of each cell, as each cell divides into 2 1N daughter cells, producing a total of 4 1N cells. During meiosis, one cell in an organism's reproductive system divides twice to form 4 1N cells. In male organisms, these gametes become sperm. In female organisms, at least one of these cells becomes an egg. In some species, including humans, only one of the 4 daughter cells produced by a female during meiosis becomes an egg. The rest dissolve back into the organism, or in some cases, are never produced. Meiosis and mitosis differ in some important ways. You can see that the processes of meiosis and mitosis are similar in many ways. However, they also have several very important differences. Only cells that are to become gametes go through meiosis. All other cells divide by mitosis. A cell that divides by meiosis goes through 2 cell divisions, but the chromosomes are not copied before the second division. In mitosis, the chromosomes are always copied before division. Daughter cells produced by meiosis, which are haploid, 1N, contain only half of the genetic material of the parent cell, 1 homolog from a chromosome pair. Daughter cells produced by mitosis, which are diploid, 2N, contain exactly the same genetic material as the parent, pairs of chromosomes. Check your reading. What are 4 ways in which meiosis differs from mitosis? 4.3 Review. Key concepts. 1. What kind of cell is produced by meiosis? 2. What is fertilization? 3. In your own words, describe the differences between meiosis and mitosis. Critical thinking. 4. Compare. How do prophase I and prophase II differ? 5. Communicate. Make a Venn diagram to show the similarities and differences between mitosis and meiosis. Challenge. 6. Synthesize. Why does meiosis II result in 4 1N cells rather than 4 2N cells? Think science. Skill. Determining relevance. Are traits linked? Fruit flies are easy to breed in a laboratory and have an assortment of easily recognized genetic traits, different eye colors, body patterns, limb characteristics, and wing shapes. For these reasons, early geneticists studied fruit flies to learn how certain traits were inherited. Sometimes the experiments produced puzzling results. Here is an example from the laboratory of Thomas Hunt Morgan. Observations. In a batch of fruit flies, most red-eyed individuals were born with short wings. In the same batch, at least one fruit fly was born with red eyes and normal-sized wings. Hypotheses. Morgan and his co-workers made these hypotheses about the inherited traits. The gene for red eyes and the gene for short wings are linked together on a fruit fly's chromosomes. These linked genes are usually inherited together. Sometimes during meiosis, one of the linked genes will cross over from one chromosome to a homologous one. When this happens, a fruit fly will be born with one but not both of the linked genes, red eyes without short wings. Genes that are farthest from each other on a chromosome are most likely to become separated and cross over during meiosis. Genes that are closest, linked to each other, are least likely to. Further discoveries. By studying the results of many breeding experiments, Morgan and his student, Alfred Sturtevant, could determine which genes were closest and farthest from each other on the same chromosome. From this information, they drew a simple map showing the location of each of the fruit fly's linked genes. Determine relevance. On your own. Look at the map of a chromosome on this page. Which of the traits are most likely to be inherited together? Which might be most easily separated and cross over during meiosis? As a group, is it reasonable to think that information about a fruit fly's genes could apply to the genes of a human being? Discuss this topic in a small group and see if the group can agree. Timelines in Science. The Story of Genetics. The Human Genome Project, DNA Evidence in Criminal Cases, Cloning. News about genetics is everywhere. Some of the most exciting research in science today involves genes. The timeline shows that some important concepts that underline the study of genetics were discovered relatively early. You will notice the influence of two major advances in technology: the development of the microscope during the 1600s and the development of computer technology during the later half of the 1900s. The boxes below the timeline show how technology has led to new understanding and to applications of those understandings. 1665. Cells discovered. Robert Hooke uses a microscope to study living matter. What he sees and then records in this drawing are tiny repeating units, which he calls cells. 1674. Cells are everywhere. Anton von Leeuwenhoek uses a microscope to study pond water and discovers the water is full of microscopic organisms, some made of single cells. These drawings show some of what he saw. 1831. Cells have structure. As the power of microscopes improves, scientists start to describe the inner structure of cells. Robert Brown describes a central structure found in many cells, a structure that he calls the nucleus. 1866. Austrian monk describes patterns of heredity. Gregor Mendel's experiments with garden peas show that traits are passed on from parents to offspring in predictable patterns. 1882. Cells divide, chromosomes split. Scientists observe how cells divide. Walter Fleming describes how structures within the cell separate. These structures are the chromosomes. Chromosomes determine the traits of living things. 1928. Researchers study the chromosomes of fruit flies and find genes. Working with fruit flies, Thomas Hunt Morgan discovers that genes are found in specific locations on chromosomes. Application. Corn in every shape and size. Native Americans grew 700 different kinds of popcorn. Their popcorn plants were a kind of grass with big, hard seeds that exploded when they were heated. People who didn't want to have to explode their corn to eat it chose plants with softer seeds and grew them, and then chose the softest of those seeds to plant. Over hundreds of years, by choosing which plants to grow, people produced what we now eat as corn on the cob. Technology. Seeing into the cell. Single glass lenses, such as the one von Leeuwenhoek used, were available as long ago as 1267. The compound microscope was first made in 1595, but it was over 200 years before it provided clear images. Until the 1930s, all microscopes focused light on objects. Eventually, light microscopes could magnify objects up to 2,000 times. 1944. DNA, genetic material. Researchers studying Streptococcus transformation find that bacterial cells get their characteristics from DNA. 1951. Scientists capture image of DNA. Scientists searching for the secret of DNA structure get an enormous clue when Rosalind Franklin uses X-ray crystallography to create an image of DNA. Maurice Wilkins, James Watson, and Francis Crick are awarded the Nobel Prize in 1962 for building a model of the DNA double helix molecule. 1973. DNA recombined. In an amazing breakthrough, scientists have cut DNA from two different sources and recombined the DNA. The new DNA molecule reproduces when placed inside a bacterium. Such bacteria can be used to make proteins useful to humans. 1984. Chinese scientists alter fish. In an effort to produce fast-growing fish for food, a team working with Zhouyin Zhu has made the first genetically modified GM fish. 1984. Living things have genetic fingerprints. Human fingers have their own unique fingerprints. In a similar way, the DNA of different people has its own unique patterns. These DNA fingerprints are compared here. 2000. Scientists sequence human genome. Two groups of researchers, Celera and the Human Genome Project, succeed in publishing the first draft of the sequence of DNA for all the chromosomes in the human body. Technology. Seeing molecules. In the 1930s, a microscope came into use that focuses a beam of electrons instead of a beam of light on an object. Now we can see things as small as the molecules inside cells. Application. DNA frees innocent prisoner. Kevin Green was convicted of murder and spent 16 years in prison. While he was in jail, the California Department of Justice created a DNA database that contained the DNA fingerprints of many other convicted felons. When Green's defenders compared the DNA found at the murder scene with the DNA fingerprints in the database, they found that it matched someone else's fingerprint. The real murderer confessed, and Green is now a free man, thanks to genetics. Application. Saved by a gene donor. In 1986, a baby girl named Ashanti De Silva was born. One single mistake in her DNA meant that Ashanti's body could not make an important disease-fighting protein. In 1981, researchers had figured out how to move a working gene from one mammal to another. Ashanti became the first person ever to receive a gene from someone else. Ashanti's doctors injected some of her white blood cells with healthy copies of the sick gene. Now her white blood cells worked. Researchers and doctors are trying to apply the same techniques to other genetic disorders. There is still much work to be done. Into the future. Genetics is a young science. The timeline spans 350 years, but the real study of genetics began in 1900 with the rediscovery of the work of Gregor Mendel. Since then, scientists have determined the structure and function of DNA and ways to use this knowledge. In medicine, genetics is used to identify genes that play a role in inherited diseases. Questions remain about how this knowledge can be used to treat or even prevent disease. In agriculture, genetics is used to modify the genes of plants and animal stocks to give them desirable traits, such as resistance to disease. Questions remain about what effect modified genes might have once they enter a population of plants or animals. In biology, genetics is used to determine how different types of organisms have changed over time, and how one species relates to another. Questions remain about whether similar genes found in different organisms behave in the same way. In society, genetic profiles are used to help solve crimes or make identifications. Questions remain about how to protect individuals and their personal information. Activities. Reliving history. Use a hand lens or microscope to study water from a pond or puddle. See if your sample contains structures similar to those drawn by Van Leeuwenhoek in 1674. Writing about science. Biography. Sharing information is important to scientific discovery. Learn more about individuals or groups involved in the discovery of DNA structure or sequencing the human genome. How important was cooperation in their work?
Chapter 5. DNA and Modern Genetics. The Big Idea. DNA is a set of instructions for making cell parts. Key concepts. Section 5.1. DNA and RNA are required to make proteins. Learn about DNA, RNA, and protein synthesis. Section 5.2. Changes in DNA can produce variation. Learn about the effects of changes in DNA and how some changes can cause genetic disorders. Section 5.3. Modern genetics uses DNA technology. Learn about some applications of DNA technology and the Human Genome Project. 5.1. Key concept. DNA and RNA are required to make proteins. Before, you learned traits pass from parents to offspring in predictable patterns. Traits are passed on through genes. In sexual reproduction, offspring get half their genes from each parent. Now you will learn how the structure of DNA stores information the cell needs, how DNA is copied, how RNA uses the information from DNA to make proteins. DNA is the information molecule. DNA is a molecule that stores information. That's all it does. You could compare the information in DNA to the books in your local library. You might find a book describing how to bake a cake, make a model sailboat, or beat your favorite computer game. The books, however, don't actually do any of those things. You do. The books in the DNA library carry all the information that a cell needs to function, to grow, and to divide. However, DNA doesn't do any of those things. Proteins do most of the work of a cell and also make up much of the structure of a cell. Proteins and amino acids. Proteins are large molecules that are made up of chains of amino acids. 20 different amino acids come together in enough combinations to make up the thousands of different proteins found in the human body. Some proteins are small. For example, lysozyme is a digestive protein that is made up of a sequence of 129 amino acids. Some proteins are large. For example, dystrophin is a huge structural protein that is made up of 3,685 amino acids. Check your reading. What is the relationship between proteins and amino acids? DNA stores the information that enables a cell to put together the right sequences of amino acids needed to produce specific proteins. Scientists describe DNA as containing a code. A code is a set of rules and symbols used to carry information. For example, your computer uses a code of ones and zeros to store data and then translates the code into the numbers, letters, and graphics you see on a computer screen. To understand how DNA functions as a code, you first need to learn about the structure of the DNA molecule. DNA and the genetic code. The DNA molecule takes the shape of a double-stranded spiral, which, as you can see from the diagram, looks something like a twisted ladder. In Chapter 2, you read about different subunits that make up the molecules found in cells. Nucleotide subunits make up each of the two strands of the DNA molecule. One part of the nucleotide forms the side rail of the DNA ladder. The other part, the nucleotide base, forms the rung. Actually, two bases come together to form the rung, as one nucleotide base attaches to another from the opposite strand. You can see how the parts fit together in the diagram to the left. There are 4 different nucleotides in DNA, identified by their bases: adenine, A, thymine, T, cytosine, C, and guanine, G. Because of differences in size and shape, adenine always pairs with thymine, AT, and cytosine always pairs with guanine, CG. The bases fit together like two pieces of a jigsaw puzzle. These bases are often referred to simply by their initials, A, T, C, and G. The phrase, "all tigers can growl," may help you remember them. It is the sequence, the order, of bases in a strand of DNA that forms the code for making proteins. Like a list of ingredients in a recipe book, a set of bases specifies the amino acids needed to form a particular protein. The cookbook uses just 4 bases, A, T, G, and C, to code for about 20 amino acids. A code of 2 bases to 1 amino acid gives only 16 possible combinations. However, a code of 3 bases to 1 amino acid gives 64 possible combinations. The genetic code is, in fact, a triplet code. A specific sequence of 3 nucleotide bases codes for 1 amino acid. For example, the triplet TCT on a strand of DNA codes for the amino acid arginine. Some amino acids have 2 different codes, others have 3, and some have 4. A gene is the entire sequence of the bases that codes for all the amino acids in a protein. Each gene is made up of a sequence of bases at a particular location on the DNA. Replication. When a cell divides into 2 cells, each daughter cell receives an identical copy of the DNA. Before a cell divides, all of its DNA is copied, a process referred to as replication. Let's follow the process through for one DNA molecule. First, the 2 strands of DNA separate, almost like 2 threads in a string being unwound. Nucleotides in the area around the DNA match up, base by base, with the nucleotides on each DNA strand. C matches up with G, and A matches up with T. When replication is complete, there are 2 identical DNA molecules. Each molecule has 1 strand of old DNA and 1 strand of new DNA. 1. The parent DNA molecule is made up of 2 strands. 2. The 2 strands separate and are used as templates. 3. 2 new identical DNA molecules are formed. During replication, each strand of DNA is used as a template to produce a copy of the other strand. A template is a pattern or shape that produces a matching or complementary product. If you've ever made a plaster model of your hand, you've worked with a template. You press your hand into a soft material that leaves a mold of your hand. You then pour liquid plaster into the mold to produce a copy of your hand. The mold is a template. Its shape allows you to make a complementary shape that matches your hand. RNA is needed to make proteins. DNA is not used to make proteins directly. Translating the genetic code of DNA involves another type of molecule, RNA. RNA, or ribonucleic acid, carries the information from DNA to a ribosome, where the amino acids are brought together to form a protein. DNA actually codes for RNA. 3 different types of RNA are involved in making proteins. They are named for their functions. Messenger RNA, mRNA. Ribosomal RNA, rRNA. Transfer RNA, tRNA. In prokaryotic cells, RNA and proteins are both made in the cytoplasm. In eukaryotic cells, DNA is copied in the nucleus, then RNA moves to the cytoplasm where the proteins are made. Transcription. The process of transferring information from DNA to RNA is called transcription. The chemical structure of RNA is quite similar to the structure of DNA. Both are made up of 4 types of nucleotide subunits. 3 of the bases that make up RNA are the same as in DNA: guanine, G, cytosine, C, and adenine, A. However, the 4th base is uracil, U, not thymine. During transcription, DNA is again used as a template, this time to make a complementary strand of RNA. Only individual genes are transcribed, not a whole DNA molecule. The DNA again opens up, just where the gene is located. As shown in the diagram above, RNA bases match up to complementary bases on the DNA template. Adenine pairs with uracil, AU, and cytosine pairs with guanine, CG. Transcription is different from replication in some important ways. Only one strand of DNA is transcribed, which means just a single strand of RNA is produced. When transcription is complete, the RNA is released. It does not stay attached to DNA. This means that many copies of RNA can be made from the same gene in a short period of time. At the end of transcription, the DNA molecule closes. Translation. Replication and transcription involve passing along information that is coded in the language of nucleotide bases. To make proteins, cells have to translate this language of nucleotide bases into the language of amino acids. 3 specific bases equal 1 amino acid. The actual assembly of the amino acids in their proper sequence is the translation. Translation takes place in the cytoplasm of a cell. It involves all 3 types of RNA. Proteins are made on ribosomes, structures that are made up of ribosomal RNA and proteins. If you think of DNA as a cookbook for making different proteins, and mRNA as a recipe for making a protein, then the ribosome is the place where the cooking gets done. In this analogy, tRNA gathers the ingredients, which are amino acids. A tRNA molecule is shaped in such a way that one end of it can attach to a specific amino acid. The other end of tRNA has a triplet of bases that is complementary to a triplet of bases on mRNA. Transfer RNA does the actual translation of bases to amino acid when it matches up with mRNA. The diagram on page 140 shows the whole process. 1. Translation begins when a ribosome attaches to the beginning end of an mRNA molecule. 2. A tRNA molecule carrying an amino acid matches up to a complementary triplet on mRNA on the ribosome. 3. The ribosome attaches one amino acid to another as it moves along the mRNA molecule. 4. The tRNA molecules are released after the amino acids they carry are attached to the growing chain of amino acids. 5. The ribosome completes the translation when it reaches the end of the mRNA strand. The newly made protein molecule, in the form of a chain of amino acids, is released. Check your reading. Describe how the 3 different types of RNA work together in protein synthesis. The process of making proteins is basically the same in all cells. The flow of information in a cell goes from DNA to RNA to protein. 5.1. Review. Key concepts. 1. Describe the shape of the DNA molecule and how nucleotide bases fit into that structure. 2. What is a protein and what is it made up of? 3. Identify 3 types of RNA involved in protein synthesis and briefly describe what they do. Critical thinking. 4. Infer. What might happen if the wrong amino acid is put on a tRNA molecule? 5. Apply. Copy the following sequence of DNA bases: A, T, C, A, G, G. Write the complementary mRNA and tRNA sequences for this. Challenge. 6. Synthesize. Study the sequences you wrote for question 5. How does the tRNA sequence compare to the original DNA sequence? 5.2. Key concept. Changes in DNA can produce variation. Before, you learned DNA contains information in the form of a sequence of bases. Genes code for RNA and proteins. DNA is transcribed into RNA, which is used to make proteins. Now you will learn about mutations, any changes in DNA, about the possible effects of mutations, about pedigrees and how they are used. DNA sequences can change. Differences or variations in DNA are what make one organism different from another. The number of differences in the DNA sequences between 2 species is large. Each human cell with its 46 chromosomes contains an astounding 3 billion base pairs in its DNA. A yeast cell, by comparison, has 12 million base pairs in its DNA. The number of differences between any 2 individuals of the same species is small. For example, about 99 9/10% of the DNA in the cells of 2 different humans is the same. Just 0.1% variation in DNA makes you the unique person you are. That averages out to 1 base in 1,000. How can there be such great variety among people if their DNA is so similar? The reason is that of the 6 billion base pairs in human DNA, only 5% are in the genes that code for RNA and proteins. As you learned in Chapter 4, genes and their interaction with the environment are what determine the traits of a person. Differences in genes affect the height of people, or the color of their eyes, hair, or skin. Genes produce variation because the type or amount of the proteins they code for can vary from person to person. For example, skin color comes from a protein called melanin. The amount of melanin an individual produces affects the color of their skin. Given the huge number of base pairs in the DNA of any organism, it is not surprising that errors occur when DNA is copied. DNA is also affected by the environment. For example, exposure to ultraviolet radiation or X-rays can damage DNA. Both natural and human-made toxins, which are harmful chemicals, can also damage DNA. Any change in DNA is called a mutation. Cells have different ways to repair mistakes in a DNA sequence. Certain enzymes actually proofread DNA, for example, correcting mismatched base pairs. Other enzymes enable damaged DNA to be fixed. Check your reading. What is a mutation? When a mutation occurs in a gene, the coding region of DNA, the wrong amino acid might be placed in the amino acid chain. If this happens, there are three possible outcomes. 1. The mutation causes no effect. Since some amino acids have more than one code, a mutation may not change the resulting protein. Also, since each cell has two sets of DNA, even if one gene is not working, enough protein may be produced. 2. The effect of a mutation is minor. A change in the genes that control the amount of melanin produced could affect not only how light or dark a person's skin is, it could also affect eye or hair color. The change in this case is a change in appearance. 3. The effect of a mutation is great. The effect can be good, such as a plant having an increased resistance to disease. Or the effect can be bad, causing a genetic disorder or disease. Remember, only 5% of human DNA is in genes. If a mutation occurs in a non-coding region of DNA, then chances are that the mutation will have no effect. Such a mutation is neutral. Mutations can cause genetic disorders. A genetic disorder is a disease or condition that results from mutations that affect the normal functioning of a cell. Sometimes these disorders are inherited, passed on from parent to offspring. Examples of inherited disorders include Tay-Sachs disease, cystic fibrosis, sickle cell disease, and albinism. Other genetic disorders result from mutations that occur during a person's lifetime. Most cancers fall into this category. Check your reading. What is a genetic disorder? Sometimes a person carries a tendency for a disease, such as diabetes, glaucoma, Alzheimer's disease, or emphysema. In some cases, a person's behavior can help prevent the disease. Cigarette smoke is a leading cause of lung cancer. Smoke also greatly increases the risk of people with a genetic tendency for emphysema to develop that disease. Sickle cell disease is an interesting example of how a mutation can have more than one effect. The mutation occurs in one of the genes that code for hemoglobin. Hemoglobin is a protein that carries oxygen in red blood cells. The mutation causes one amino acid to be replaced with another. Sickle cell disease is a recessive disorder. Only people who carry two recessive alleles are affected. Recall that an allele is one form of a gene. Because of the amino acid change, some red blood cells can take on a sickle shape. See the photograph at the right. The pedigree below shows the pattern of inheritance of the sickle cell allele through 3 generations of a family. A pedigree is a diagram of family relationships that includes 2 or more generations. Sickle cell disease is a severe disease. Sickled red blood cells tend to break more easily than normal red blood cells. People with sickle cell disease do not get enough oxygen delivered to their body tissues, and the tissues become damaged. The disease is common in Africa and parts of India and the Middle East. What is interesting about the sickle cell allele is that it provides protection against dying of malaria. Malaria is a severe disease, also common in Africa, India, and the Middle East. It is caused by microscopic organisms that reproduce in red blood cells. Scientists do not yet completely understand why people with the sickle cell allele are better able to survive malaria. However, the effect of this protection is that the sickle allele remains common in populations that live in regions where malaria is common. Cancer is a genetic disorder that affects the cell cycle. Cancer is not a single genetic disorder, but rather it is a group of disorders. All cancers are characterized by the uncontrolled division of cells. Normally, cells in a multicellular organism function to maintain the health of an organism. Cell division is controlled so that an organism has the number of cells it needs to function. Cancer cells are, in a way, selfish cells. Where normal cells stay within the same tissue, cancer cells spread quickly and can invade other tissues. A normal cell has a definite lifespan. Cancerous cells become immortal. They divide indefinitely. Check your reading. What is a characteristic of all cancers? Most cancers are caused by mutations to DNA that happen during a person's lifetime. Some mutations come from mistakes made during replication, but many are caused by harmful chemicals often referred to as carcinogens. Many plants naturally produce carcinogens in their tissues. Nicotine is a carcinogen naturally found in tobacco leaves. There are other carcinogens in tobacco. Ultraviolet and nuclear radiation, as well as X-rays, can also cause cancer. That is why, if you get an X-ray at the doctor's or dentist's office, the part of your body not being X-rayed is protected by a lead apron. Some people may inherit a tendency for a particular cancer. That does not mean the cancer will occur. Cancer involves a series of mutations. What is inherited is a mutation that is one step in the series. The disease occurs only if other mutations come into play. 5.2. Review. Key concepts. 1. What is a mutation? 2. How do mutations affect an organism? 3. What effect does cancer have on the cell cycle of a cancerous cell? Critical thinking. 4. Infer. A mutation in a triplet code that ends up coding for the same amino acid is referred to as a silent mutation. In what sense is it silent? 5. Provide examples. Identify 3 causes of genetic disorders and give an example of each. Challenge. 6. Analyze. Why are genetic diseases carried by genes on the X chromosome more common in male offspring than female offspring? Hint. Think about how X and Y chromosomes are distributed in males and females. 5.3. Key concept. Modern genetics uses DNA technology. Before, you learned mutations are changes to DNA. Not all mutations have an effect on an organism. Mutations can lead to genetic disorders. Now you will learn how scientists can change organisms by changing DNA. About some applications of DNA technology. About some issues surrounding the use of DNA technology. Think about. What type of animal is this? Look at the photograph of the animal to the right. The cells of this animal contain DNA from 2 different species. For a long time, humans have been able to mix genes by breeding together animals of different but similar species. Now scientists have the technology to mix together genes from 2 very different species by inserting genes from one organism into the cells of another. What do the characteristics of this animal suggest about the source of its genes? Changes in DNA can change an organism. Organisms change over time. Changes come about because of mutations in DNA. Random changes in DNA may introduce new traits into an organism. Over time, certain traits may become more common in one group of organisms as they interact with the environment and each other. Are all changes in a group of organisms random? There are dogs, such as bloodhounds, that are particularly well suited to tracking. There are cows that give large quantities of milk, and crops that produce large quantities of grain. Changes such as these are not random, but result from careful breeding directed by humans. Selective breeding. For thousands of years, humans have been carefully selecting and breeding certain plants and animals that have desirable traits. As the years have passed, horses have gotten faster, pigs have gotten leaner, and corn has become sweeter. Selective breeding is the process of selecting and breeding parent organisms to pass on particular traits to the offspring. Selective breeding can be successful as long as the desirable traits are controlled by genes. In fact, what these early farmers were actually selecting were alleles, particular versions of a gene. The alleles were already present in some members of the population. People were not changing DNA, but they were causing certain alleles to become more common in a particular breed. The different dog breeds are a good example of this. All dogs share a common ancestor, the wolf. However, thousands of years of selective breeding have produced dogs with a variety of characteristics. Check your reading. How does selective breeding affect DNA? Genetic engineering. Within the last 50 years, it has become possible to directly change the DNA of an organism. Genetic engineering is the process in which a sequence of DNA from an organism is first isolated, then inserted into the DNA of another organism, changing that organism's DNA. The DNA that is engineered often codes for some particular trait of interest. Using technology, scientists can take a gene from one species and transfer it into the DNA of an organism from another species. The resulting organisms are referred to as genetically modified, GM, or transgenic. Check your reading. What are 3 steps involved in genetic engineering? One application of genetic engineering across species involves making plants more insect resistant. Genetic engineers have isolated genes in microorganisms that produce natural insect-killing chemicals, or pesticides. They have succeeded in transferring these genes into the DNA of crop plants, such as corn and soybeans. The cells of the genetically modified plants then produce their own pesticide, reducing the amount of chemical pesticide farmers need to use on their fields. Genetic engineering can address very specific needs. For example, in many parts of the world, soils are poor in nutrients, or the soil may contain salts. Such soil is not good for growing food crops. Genetic engineers have inserted a gene from a salt-tolerant cabbage into tomatoes. The salt-tolerant tomatoes can grow in soil that natural tomatoes cannot grow in. These tomatoes can also be grown using brackish water, which is water with a higher salt content than fresh water. There are risks and benefits associated with genetic engineering. Genetic engineering offers potential benefits to society, but also carries potential risks. Probably most people in the United States have eaten foods made from genetically modified corn or soybeans. The plants have bacterial genes that make them more resistant to plant-eating insects. This increases food production and reduces the amount of chemical pesticides needed. Less chemical pesticide on the ground reduces the risk of environmental pollution. However, many people worry that the natural pesticides produced by a genetically modified plant might have some effect on humans. What if genetically modified plants crossbreed with other plants and give protection to plants that are considered weeds? There is also the question of how to let people know if the food they eat is genetically modified. Many people think that such food should be labeled. Check your reading. What are some risks and benefits associated with using genetic engineering in food crops? There is uncertainty about how the DNA of genetically modified organisms might affect natural populations. For example, scientists are working with salmon that are genetically modified to grow more quickly. Fish are an important food source, and natural fish populations are decreasing. However, the salmon are raised in pens set in rivers or the sea. If the fish escape, they may breed with fish from wild populations. Government officials have yet to decide whether the benefit of having these fast-growing fish is worth the risk to wild populations. DNA technology has many applications. DNA technology is used in many different ways. It can be used to add nutrients to foods to make them more nutritious. It can be used to produce new and better drugs for treating disease. DNA technology can also be used to determine whether a particular drug might cause side effects in an individual, and it can be used to screen for and perhaps treat genetic disorders. DNA identification. You may have seen news stories about how DNA evidence is used to solve a crime. Law enforcement specialists gather as much DNA evidence as they can from a crime scene. For example, skin, hair, or blood. In a laboratory, they scan about 10 regions of the DNA that are known to vary from individual to individual. They use this information to produce a DNA profile, a DNA fingerprint. This fingerprint is unique to a person, unless that person has an identical twin. If DNA analysis of tissue found at the crime scene matches the DNA fingerprint of a suspect, then police know the suspect was at the scene. The more matches found between crime scene DNA and the suspect's DNA, the higher the probability that the suspect is guilty. Experts currently recommend that at least 4 to 6 DNA regions be matched to establish a person's guilt. The chances are very small that another person would have exactly the same DNA profile for all the DNA regions tested. Of course, the courts also take other forms of evidence into account before an individual is convicted of a crime. Studying genomes. One of the most challenging scientific projects ever undertaken was the Human Genome Project. A genome is all the genetic material in an organism. The primary goal of the project was to sequence the 3 billion nucleotide pairs in a single set of human chromosomes. The initial sequence was published in 2001. Scientists are now working to identify the approximately 30,000 genes within the human genome. Scientists have completed sequencing the genomes of many organisms. These organisms, often referred to as model organisms, enable scientists to compare DNA across species. Many of the genes found in model organisms, such as the fruit fly and mouse, are also found in the human genome. Scientists are aware that there are many ethical, legal, and social issues that arise from the ability to change DNA. We as a society have to decide when it is acceptable to change DNA and how to use the technology we have. Cloning is a technique that uses technology to make copies. It can be applied to a segment of DNA or to a whole organism. Cloning has been used in bacteria to produce proteins and drugs that help fight disease. Human insulin, which is used to treat people with a certain form of diabetes, is now produced in large quantities as a result of cloning techniques. The same technology, which is so helpful in one application, can be a cause of concern when applied in a different way. In 1996, scientists produced the first clone of a mammal, a sheep named Dolly. All of Dolly's DNA came from a single body cell of another sheep. The ability to clone such a complex animal raised many concerns about future uses of cloning. This, as well as many other possible applications of technology, makes it important that people understand the science of genetics. Only then can they make informed decisions about how and when the technology should be used. 5.3 Review. Key concepts. 1. What is a genetically modified organism? 2. What is the Human Genome Project? 3. List 3 different applications of DNA technology. Critical thinking. 4. Compare and contrast. How is selective breeding different from genetic engineering? How is it the same? 5. Analyze. Do you think a genetically modified trait in an organism can be undone? Why or why not? Challenge. 6. Analyze. Why might a genetically engineered drug, such as insulin, be better for treatment of disease than a drug that is manufactured chemically? Extreme science. DNA of extinct animals. Modern genetics meets the dodo and the solitaire. Hunted to extinction. The dodo bird was first sighted around 1600 by Portuguese sailors arriving on the shores of the island of Mauritius in the Indian Ocean. Portuguese sailors hunted the dodo, which was unable to fly, and used its meat for food. The bird, never having had contact with humans, did not run away. Only a mere 80 years later, the dodo was extinct. DNA evidence. Few bone specimens of the dodo bird remain today. Scientists collected and analyzed genetic material from preserved dodo specimens and specimens of another similar extinct bird called the solitaire bird. The DNA evidence was compared with the genetic material of about 35 species of living pigeons and doves. The DNA had a story to tell. Evidence suggests that the dodo and solitaire bird were close relatives. Their nearest living relative is a species of pigeon found in nearby Southeast Asia. From this evidence, scientists hypothesized that the dodo and solitaire bird species became separate almost 25 million years ago. In the geographic location of the island of Mauritius, the dodo developed its distinct characteristics, which eventually led to its extinction. Explore. 1. Make inferences. How can scientists use what they know from analyzing dodo bones to help them form conclusions about the physical characteristics of the bird? 2. Challenge. Several factors contributed to the extinction of the dodo bird. Look online to find out more about these factors. How can learning about what happened to the dodo help save today's endangered species from extinction?
Timelines in Science. The Story of Genetics. The Human Genome Project. DNA evidence in criminal cases. Cloning. News about genetics is everywhere. Some of the most exciting research in science today involves genes. The timeline shows that some important concepts that underlie the study of genetics were discovered relatively early. You will notice the influence of two major advances in technology: the development of the microscope during the 1600s and the development of computer technology during the later half of the 1900s. The boxes below the timeline show how technology has led to new understanding and to applications of those understandings. 1665. Cells discovered. Robert Hooke uses a microscope to study living matter. What he sees and then records in this drawing are tiny repeating units, which he calls cells. 1674. Cells are everywhere. Anton van Leeuwenhoek uses a microscope to study pond water and discovers the water is full of microscopic organisms made of just single cells. These drawings show some of what he saw. 1831. Cells have structure. As the power of microscopes improve, scientists start to describe the inner structure of cells. Robert Brown describes a central structure found in many cells, a structure that he calls the nucleus. 1866. Austrian monk describes patterns of heredity. Gregor Mendel's experiments with garden peas show that traits are passed on from parents to offspring in predictable patterns. 1882. Cells divide, chromosomes split. Scientists observe how cells divide. Walter Fleming describes how structures within the cell separate. These structures are the chromosomes. Chromosomes determine the traits of living things. 1928. Researchers study the chromosomes of fruit flies and find genes. Working with fruit flies, Thomas Hunt Morgan discovers that genes are found in specific locations on chromosomes. Application. Corn in every shape and size. Native Americans grew 700 different kinds of popcorn. Their popcorn plants were a kind of grass with big hard seeds that exploded when they were heated. People who didn't want to have to explode their corn to eat it chose plants with softer seeds and grew them, and then chose the softest of those seeds to plant. Over hundreds of years, by choosing which plants to grow, people produced what we now eat as corn on the cob. Technology. Seeing into the cell. Single glass lenses, such as the one von Leeuwenhoek used, were available as long ago as 1267. The compound microscope was first made in 1595, but it was over 200 years before it provided clear images. Until the 1930s, all microscopes focused light on objects.
Frontiers in Science. Life by degrees. What happens when Earth's climate changes? Scientists are studying how climate change has influenced the evolution of life on Earth. Climate and life. Throughout its history, Earth's climate has changed many times. Often the changes are gradual. They may seem small. However, an average global temperature change of just a few degrees can have a large impact on climate. Small changes in climate then cause big changes for plants and animals. Before there were humans to record events, Earth recorded its changes in its rocks and fossils. For example, scientists get a sense for Earth's climate at different times in the distant past by looking at fossils, the remains and traces of living things. If scientists find fossils of tropical plants in places near the Arctic Circle, then they may conclude that the climate in those places was different in the past. Scientists have found that warmer climates lead to a greater diversity of organisms. One researcher examined fossils of tiny organisms called phytoplankton. During cooler climate periods, there were fewer types of phytoplankton than during warmer periods. The same is true for other organisms. Peter Wilf and Konrad Lavandera studied fossil plants. They were especially interested in the marks they found on the plants. The marks were left by plant-eating animals who bit the leaves. The warmer the climate was, the more types of plants there were, and the more kinds of animals were eating the plants. Mass extinction. Several times in Earth's past, many kinds of animals and plants have disappeared in a relatively short time. These events are called mass extinctions. While we don't know for sure what causes them, most scientists think climate change plays a role in mass extinctions. The largest mass extinction in Earth's history happened at the end of the Permian period, about 248 million years ago. Scientists estimate that 90 to 95% of animal species that lived in the water died out. About 3/4 of the vertebrates, or animals with backbones, living on land died out too. Turn of events. What caused this extinction? Using fossils, scientists have concluded that Earth's climate became cooler. Material from erupting volcanoes may have blocked sunlight long enough to cool the Earth. The cool temperatures and lack of sunlight may have killed plants and animals. Scientist Peter Ward has been studying the Permian extinction. He looked at ancient African rivers and found evidence that rivers had become clogged with soil. Plants normally holding soil in place may have been wiped out, causing the soil on the riverbanks to loosen. The plant extinction would also have led to animal extinction. What hit them? Not all scientists agree about what caused the Permian extinction. If an asteroid hit Earth, it would push massive amounts of dirt and dust into the air. This could block sunlight and create a cooler climate. An increase in volcanic eruptions is another possible cause. The most famous extinction of all took place at the end of the Cretaceous period. The extended winter that may have followed a meteor impact caused many large animals, including dinosaurs, to become extinct. In a new climate, some species thrive and survive. They spread out and over time evolve to fill empty niches or unique roles in the environment. For example, before the Cretaceous extinction, the only mammals were small. After the dinosaurs became extinct, large mammals could fill the roles of large plant eaters and meat eaters. Even today, climate change continues. Earth's average temperature rose about 0.5 degree Celsius in the 20th century. Studying how past climate changes shaped life helps scientists predict how it may affect us in the future. Unanswered questions. Scientists have learned a lot about climate change and mass extinctions by studying fossils. There are many questions still to be answered. What caused changes in Earth's climate? What else might have caused mass extinctions? How might climate change affect life on Earth in the future?
Chapter 6. The History of Life on Earth. The Big Idea. Living things, like Earth itself, change over time. Key Concepts. Section 6.1. Earth has been home to living things for about 3.8 billion years. Learn how fossils help explain the development of life on Earth. Section 6.2. Species change over time. Learn how species develop and change. Section 6.3. Many types of evidence support evolution. Learn about the evidence scientists use to support evolution. 6.1. Key Concept. Earth has been home to living things for about 3.8 billion years. Before you learned, living things are diverse. Living things share common characteristics. A species is a group of living things that can breed with one another. Now you will learn how scientists use fossils to learn about the history of life, about patterns in the fossil record, about mass extinctions. Fossils provide evidence of earlier life. Imagine watching a movie about the history of life on Earth. The beginning of the movie is set 3.8 billion years ago. At that time, the ocean would have been the setting. All living things lived in the sea. The end of the movie would show Earth today, a planet that is home to millions of species living on land as well as in water and air. Of course, learning about the history of life isn't as easy as watching a movie. Modern ideas about life's history involve careful observation of the available evidence. Much of this evidence is provided by fossils. Fossils are the remains of organisms preserved in the earth. Fossils provide a glimpse of a very long story. In some ways, observing a fossil is like hitting the pause button on your video machine or looking at a snapshot of another time. Types of fossils. You may have learned that fossils are the imprints or remains of once living things. Most fossils are hard body parts, such as bone. Perhaps you have seen displays of dinosaur skeletons in museums. These displays include fossil bones, such as the jawbone to the left. Other fossils form when minerals replace the remains of organisms or parts of organisms. The trilobite fossil, shown in the middle photograph, is an example of this type of fossil. Fossils also include prints made by organisms. Very rarely, people find fossils that are the original remains of entire organisms. Explorers have found the frozen bodies of animals called woolly mammoths that lived about 10,000 years ago. The bodies of insects can be preserved in sap from plants. Finding the age of fossils. How can scientists tell that the first organisms lived in oceans, or that dinosaurs lived on land and that they disappeared 65 million years ago? These questions and others can be addressed by determining the age of fossils. There are two approaches to dating fossils: relative dating and absolute dating. In relative dating, one fossil is compared with another fossil. The relative age tells you whether a fossil formed before or after another fossil. The places where fossils are discovered provide information about their relative ages. Much of Earth's crust is rock, and rock forms over long periods of time. Understanding when and how rock forms gives scientists information about the sequence of events in Earth's history. Materials such as sand and mud may settle to the bottom of a body of water. Over many millions of years, layers harden into rock. Shells and other remains of organisms can be trapped in those layers, forming fossils. Newer fossils are usually found in the top layers of rock, while older fossils are in the lower layers. The absolute age of a fossil tells you when it was formed. To find the absolute age, scientists study the radioactive elements found in rocks and fossils. Some of these elements, such as uranium, decay at a very precise rate into more stable elements, such as lead. Thus, by measuring the amount of uranium and the amount of lead in an object, scientists can determine the object's age. The more lead it has, the older it is. Check your reading. What are the two ways scientists can determine the age of fossils? Assembling the fossil record. By combining absolute dating with relative dating, scientists can estimate the age of most fossils. The information about the fossils found in a particular location is called the fossil record. By assembling a fossil record, scientists can identify the periods of time during which different species lived and died. Scientists have used the fossil record to develop an overview of Earth's history. More complex organisms developed over time. One of the most striking patterns that scientists find when they study the fossil record involves the development of more complex organisms. Below you will see how scientists have reconstructed the history of a modern city to show how life has developed over time. Recall that the first organisms were made up of single cells. Most organisms living today are single-celled. However, more and more species have developed more and more complex cells and structures over time. Unicellular organisms. Unicellular organisms are organisms made up of a single cell. The organisms in the ocean 3.8 billion years ago were made of simple single cells. Some of these organisms are responsible for the amount of oxygen that now makes up our atmosphere. The early atmosphere did not contain as much oxygen as it now does. As the atmosphere changed, so did life on Earth. Different types of single cells developed over time. Over millions of years, the cells of organisms became more complex. Today, there are different species of life that include organisms made up of many cells. Multicellular organisms. Around 1.2 billion years ago, organisms made up of many cells began to live in Earth's oceans. Multicellular organisms are living things made up of many cells. Individual cells within multicellular organisms often perform specific tasks. For example, some cells may capture energy. Other cells might store materials. Still others might carry materials from one part of the organism to another. The most complex species of multicellular organisms have cells that are organized into tissues, organs, and systems. Recall that all organisms have similar needs for energy, water, materials, and living space. For almost 3 billion years, these needs were met only in oceans. According to fossil records, the earliest multicellular organisms were tiny seaweeds. The earliest animals were similar to today's jellyfish. Check your reading. Explain how unicellular and multicellular organisms differ. Life on land. Consider the importance of water. Without it, you and most other living things would not be able to live. About 500 million years ago, the first multicellular organisms moved from water to land. In order to survive, these living things needed structures to help them get water. The first land-dwelling organisms were simple plants and fungi. Plants were able to obtain water from the soil through structures called roots. Fungi absorbed water from plants as well as from the soil. Insects were also probably among the first living things to inhabit land. Plants provided insects with food and shelter. After insects, animals such as amphibians and reptiles began living on land. They were followed by birds and mammals. Earth's history includes mass extinctions. About 10,000 years ago, the last woolly mammoth died without any offspring. At that time, the species became extinct, which means it disappeared. The only way that we know that some species, such as woolly mammoths, ever existed is through the fossil record. During Earth's history, there have been several periods when huge numbers of species have died or become extinct in a very short time. These events are called mass extinctions. Although the fossil record shows a pattern of mass extinctions, two of these extinctions are particularly interesting. These are the Permian extinction and the Cretaceous extinction. The causes of these mass extinctions remain unknown. Permian extinction. About 250 million years ago, approximately 90% of the species living in the ocean became extinct. At the same time, many land-dwelling animals disappeared. Scientists who have studied Earth's history think that Earth's land masses joined together, forming one enormous continent. This event would have changed the climate on land and the conditions within Earth's waters. Cretaceous extinction. Fossils show that around 140 million years ago, animals called dinosaurs lived all over the planet. However, the fossil record for dinosaurs ends about 65 million years ago. At the same time, more than half of the other species living on Earth became extinct. How do scientists explain the extinction of so many species? One possibility is that a meteorite, a very large object from space, collided with Earth. The collision and its aftereffects wiped out most of the existing species. The remains of such a collision, the Chicxulub crater, can be found off the coast of Mexico. The computer graphic on page 15 shows the area of impact. The pattern in the fossil record shows that mass extinctions were followed by periods during which increasing numbers of new species developed. There may be a connection between the extinction of one species and the development of new species. For example, the extinction of dinosaurs may have made it possible for new species of mammals to develop. Check your reading. What do scientists think caused the most recent mass extinction? 6.1 Review. Key concepts. 1. How do fossils help scientists understand the history of life? 2. How do scientists know that the first organisms were simple unicellular organisms? 3. What is extinction? Give an example of a mass extinction and its results. Critical thinking. 4. Synthesize. How do absolute dating and relative dating help scientists assemble a fossil record for an area? 5. Sequence. Draw a timeline showing the sequence of 3 major events in the history of life. Include the following terms on your timeline: unicellular, multicellular, ocean, land. Challenge 6. Predict. Using the Denver reconstruction as your model, explain how you would reconstruct the history of the environment in your town. 6.2 Key concept. Species change over time. Before you learned, fossils are evidence of earlier life. More complex organisms have developed over time. Mass extinctions contributed to the development of Earth's history. Now you will learn about early ideas and observations on evolution, how Darwin developed his theory of natural selection, how new species arise from older species. Think about, how have telephones changed over time? Today, people across the world can communicate in many different ways. One of the most common ways is over the telephone. Looking at the 2 pictures, can you describe how this form of communication has changed over time? Scientists explore the concept of evolution. In a general sense, evolution involves a gradual change over time. You could say that the way humans communicate has evolved. Certainly, telephones have changed over time. The first telephones were the size of a shoebox. Today, a telephone can fit in the palm of your hand and can send images as well as sound. In biology, evolution refers to the process through which species change over time. The change results from a change in the genetic material of an organism and is passed from one generation to the next. Check your reading. What is evolution? Early ideas. In the early 1800s, a French scientist named Jean-Baptiste de Lamarck was the first scientist to propose a model of how life evolves. He became convinced that the fossil record showed that species had changed over time. He proposed an explanation for evolution based on the idea that an individual organism can acquire a new trait during its lifetime and then pass that trait on to its offspring. For example, Lamarck suggested that when giraffes stretch their necks to reach the leaves of tall trees, they pass the result of this stretching, a longer neck, to the next generation. Lamarck was a highly respected scientist, but he was unable to provide any evidence to support his idea. Check your reading. How did Lamarck explain the process of evolution? Darwin's observations. About 50 years after Lamarck, the British naturalist Charles Darwin published what would become the basis of the modern theory of evolution. As a young adult, Darwin spent 5 years as a naturalist aboard the Beagle, a ship in the British Navy. The map below shows the route Darwin traveled. As he sailed along the coast of South America, he studied rock formations and collected fossils. He also began to compare the new animals he was seeing with ones from his own country. The differences he saw in animals became more obvious when he visited the Galapagos Islands, a chain of volcanic islands about 950 kilometers, 600 miles, off the South American coast. On the 18 Galapagos Islands, plants and animals not only differed from those he saw on the mainland, but some differed from island to island. Darwin observed several types of tortoises on the islands. Tortoises with short necks were living in damp areas with abundant plant life that grew close to the ground. Longer-necked tortoises were living in dry areas with cacti. He considered whether the length of their necks made it possible for the tortoises to live in different environments. Darwin also found many different types of birds, called finches, living on the islands. Some finches were common in the treetops, while others lived in the lower shrubs of a neighboring island. Among the different islands, he noticed a variety of beak shapes and sizes. Some finches had heavy, short beaks, useful for pecking trees or seeds, while others had small, thin beaks that could be used for capturing insects. These observations caused Darwin to wonder if the birds had evolved from similar species. Natural selection explains how living things evolve. After Darwin returned home to England in 1836, he spent several years analyzing the observations and specimens he had collected on his voyage. He struggled to develop an explanation that would account for the amazing diversity of species he saw and for the relationships between them. By 1844, he had developed a hypothesis based in part on an insight from one of his hobbies, breeding pigeons. Darwin knew from personal experience that breeders can produce new varieties of an animal over time. The process breeders use is called artificial selection. For example, breeders produce a new breed of dog by selecting dogs that have certain desired traits, and then allowing only those individuals to mate. From the resulting litters, they again selectively breed only the individual dogs with the desired traits. By repeating this process generation after generation, a new breed is produced. Check your reading. What is artificial selection? Darwin's insight was that a similar process might be going on in nature. He proposed that through a process he called natural selection, members of a species that are best suited to their environment survive and reproduce at a higher rate than other members of the species. Darwin based this idea on a few key principles. These are overproduction, variation, adaptation, and selection. Overproduction. Take a look at how Darwin's ideas are useful for the study of salmon. When a plant or an animal reproduces, it usually makes more offspring than the environment can support, as you can see in the diagram on page 183. A female salmon may lay several thousand fertile eggs, but not all of them will hatch. Only a few hundred of the salmon that hatch from the eggs will survive disease and avoid fish-eating predators. Several dozen of these survivors will live to adulthood. An even smaller number will successfully reproduce. Variation. Within a species, there are natural differences, or variations, in traits. For example, if you looked very closely at thousands of salmon, you might see slight differences among individuals. Some might have larger fins. Others might have distinct patterns of spots on their scales. Many of the differences among individuals result from differences in the genetic material of the fish. Sometimes the genetic material itself changes, causing a new variation to come about. A change in the genetic material is referred to as a mutation. As the fish with the new variation reproduces, the trait gets passed on to its offspring. Therefore, genetic variations are passed on from one generation to the next. Adaptation. Sometimes a mutation occurs that makes an individual better able to survive than other members of the group. An adaptation is any inherited trait that gives an organism an advantage in its particular environment. For example, a slight change in the shape of a tail fin may increase a fish's chance of survival by helping it swim faster and avoid predators. Selection. Darwin reasoned that individual organisms with a particular adaptation are most likely to survive long enough to reproduce. As a result, the adaptation becomes more common in the next generation of offspring. As this process repeats from generation to generation, more members of a species show the adaptation. Consider the shape of the salmon. If a change in the tail fin makes the salmon better able to move upstream and lay eggs, scientists say this trait has been selected for in this environment. In other words, the species is evolving through natural selection. New species develop from earlier species. Darwin's personal observations and the work of another scientist, Alfred Wallace, led Darwin to write about this new concept of evolution. In 1859, after more than 20 years of work, Darwin published his ideas in his book, On the Origin of Species. This work led the way for our modern understanding of how new species arise. Speciation. Speciation is the evolution of new species from an existing species. Speciation may occur when the environment changes dramatically, or when the environment changes gradually. The Galapagos finch populations Darwin studied showed evidence of speciation. Isolation. Darwin's trip to the Galapagos Islands showed him an important point about speciation. Many new species had evolved after populations were separated from the mainland and were not able to breed with their mainland relatives. Darwin reasoned that isolation of populations by geographical or other barriers could contribute to the process of speciation. A species of fish called cichlids shows how a physical barrier contributes to speciation. In Lake Tanganyika, one of the largest lakes in the world, there are over 150 species of cichlids. Members of one particular genus, Tropheus, originally lived along the rocky shore and couldn't cross the open water. The climate and geology of the area caused the lake's water level to rise and fall many times over thousands of years. As the water level changed, a new rocky habitat was formed, and some populations of cichlids became isolated from each other. The isolated populations were unable to interact with each other because they couldn't cross open waters. As a result, genetic differences began to add up in these populations. The cichlid populations now represent distinct species. They have developed unique characteristics and cannot breed with each other. See diagram on page 184. Today's scientists understand that isolation is essential to speciation. For a species to separate, two populations must be prevented from reproducing with each other. A geographic boundary, like an ocean or mountain range, can result in isolation. Two populations of a species can also be isolated if they feed on different things, or reproduce at different times of the year. As the cichlids in Lake Tanganyika show, the mutations in one isolated group may differ from another. Two or more populations may evolve differently from each other. The result is speciation, which has contributed to the biodiversity on Earth. Check your reading. What is a key factor that can lead to speciation? 6.2. Review. Key concepts. 1. How did Lamarck's ideas differ from Darwin's? 2. What did Darwin observe in the finch populations that supported his idea of natural selection? 3. Explain how isolation helps speciation. Critical thinking. 4. Hypothesize. Two species of grasses are separated by a tall mountain range. A third species of grass shares some characteristics with each of the other two species. It inhabits a small valley, surrounded on all sides by mountains. Form a hypothesis for the origin of the third species. Challenge. 5. Predict. The Arctic hare lives in snow-covered mountains in Canada. The hare is hunted by foxes, wolves, and owls. Which trait is more likely to be inherited by new generations of hares? White fur or black fur? 6.3. Key concept. Many types of evidence support evolution. Before you learned, natural selection explains part of the process of evolution. New species develop from earlier species. Now you will learn how scientists develop theories, about the evidence Darwin used to support evolution, about additional evidence most scientists use today. Observations provide evidence for theories. In this chapter, you've learned about important observations that scientists have used to understand the history of living things. These observations provided Darwin with information he used to describe his ideas about evolution. Darwin, like all good scientists, was skeptical about his observations and conclusions. Although the historic trip on the Beagle took place between 1831 and 1836, Darwin didn't publish the book explaining his theory until 1859. In order to understand the importance of Darwin's work, it is also important to understand the meaning of the term theory. A scientific theory is a statement based on observation and experiment. If continued observation and experiment support the statement, it may become widely accepted. A theory that has been widely accepted is used to explain and predict natural phenomena. The chart above will help give you an idea of how a theory works and what evidence has been used to support evolution and the theory of natural selection. Check your reading. How do scientists support theories? Fossil evidence supports evolution. You have read that Darwin collected many specimens of fossils on his trip. These specimens provided evidence that species existing in the past were very similar to species living during Darwin's time. For example, the fossil of an extinct animal called the Glyptodon resembles the modern armadillo, an animal found today in South America. The geographic information about many fossils provides evidence that two species with a common ancestor can develop differently in different locations. An ancestor is an early form of an organism from which later forms descend. The idea of common ancestors is important to the theory of natural selection and to the evidence that supports the theory. Scientists comparing modern plants and modern algae to fossil algae can tell that they all share a common ancestor. Check your reading. What is a common ancestor? Biological evidence supports evolution. Today, scientists continue to study fossil evidence as well as biological evidence to support the concept of evolution. They have even returned to the Galapagos to further investigate Darwin's work. What they have found gives strength to the theory he proposed nearly 150 years ago. Returning year after year, These scientists are able to follow and record evolutionary changes as they are unfolding. The biological evidence they study includes the structure and the development of living things. This work has helped scientists identify relationships between organisms that exist today. In addition, their observations suggest how modern organisms are related to earlier species. Similarities in structure. Evidence for evolution can be observed within the physical structures of adult organisms. Scientists who study evolution and development consider two types of structural evidence. They are vestigial organs and similar structures with different functions. Check your reading. What are two types of structural evidence? Vestigial organs are physical structures that were fully developed and functional in an earlier group of organisms, but are reduced and unused in later species. In the bodies of whales, there are small leg bones that are vestigial. The skeletons of snakes also have traces of leg-like structures that are not used. These vestigial organs help researchers see how some modern organisms are related to ancestors that had similar structures. Similar structures with different functions. Scientists studying the anatomy of living things have also noticed that many different species share similar structures. But these structures are used differently by each species. For example, lizards, bats, and manatees have forelimbs that have a similar bone structure. As you can see from the diagram on page 31, there is one long bone that goes from a shoulder structure to a wrist structure. But obviously, a lizard, a bat, and a manatee use this structure in different ways. This similarity in structure indicates that these organisms shared a common ancestor. The process of natural selection caused the variations in form and function that can be observed today. These organisms lived in different environments, and so were under different pressures. For lizards, the environment was land. For bats, it was the air. And for manatees, the water. The environment influenced the selection of traits. Similarities in development. Scientists in the 1700s were fascinated by the fact that various animals looked similar in their earliest stages of life. They noted that as the organisms developed, they became less and less alike. Today, scientists continue to compare the developmental stages of different species. The adult stages of many species do not look similar. For example, a rabbit does not look anything like a chicken. However, studying reveals that the early life stages of a chicken and a rabbit are similar. An organism that is in an early stage of development is called an embryo. In the diagram below, notice the development of three different species: a chicken, a rabbit, and a salamander. In the early stages of development, the embryos of all three organisms look similar. As they continue to develop, they begin to take on distinct characteristics. The chicken has a structure that starts to resemble a beak. The salamander begins to look as if it adapted for life near water. In their adult stages, these three species no longer look similar. Genetic evidence supports evolution. The key to understanding how traits are passed from one generation to the next lies in the study of DNA, the genetic material found in all organisms. DNA contains the information all organisms need to grow and to maintain themselves. When organisms reproduce, they pass on their genetic material to their offspring. DNA contains a code that a cell uses to put together all the materials it needs to function properly. The code is made up of 4 different chemical subunits, called bases. The bases are symbolized by the 4 letters A, T, C, and G. Located within DNA are individual genes. A gene is a segment of DNA that relates to a specific trait or function of an organism. Each gene has a particular sequence of bases. The cell takes this sequence and translates it into the chemicals and structures the organism needs. Scientists studying genes have identified a gene called the clock gene in many mammals. This particular gene relates to the function of sleeping and waking. As scientists learn more, they can identify patterns of behavior in different organisms. The chart on page 194 compares the DNA sequence of part of the clock gene in both humans and mice. Check your reading. What is a gene? Scientists can tell how closely organisms are related by comparing their DNA. The more matches there are in the sequence of bases between two organisms, the more closely related they are. For example, almost all of the genes found in a mouse are also found in a human. Even though the two organisms appear so different, much of the functioning of their cells is similar. 6.3. Review. Key concepts. 1. Describe in your own words how scientists use the word theory. 2. What type of evidence did Darwin use to support his theory of evolution? 3. Identify 3 different types of evidence that today's scientists use to support the theory of evolution. Critical thinking. 4. Analyze. Describe 3 characteristics of a scientific theory. Explain how Darwin's theory of evolution is an example of a scientific theory. Challenge. 5. Predict. If you were looking at the sequence within the genes of 2 species, how would you predict that the 2 species are related? Think science. Skill. Evaluating hypotheses. How did the deep-sea angler get its glow? A fish that uses a fishing pole to catch food might seem odd. However, anglerfish do just that. The fish have a modified spine that extends from their head, almost like a fishing pole. At the end is a small piece of tissue that is similar in shape to a small worm. The tissue functions like a lure that a fisherman uses to catch fish. The anglerfish wiggles its lure to attract prey. If the prey fish moves in close enough, the anglerfish opens its mouth and swallows the prey whole. The fishing poles of abyssal anglerfish that live in the deep sea, have an interesting adaptation. The lure actually glows in the dark. It is bioluminescent. Observations. From laboratory research and field studies, scientists made these observations. There are more than 200 species of anglerfish. Many of these live in deep water. Shallow water species do not have glow-in-the-dark lures. Only female abyssal anglerfish have a pole. They do not have pelvic fins and are not strong swimmers. Other deep-sea organisms, including bacteria, jellyfish, even some squid, are bioluminescent. Hypotheses. Consider these hypotheses. The ancestors of abyssal anglerfish lived in shallow waters. Some of these fish drifted into deep waters. A bioluminescent lure helped some survive. Light does not reach down to the bottom of the deep sea. Bioluminescence provides an advantage for the anglerfish because it makes its lure noticeable. A bioluminescent lure is more valuable to a female abyssal anglerfish than the ability to swim. Evaluate each hypothesis. On your own. For each hypothesis, think about whether all the observations support it. Some facts may rule out some hypotheses. Others may support them. As a group, decide which hypothesis is the most reasonable. Discuss your thinking and conclusions in a small group and see if the group can agree.
Chapter 7. Classification of Living Things. The Big Idea. Scientists have developed a system for classifying the great diversity of living things. Key concepts. Section 7.1. Scientists develop systems for classifying living things. Learn about why scientists classify living things and about taxonomy. Section 7.2. Biologists use 7 levels of classification. Learn about scientific names, how to classify organisms in 7 levels, and dichotomous keys. Section 7.3. Classification systems change as scientists learn more. Learn how classification systems have changed based on features of cells. 7.1. Key concept. Scientists develop systems for classifying living things. Before, you learned natural selection helps explain how new species develop. Evidence indicates that species change over time. New species develop from ancestral species. Now you will learn why scientists classify living things, that taxonomists study biological relationships, about evidence used to classify organisms. Think about, how are these organisms similar? Both a worm and a caterpillar share many characteristics. Both have long, skinny bodies that are divided into segments, but an earthworm moves underground, has no legs or eyes, and can grow back segments that are lost. A caterpillar crawls above ground and is just one part of a butterfly's life cycle. As you read this chapter, think about whether you would classify these animals together or separately. Scientists classify millions of species. About 400 years ago, scientists who studied insects classified them based upon their appearance and behavior. If animals looked alike, researchers concluded that they were related. In the last few centuries, scientists have realized that appearances can suggest false connections. Although caterpillars look like worms, they are actually an earlier stage of a butterfly's life. For some people, the world seemed to grow larger during the 1600s. Travelers sailed to distant lands and oceans. Scientists went on many of these trips, observing and collecting samples of living things they had never seen before. In addition, the microscope allowed scientists to see tiny organisms that had been invisible before. But how could scientists organize and talk about this wonderful new knowledge? Classification and taxonomy. Two scientific processes deal with classifying and naming living things. Classification is the process of arranging organisms into groups based on similarities. Taxonomy is the science of naming and classifying organisms. A good system of classification allows you to organize a large amount of information so that it is easy to find and to understand. The system should provide a tool for comparing very large groups of organisms as well as smaller groups. Large groups might include all animals. Smaller groups might include birds, reptiles, or mammals. A good system of taxonomy allows people to communicate about organisms. Before the 1700s, scientists had not agreed on a system of naming and grouping organisms. Take, for example, the common wild briar rose. Some scientists called it Rosa sylvestris inodora seu canina, odorless woodland dog rose. Others used the name Rosa sylvestris alba cum rubore folio glabro, pinkish-white woodland rose with hairless leaves. Plus, any scientist studying a species could change the name. These long Latin names may sound confusing, but even common names can be confusing. In England, the bird called a robin, Britain's national bird, is only distantly related to the bird called a robin in the United States, even though they both have red feathers on their chests. A daddy longlegs could be either a long-legged relative of spiders in the United States or a long-legged relative of mosquitoes in England. Clearly, biologists need both a system for organizing and a system for naming. Each name should refer to one specific type of organism. That way, scientists can use the species name and be sure that everybody knows exactly which organism they are talking about. Check your reading. What is the difference between classification and taxonomy? Using classification. To classify organisms, scientists use similarities and differences among species. Sometimes these differences are easy to see, such as whether an animal has fur, feathers, or scales. Other times, Seeing the differences requires special laboratory equipment, such as equipment to study DNA. A classification system can help you identify unfamiliar organisms. For example, if you had never heard of a caracal but were told that it was a kind of cat, you already would know many things about it. It has fur, fangs, and sharp claws. It's a meat eater, not a plant eater. You would know these things because the caracal shares those characteristics with all of the members of the cat family. If you looked up caracal in an encyclopedia, you'd find that your guesses were right. The caracal is a small wildcat native to Africa, the Middle East, and India. It weighs about 13 to 19 kilograms, 29 to 42 pounds. The name caracal comes from a Turkish word meaning black-eared. The more characteristics two organisms share, the more similar their names should be in the classification system. The caracal, a pet cat, and all the cats below are different in size, habitat, and other characteristics. But they also have many similarities, and all belong to the cat family Felidae. Taxonomists study biological relationships. Scientists need a simple, standard way of arranging all of the different species. The science of taxonomy is related to the Greek word taxis, which means arrangement. Taxonomists are the scientists who classify and name organisms based on their similarities and differences. A taxon is a group of organisms that share certain traits. Taxons can be broad, like animals and plants, or more specific, like cats and roses. As you learned in Chapter 1, living things evolve over time. A single species found in a fossil record might be the ancestor of many different species found on Earth today. Taxonomists study the relationships between species, trying to discover how one species evolved as compared to another species. Species that share ancestors are grouped together. To determine how to classify organisms, scientists compare a variety of characteristics or traits. A trait is a characteristic or behavior that can be used to tell two species apart, such as size or bone structure. If two organisms share a trait, taxonomists try to determine if they share the trait because they share an ancestor. Check your reading. How do taxonomists use biological relationships to classify organisms? Look at the photographs and try to determine to which organism a leafy sea dragon is more closely related. The leafy sea dragon shares traits with all of the other organisms pictured. For example, the sea dragon and the sargassum seaweed look similar, with greenish wavy fronds. But the sea dragon is an animal that moves, gets food from other organisms, and breathes oxygen. The sargassum seaweed is not an animal. It is a type of algae. The sargassum fish shares more traits with the sea dragon, but its body is a much different shape and has scales. In fact, the leafy sea dragon is an animal that is closely related to a seahorse. Both have heads and bodies with similar shapes, and neither has scales. The seahorse shares more traits with the leafy sea dragon than with the other two organisms. Taxonomists take evidence and try to reconstruct the evolution of a species. Then they place the species in the classification system. Scientists use physical evidence, such as fur, bones, and teeth. They also use genetic evidence, which is found within an organism's DNA. Physical evidence. The primary tools early scientists used for taxonomy were their eyes and measuring devices. They collected examples of organisms and noted characteristics such as color, size, weight, and how groups of organisms obtain energy. Scientists who studied animals observed the internal structure as well as outward appearances. These physical features are still important today. Individuals of two species, such as the two jays shown to the left, can have many similarities as well as some differences. One obvious difference is the color pattern. Another is the area of the world in which they live. Blue jays live east of the Rocky Mountains, and Steller's jays live west of the Rockies. The common names and the scientific names reflect the differences and the common ancestor. Blue jay, Cyanoceta cristata, and Steller's jay, Cyanoceta stellari. Skeletons, shells, and other hard parts of organisms become fossilized more easily than soft parts do. Scientists can observe and measure fossilized bones or pieces of bones and compare them with each other. They can also compare bones of species that are extinct with bones of modern species. From such studies, scientists can determine many things about the organism. Physical evidence provides clues about how an organism may have lived, how it moved, or what type of food it ate. All of this physical evidence helps scientists see that all living organisms are related by evolution. Some are more closely related than others. This means they share a more recent ancestor. Check your reading. How could comparing fossilized bones with a modern animal's bones help you see the modern animal's evolutionary history? Genetic evidence. In the early 20th century, scientists discovered that organisms inherit their traits through structures called genes. In the mid-1950s, they observed that genes are made of DNA and that DNA stores coded information. Today, scientists can use laboratory machines to catalog each component of an organism's DNA. With that information stored on a computer, scientists can compare the components of a gene from one organism with the components of the same gene from another organism. Genetic evidence usually supports physical evidence, but not always. Consider the example shown on page 49. For years, taxonomists argued about how to classify the small reddish animal from China. Its scientific name is Ailurus fulgens, and the common name is red panda. Later, scientists discovered a larger bear-like animal in China, which they called the giant panda. Both pandas ate only bamboo, shared a common name, and their faces looked similar. Scientists concluded that they were related to each other and to raccoons. However, molecular evidence has shown that the red panda is more closely related to raccoons, and the giant panda is more closely related to bears. 7.1. Review. Key concepts. 1. Describe the benefits of classifying species. 2. Why do taxonomists study biological relationships? 3. How do scientists use genetic evidence when classifying organisms? Critical thinking. 4. Analyze. Why do people need a universal system of naming organisms? 5. Predict. The animal called a marbled godwit is a bird. What traits would you predict it has? Challenge. 6. Synthesize. Suppose you found two species of cave-dwelling lizards without eyes living on opposite sides of the world. Explain how you would try to determine if the two species were closely related. 7.2. Key concept. Biologists use 7 levels of classification. Before, you learned classification is a system of organization. Evidence is used to classify organisms. Now you will learn about scientific names, about 7 levels of classification, how to use a dichotomous key. Linnaeus named about 4,000 species. Scientists name species and arrange them into groups. One scientist named Carolus Linnaeus developed systems for both naming species and organizing them into groups. All 4,000 species that Linnaeus named were plants or animals. Today, scientists have named over 1 million species. Linnaeus used appearance to group species. As you have read, modern scientists also use appearance, along with other types of evidence, to arrange species into groups. Naming species. Sometimes using only one word to name an organism isn't specific enough. If you are telling a friend about your favorite writer, you might name Mary Oliver, or Mary Whitebird, or Mary Shelley. Using only Mary won't help your friend know of the author you name, so you use two words. In a similar way, scientists use two words to name organisms. A genus is a group of species that have similar characteristics. For example, the genus Ursus groups all of the animals known as bears. Included in this genus are Ursus arctos, grizzly bears, and Ursus maritimus, polar bears. Members of the same genus are closely related. The system for naming species developed by Linnaeus is the basis of modern taxonomy. We call this system binomial nomenclature. Binomial means two names, and nomenclature means list of names. So binomial nomenclature describes a system of naming something using two names or words. Most scientific names are Latin terms. Using scientific names. Linnaeus' system of binomial nomenclature made communication about certain species much easier. When naming an organism, the use of a genus name as well as a species name is necessary. If the genus name is not included in the scientific name, the identity of a species can be a mystery. For example, the species name of the three different species shown above is Gracilis. The word gracilis means graceful or slender in Latin. Aubrietta gracilis is a type of flower found in a rock garden. Cameleo gracilis is a type of lizard called a chameleon. Mammillaria gracilis is a type of cactus. People follow certain rules when they write scientific names. The genus name comes first. The first letter is capitalized, and the entire name is in italics. The species name is also written in italics, follows the genus name, and the first letter is in lowercase. Check your reading. What is the difference between a genus and a species? In addition to species and genus, the classification system includes several larger groups. Each larger group includes one or more smaller groups. Turn to page 54 to read about the larger groups in our modern system of classification. Organisms can be classified into 7 levels. You've read about species and genus, the most specific levels of the classification system most scientists use today. There are 7 levels that describe a species. The largest level is the kingdom, the group containing the most species. The 7 levels of classification for a spotted turtle and a house cat are listed below. 1. Kingdom. Animalia. The animals. 2. Phylum. Chordata. Animals with backbones. 3. Class. Mammalia. Mammals or furry animals that nurse their young. 4. Order. Carnivora. Carnivores or animals that kill and eat other animals. 5. Family. Felidae. The cat family. 6. Genus. Felis. House cats, cougars, and many others. 7. Species. Catus. All house cats, no matter what their breed. Like the cat, the turtle is also classified into 7 levels. However, only the 2 largest levels, Animalia and Chordata, are the same as the classification for a house cat. The more names an organism shares with another organism, the more closely related the 2 organisms are. Cats and turtles are both animals with backbones, but are otherwise different. Spotted turtles have more traits in common with snakes and lizards than with cats. Lizards, snakes, and turtles all belong in the class Reptilia. Phyla are more specific than kingdoms. Classes are more specific than phyla, and so on. The illustration on page 55 shows how kingdom is the broadest grouping of organisms and species is the most specific. Scientists can compare very broad categories of organisms, such as kingdoms and phyla. Or they can compare very specific categories, such as species. If scientists wish to compare all the different types of turtles to one another, then they will compare the organisms in the order Testudinus. But if the scientists want to compare turtles that live in or near water, then they will compare only organisms in the family Emydidae. You can remember the classification levels and their order with this memory aid. Kings play chess on fat green stools. The first letter of each word is the same as the first letter in each level of classification. Kingdom, phylum, class, order, family, genus, and species. A complete classification of humans goes like this: Kingdom, Animalia, phylum, Chordata, class, Mammalia, order, Primates, family, Hominidae, genus, Homo, species, Sapiens. Check your reading. Which level of classification in the 7-level system includes the most species? Dichotomous keys and field guides help people identify organisms. With millions of organisms on Earth, how could a specific one be identified? Even if you know some of the larger categories, it can be difficult to find the species, genus, or even family name of many organisms from a long list of possibilities. Take a beetle, for example. Even if you knew that it is in the kingdom Animalia, phylum Arthropoda, animals with jointed legs, class Insecta, insects, and order Coleoptera, hard-winged insects, you'd still have to choose among 300,000 known species of beetles that have been discovered around the world. Taxonomists have come up with a tool to identify organisms such as this beetle. A dichotomous key asks a series of questions that can be answered in only 2 ways. Your answer to each question leads you to another question with only 2 choices. After a number of such questions, you will identify the organism. One example of a dichotomous key for trees is shown on page 57. The questions in a dichotomous key gradually narrow down the list of possible organisms. The questions can ask about any trait. The idea is simply to make identifying an organism as easy as possible. The dichotomous key for trees, for example, asks a set of questions that only ask about the traits of the leaves. Leaves are usually easy to get from a tree that needs to be identified, and they include many characteristics that can be used to tell different trees apart. Another tool for identifying organisms is a field guide. Field guides include paintings or photographs of familiar species. Flower guides may start with a flower's color. Bird guides are arranged by orders and families. Field guides also include maps showing where organisms live. Check your reading. What 2 tools have taxonomists developed to identify organisms? 7.2. Review. Key concepts. 1. What is binomial nomenclature? 2. Write the names of the 7 levels of classification. Which level contains the most organisms? 3. What makes a dichotomous key easy to use? Critical thinking. 4. Summarize. What were Carolus Linnaeus's main contributions to taxonomy? 5. Compare and contrast. Compare a dichotomous key with a typical field guide. What are the strengths and weaknesses of each? Challenge. 6. Synthesize. Predict what differences you might find among organisms in the same species. Extreme science. New insect species. The undiscovered. Everyone agrees that insects are the largest group of animals on Earth, but nobody knows exactly how many insect species exist. Some estimates predict that there are as many as 30 million. However, only about 900,000 have been classified. 29 million insect species may be waiting to be discovered. Where in the world? Many of the new insect species are found in tropical forests of South America and Asia, but plenty may be hiding close to your own home. The most massive bug in Southern California went undiscovered until April 4, 2002. This wingless relative of the Jerusalem cricket looks something like a puffed-up 3-inch-long ant. While studying for her graduate degree, Christina Sandoval captured insects in Santa Barbara, California. She caught an unidentified species of walking stick insect, which she named after herself, Timema christini. The Hanford Nuclear Reservation in Washington State was closed to the public for about 50 years. After it opened for cleanup, the Nature Conservancy found 27 new insect species in just 4 years, including a new micromoth less than 1/8 inch long. Scientists think that over 1/3 of the estimated 164,000 insect species in the United States have yet to be discovered and named. Start looking. Who knows where they'll be? A whole new order. In March 2002, for the first time in 87 years, a whole new order of insects was discovered. Insects in this order look like a cross between stick insects, praying mantises, and grasshoppers. Upon its discovery, the order was nicknamed gladiators. Now called Mantophasmatodia, the gladiator bugs raised the total number of insect orders to 31. Explore. 1. Analyze. List some things about an insect that could be included in its species name. Tell why each is important. 2. Challenge. Scientists recently discovered a new centipede in New York's Central Park, the first new species in the park in over 100 years. Centipedes are related to insects. Find out what centipedes and insects have in common and how they differ. 7.3. Key concept. Classification systems change as scientists learn more. Before you learned, scientists give each species a unique scientific name. There are 7 levels of classification. Dichotomous keys help us identify organisms. Now you will learn about the connection between new discoveries and taxonomy, about 3 domains, about 6 kingdoms. Think about, how do scientists define kingdoms? Look at this photograph of a sea urchin. It lives its life buried in or slowly moving across the ocean floor. The sea urchin's mouth is located on its underside. It feeds on food particles that settle on or are buried in the ocean floor. The sea urchin doesn't appear to have much in common with a tiger, an alligator, even a human. Yet all of these organisms belong in the same kingdom, called Animalia. Why do you think scientists would group these organisms together? Taxonomy changes as scientists make discoveries. The list of species continues to grow as scientists discover new species. In addition, taxonomists are learning more about the evolutionary history of species. As you read in Section 1, new knowledge resulted in the reclassification of species, such as the giant panda. Both the names of species and the groups into which they are arranged may change as a result of discoveries about the evolution of these species. Early scientists described 2 large groups of organisms, plants and animals. Plants were described as green and non-moving. Animals moved. Most scientists today use a system that includes 6 kingdoms. In addition, taxonomists have added a level of organization above the kingdom level. 3 domains. Microscopes and other advances in technology have allowed scientists to observe that there are 3 fundamentally different types of cells. On the basis of this observation, scientists have arranged kingdoms into larger groups called domains. For example, the domain Eukarya contains the protists, fungi, plants, and animals. The table below summarizes the relationships among the 6 kingdoms and the 3 domains. You will learn more about kingdoms in the rest of this section. The photographs below show examples of cells from each domain. One of the traits that distinguishes cells of Eukarya from cells of bacteria and archaea is the presence of a nucleus. Cells that contain a nucleus are called eukaryotic cells, and cells that do not contain a nucleus are called prokaryotic cells. The domains bacteria and archaea include only organisms with prokaryotic cells. The domain eukarya includes only organisms with eukaryotic cells. Check your reading. How are prokaryotic cells different from eukaryotic cells? 6 kingdoms. The classification system that many scientists use today has 6 kingdoms. Every known species on Earth is included in one of these 6 kingdoms. Kingdom Plantee includes plants such as trees, grass, and moss. Kingdom Animalia includes animals, from lions and tigers and bears to bugs and multicellular microbes. Kingdom Protista includes organisms that don't fit easily into animals, plants, or fungi. They are either unicellular organisms or have a simple multicellular structure. Kingdom Fungi includes mushrooms, molds, and yeasts. Kingdom Archaea contains organisms that are similar to bacteria, but have a cell structure that is so different that scientists separate them into their own kingdom. Kingdom Bacteria are unicellular organisms with no nucleus. This system may change as scientists learn more about the species in each kingdom. Before 1990, most scientists preferred a 5-kingdom system that combined archaea and bacteria into a single kingdom. However, as scientists learned of chemical differences between the cells of the species, they arranged them into 2 kingdoms. Today, some scientists suggest that the kingdom Protista should be arranged into smaller kingdoms because of the many differences among its species. Many scientists agree on a 3-domain and 6-kingdom system, similar to the one summarized on pages 61 through 63. Check your reading. Which of the 6 kingdoms include unicellular organisms? The 2 most familiar kingdoms are plants and animals. Carolus Linnaeus divided all of the species he identified into 2 large groups, plants and animals. People still use these groups to describe most living things today. But these 2 kingdoms also include unfamiliar organisms. It might seem odd that living things that are so different from each other, humans, elephants, termites, ducks, fish, worms, are all part of the same group. However, all of these organisms share some general traits, just as all plants share another set of general traits. Plantee. About 250,000 plant species live on Earth. They range from tiny mosses to the largest organisms on the planet, giant sequoia trees. The oldest living organism on our planet is a plant called the bristlecone pine. Some living bristlecone pines were growing when the Egyptians built the pyramids about 4,000 years ago. All plants are multicellular and are eukaryotes, which means their DNA is stored in the nucleus of their cells. All plants are able to make sugars using the sun's energy. Plants cannot move from place to place, but they can grow around objects, turn toward light, and grow upward. Plant cells are different from animal cells because plant cells have tough walls outside their cell membranes. Animalia. Scientists have already named 1 million species in the kingdom Animalia. Many different types of animals inhabit the planet, but more than 90% of the named species are insects. The animal kingdom also includes familiar animals such as whales, sharks, humans, bears, dogs, and fish. All animals get their energy by eating other organisms, or by eating food made by other organisms. Animals have the ability to move around for at least part of their life. Most animals have mouths and some type of nervous system. Plant and animal cells are both eukaryotic, but animal cells have no cell walls. Check your reading. What is the most abundant type of species in the animal kingdom? Other organisms make up 4 more kingdoms. Carolus Linnaeus's classification systems included the organisms he knew about in the late 1700s. Some of the organisms Linnaeus called plants, the mushrooms, molds, and their relatives, turned out to have some characteristics very different from those of plants. Biologists now put fungi in a kingdom of their own. Three other kingdoms consist mainly of microscopic organisms. These are Protista, Archaea, and bacteria. Most organisms on Earth are classified as bacterial or archaea. These are prokaryotic organisms, which have small, simple cells and no nuclei. Check your reading. What are the four kingdoms besides Plantae and Animalia? Protista. The kingdom Protista includes a wide variety of organisms. Most protists are unicellular. Protists that are multicellular have structures that are too simple to be classified as animals, plants, or fungi. All protists have large complex cells with a true nucleus, eukaryotes. Some eat other organisms as animals do. Some get energy from sunlight as plants do. Some protists resemble fungi. However, protists that are multicellular do not have as many specialized cells or structures as plants, animals, and fungi. Many protists live in pond water or seawater. The largest of these unicellular species are barely visible without a microscope. However, large organisms such as seaweeds are also classified as Protista. Some seaweeds can grow hundreds of feet in a single year. Different groups of protists evolved from different ancestors. Scientists still debate whether kingdom Protista should be classified as one kingdom or should be split into several kingdoms. Fungi. Every time a loaf of bread is baked, a fungus is responsible for the rising dough. One group of fungi called yeasts makes it possible for us to make bread and many other food products. Another type of fungi that people eat includes some mushrooms. A mushroom grows in thin threads underground, and only the small cap breaks above the ground. Fungi are usually divided into three categories: mushrooms, molds, and yeasts. The trait that separates fungi from other organisms is that fungi take in nutrients from their surroundings instead of eating other organisms or using sunlight. Both plants and fungi remain rooted in one place. Most fungi have cell walls similar to the cell walls of plants. Unlike plants, however, many fungi act as decomposers, breaking down dead or decaying material into simpler parts that can be absorbed or recycled by other organisms. Archaea. In the mid-1990s, a researcher studying the genes of some bacteria discovered that although they resembled bacteria in size and cell type, some species had very specific genetic differences. After more study, scientists decided to call these organisms Archaea. They differ so much that scientists now classify Archaea in a separate kingdom or domain of Archaea. In some ways, Archaea appear to be more related to eukaryotes, organisms with complex cells containing nuclei, than to bacteria. Archaea do not have nuclei, but their cell structure is different from that of bacteria. Like bacteria, Archaea live in many environments, especially in the ocean. But they also live in some very extreme environments, such as boiling mud near geysers, hot vents at the bottom of the ocean, salty ponds, and deep under the sand. Check your reading. Which traits classify an organism as part of the kingdom Archaea? Bacteria. Bacteria live nearly everywhere on Earth. This kingdom includes organisms that cause human disease and spoil food. But most of these organisms are helpful members of biological communities. All bacteria are unicellular organisms. They have small, simple cells without a nucleus. Most bacteria have a cell wall outside the cell membrane, but this wall is not the same as the cell wall of plants. Bacteria reproduce by dividing in two and can produce many new generations in a short period of time. Species and environments change. In the last chapter, you read about the ways species change over time. You have also read how the evolutionary history of species helps scientists classify living things. Scientists have named over 1 million species and placed them into 6 kingdoms. In addition, scientists estimate that there are millions, maybe tens of millions, more species that haven't been discovered. Scientists have also discovered forms of life preserved in the fossil record. Some of those organisms are the ancestors of organisms that live today. Species evolve over time as individual organisms and environments change. Individual organisms are faced with many other pressures that affect daily lives. These pressures may come from changes in their living space, in the availability of food or other resources, or from other organisms. In the next chapter, you will read about how groups of species are affected by changes in their surroundings. 7.3 Review. Key concepts. 1. What are the names of the 6 kingdoms used in the classification system? 2. How are species sorted into the various kingdoms? Critical thinking. 3. Communicate. Make a table with columns headed Characteristics, Animalia, and Plantee. Using as many rows as needed, list characteristics that differ between these 2 kingdoms. 4. Analyze. Explain how fungi differ from plants. Challenge. 5. Analyze. One bacterium has a membrane surrounding its DNA. Should this organism be classified with the eukaryotes? Why? Or why not? Timelines in science. Life unearthed. How do scientists know about life on Earth millions of years ago? They dig, scratch, and hunt. The best clues they find are hidden in layers of rock. The rock-locked clues, called fossils, are traces or remains of living things from long ago. Some fossils show the actual bodies of organisms, while others, such as footprints, reveal behavior. Before 1820, most fossil finds revealed the bodies of ocean life. Then large bones of lizard-like walking animals began turning up, and pictures of a new, terrible lizard, or dinosaur, took shape. Later, discoveries of tracks and nests showed behaviors such as flocking and caring for young. Even today, discoveries of living fossils, modern relatives of prehistoric species, have offered us a rare glimpse of the activity of early life. 1824. Giant lizards from fragments. William Buckland describes Megalosaurus, a giant crocodile-like animal he studies from only a few bits of jaw, teeth, ribs, pelvis, and one leg. A year later, Gideon Mantell assembles Iguanodon, a similar animal, from fossil bones. 1909. Burgess Shale shows soft bodies. In the Burgess Pass of the Canadian Rocky Mountains, Charles Walcott finds fossils preserved in shale, a soft rock that preserves lace-like details, such as the soft tissues of the morella. The glimpse of life 505 million years ago is the earliest yet seen. 1938. African fisherman hauls in history. A South African fisherman pulls up a 5-foot fish he has never seen. He calls the local museum, whose curator, a naturalist, has also never seen the species. To her surprise, biologists identified as a coelacanth, a prehistoric fish thought to be extinct for more than 50 million years. 1944. New dawn for the dawn redwood. Beside a small temple, a Chinese scientist discovers the dawn redwood, or metasequoia, growing. Common in fossil specimens 100 million years old, the tree had not been seen alive in recorded history. The 1944 find starts a search, and in 1948, scientists find a small wild grove in China as well. Technology. Removing fossils with care. The technology for removing fossils from rock beds has not changed much since the 1820s. Collectors still work by hand with hammers, chisels, trowels, dental picks, and sieves. Gideon Mantell used these when he chiseled out iguanodon bones embedded in one large rock called the mantelpiece. Fossil hunters also use hand lenses and microscopes. Sometimes a protective layer is built up with glue, varnish, or another finish. For larger samples, a plaster cast often supports the fossil. Most fossils are packed using a technology found in any kitchen, a sealable plastic bag. Application: protecting fossils and dig sites. The United States Antiquities Act of 1906 preserves and protects historic and prehistoric sites. The Act requires collectors to have a permit to dig for or to pick up fossils on public lands, such as national parks. It also requires that any major find be publicly and permanently preserved in a museum. The United Nations also now designates World Heritage Sites. For example, the original Burgess Shale find in Yoho National Park in Canada is now protected by international law. Since 1906, many states and provinces in Canada have enacted their own laws about land rights and the excavation and transport of fossils. 1953. Piltdown Man, no Neanderthal. Scientists once applauded the discovery in 1912 of a Neanderthal skull in the Piltdown gravel pit, but a few had their doubts. In 1953, radioactive potassium dating proves the Piltdown Man to be nothing more than the jaw of an orangutan, placed beside human skull fragments. 1974. Lucy and upright kin found. Digging in Ethiopia, Donald Johansen finds an almost complete hominid skeleton. He names the fossil Lucy after a Beatles song. Lucy is over 3 million years old, is 3 1/2 feet tall, and has an upright stance or posture. A year later, Johansen's crew finds the first family, a group of 13 skeletons of the same species as Lucy. 1990. Largest Tyrannosaurus, Sue. Out on a walk with her dog in the South Dakota Badlands, amateur fossil hunter Sue Hendrickson discovers 3 huge bones jutting out of a cliff. Hendrickson finds the largest and most complete T-Rex skeleton yet. The 67-million-year-old Sue is now on display in the Field Museum in Chicago, Illinois. 1993. Oldest fossils are too small to see. Fossils discovered up to this point date back about 550 million years, to the dawn of the Cambrian period. J. William Schopf identifies fossils of microorganisms scientifically dated to 3.4 billion years ago. This startling find near Australia's Marble Bar opens up a vast period of time and once again reshapes theories about life's beginnings. Technology. How old is a fossil? Before 1947, scientists used a method of fossil dating called relative dating. They assigned a date to a fossil according to the rock layer in which it was found. The deeper or older the layer, the older the fossil. The discovery of radiometric dating in 1947 marked the first time a fossil's date could be pinpointed. Organic matter decays at a constant rate, so by measuring the rate of decay, you can tell the age of the matter. Radiocarbon-14 is used to tell the age of a fossil that is less than 10,000 years old. Since most fossils are older than that, scientists use other methods. Potassium argon decays more slowly than carbon. It is a more common method of dating. All types of fossil dating have margins of error or limits to accuracy. Technology. Fossil classification and DNA. There are many ways to classify fossils. Scientists look at bone structure, body posture, evidence of behavior, and environment. Microscopes are used to identify organisms too small for the eye to see. Study of DNA molecules helps to identify species when soft tissues remain intact, such as in fossils formed in amber or crystallized tree sap. In 1985, polymerase chain reaction, PCR, became the simplest method to study the DNA extracted from fossils. In PCR, parts of DNA can be copied billions of times in a few hours. Into the future. Technology is sure to play a role in future fossil finds. Scientists can communicate via laptop computers and satellites, which allow the public to follow excavations as they occur. Computer modeling helps scientists determine what incomplete skeletons looked like. It also helps them determine how dinosaurs and other living things once moved. Fossil finds can be combined with digitized information about modern living organisms and about environmental conditions. The model can test hypotheses or even help to formulate them. Another area of technology that may become increasingly applied to fossils is DNA testing to identify and help date fossils. This is more complicated in fossilized bone, as the genetic material can be fragmented. But with time, scientists may discover new techniques to extract better genetic information. DNA is also the basis for cloning, which as yet can only be applied to living organisms. Perhaps in the future, it can be applied to preserved remains. Activities: writing about science. Film script. Write your own version of the story of life on Earth. Include drawings, photographs, or video clips to illustrate your story. Reliving history. Think about the equipment archaeologists and paleontologists use on excavations. Think about their goals. Write a proposal to a local university or museum, asking them to fund your excavation.
Chapter 8. Population Dynamics. The Big Idea. Populations are shaped by interactions between organisms and the environment. Key Concepts. Section 8.1. Populations have many characteristics. Learn about the stages and factors that all populations have in common. Section 8.2. Populations respond to pressures. Learn how change can affect populations. Section 8.3. Human populations have unique responses to change. Learn how the responses of human populations are different from responses of other populations. 8.1. Key Concept. Populations have many characteristics. Before you learned, species change over time. Evolution is a process of change. A habitat is an area that provides organisms with resources. Now you will learn about stages in population dynamics, about variables that define a population, about changes that affect populations. Think about: how fast can a population grow? How big can a population grow? Suppose you started with a pair of fruit flies. That single pair can lay and fertilize 200 eggs. In 3 weeks, each pair from that batch could produce 200 flies of its own, producing up to 20,000 flies. Assume all eggs hatch, an event highly unlikely in the real world. After 3 weeks, 2 million fruit flies would be buzzing around the area. After just 17 generations, given ideal conditions, for the fruit fly that is, the mass of fruit flies would exceed the mass of planet Earth. Populations go through 3 stages. Look closely at the fruit flies above. As a group of the same species living together in a particular area, they represent a population. The particular area in which a scientist studies a population may be as large as a mountain range or as small as a puddle. Scientists study how populations of organisms change as they interact with each other and the environment. Over time, the number of individuals in a population changes by increasing or decreasing. Population dynamics is the study of why populations change and what causes them to change. In this chapter, you will learn about some of the important observations scientists have made about populations. Check your reading. What is population dynamics? One species of iguana may have several populations living on different islands. As a result, these iguana populations don't interact with each other. Yet there may be other populations of iguanas living on the islands made up of a different species. Growth, stability, and decline. As different as populations may be, Whether cacti, finches, dragonflies, or iguanas, all populations go through the same three stages of change: growth, stability, and decline. All living things need resources, such as water, energy, and living space. Populations get their resources from the environment. However, the area a population occupies can support only so many individuals. Carrying capacity is the maximum number of individuals an ecosystem can support. When a habitat contains enough resources to meet the needs of a population, the population grows rapidly. This growth stage of a population tends to be brief. On a graph, it looks like a sharp rise. The growth stage is followed by a period of stability, when the size of a population remains constant. For most populations, the stability stage is the longest stage of a population's existence. The stability stage is often followed by a decline in population size. During the growth stage, populations can increase according to two general patterns. One pattern is rapid growth, which increases at a greater and greater rate. Another pattern is gradual growth, which increases at a fairly steady rate. The two graphs below show the two different types of growth. Darwin's observations of population growth. In Chapter 1, you read about the observations and conclusions made by the naturalist Charles Darwin. In his book, On the Origin of Species, Darwin included important observations about population growth. All populations are able to grow rapidly. Populations tend to remain constant in size. There are limits to natural resources. Within a given population, there is genetic variation. Darwin recognized that organisms in most species have the ability to produce more than two surviving offspring. He knew that if there were no limits to growth, then populations would grow rapidly. However, Darwin also observed that in the real world, there are natural limits to growth, so populations tend to stabilize. In order for a species to continue, individuals must be replaced as they die. This means that, on average, one member of a population must produce one surviving offspring. If the birth rate doesn't match the death rate, a population can decline until it becomes extinct. 4 characteristics define a population. When scientists think about population dynamics, they consider 4 major characteristics. These characteristics include population size, population density, population spacing, and age structure. Population size. Population size is the number of individuals in a population at a given time. Even when the population size appears to be stable over time, changes can occur from year to year or from place to place. Population size varies from one habitat to another. It also varies within a single habitat. An area where the summers are hot and the winters are cold is a good place to observe how population size might change at different times of year. For example, the population sizes of many insects change within a year. Mosquitoes that are all around you on warm summer evenings are nowhere in sight when the temperatures fall below freezing. The size of plant populations can also change during the year. In the spring and summer, you can see flowering plants across the deserts, woods, and mountains of North America. However, by fall and early winter, when there is less rainfall and temperatures drop, many of these plants die. Below is a picture of a southwestern desert in full bloom. During the springtime months of March through May, many deserts in the United States experience a change. There is a period of rapid growth as a variety of wildflowers begins to bloom. The availability of resources, such as water, increases plant growth. By summer, the change in season brings higher temperatures and less rainfall. As a result, desert wildflowers experience a rapid decline in their population size. Check your reading. What are 2 factors that affect population size? Population density. Population density is a measure of the number of individuals in a certain space at a particular time. Population density is related to population size. If a population size increases and all of the individuals remain in the same area, then population density increases too. There are more individuals living in the same amount of space. If the size of a population in a particular area decreases, density also decreases. Some species, such as bumblebees or mice, live in populations with high densities. Other species, such as blue herons or wolves, live in populations with low densities. Check your reading. What is the difference between low density and high density? The distribution of a population across a large geographic area is its range. Within that range, population density may vary. For example, there may be more grasshoppers in the middle of a prairie than there are at the edges. The population density tends to be higher where more resources are available. Habitats located in the middle of a population range tend to have a greater population density than habitats located at the edges. Check your reading. How might population density vary within a range? Population spacing. Take a look around you as you walk through a local park. You might notice many flowers growing in open sunny spots, but few beneath the shade of large trees. The pattern in which the flowers grow is an example of population spacing. Scientists have observed 3 distinct patterns of spacing: clumped, uniform, and random. In clumped spacing, individuals form small groups within a habitat. Animals like elephants clump because of their social nature. Clumping can also result from the way resources are distributed throughout a habitat. Salamanders that prefer moist, rotten logs may be clumped where logs have fallen in their habitat. Some individuals live at a distance from each other. These individuals are uniformly spaced. Many plants that grow too close together become evenly spaced as individuals die out. Uniform spacing can protect saguaro cacti from competing for important resources in the desert. Individuals that aren't uniform or clumped space themselves randomly. Dandelions, for instance, grow no matter where other dandelions are growing. Age structure. Scientists divide a population into 3 groups based on age. Post-reproductive. Organisms can no longer reproduce. Reproductive. Organisms capable of reproduction. Pre-reproductive. Organisms not yet able to reproduce. The age structure of a population affects how much it can grow. On the graph below, the post-reproductive age range for humans is over 45. Reproductive is 14 to 44 years of age, and pre-reproductive is 0 to 14. Scientists can predict population change. Scientists use these 4 factors, size, density, spacing, and age structure, to describe a population and to predict how it might change over time. Sometimes a population changes when a particular factor changes. A population can change in response to its surroundings. Suppose a population of frogs is living in a pond where the water becomes saltier. Only those frogs that can survive in an environment with more salt will survive. Thus, the population size of frogs will probably decrease as a result of the changing conditions. By looking at population size, scientists can predict how changes affect the population. Scientists can also predict change by looking at the distribution of population. The story of the European starling provides a dramatic example of how the movement of organisms into or out of an area affects a population. In 1890, the first starlings were introduced to the United States in New York City's Central Park. Their numbers went from 60 individuals to about 200 million in just over 100 years as they expanded on the North American continent. The population of starlings rose as starlings moved into new habitats that had the resources they needed. Today's large populations of starlings can still be found across the North American continent. Even within a given habitat, the population can vary. In Central Park, for example, you can find starlings in clumps, uniformly spaced, or randomly spaced. 8.1 Review. Key concepts. 1. Describe the 3 stages of population growth. 2. Make a chart showing the 4 factors that affect population dynamics and an example of each. 3. Give an example of how a shift in age distribution can affect population growth. Critical thinking. 4. Apply. Choose a population in your neighborhood. Describe its population spacing. Is it clumped, uniformly spaced, or randomly spaced? 5. Compare-contrast. How is population size related to population density? Your answer should mention area. Challenge. 6. Predict. Explain how a heavy thunderstorm might affect the population density of birds living in the area. 8.2. Key concept. Populations respond to pressures. Before you learned, 4 characteristics are used to describe a population. Scientists study these 4 characteristics to predict population change. Now you will learn about limits to population growth, how population density affects limiting factors, about 2 reproductive strategies found within populations. Population growth is limited. No population can grow forever. Every population has a limit to its growth. For example, the cockroach has been around for more than 300 million years. This insect has outlived the dinosaurs and may persist long after humans have become extinct. Yet even if cockroaches became the only species on the planet, several factors would limit their population size. Birth, death, immigration, and emigration. When scientists study how a population changes, they must consider 4 things: birth, death, immigration, and emigration. There is even a simple formula to help scientists track population change. Population change equals birth plus immigration minus death plus emigration. It is too simple to say that a high birth rate means population growth, or that many deaths mean population decline. Immigration is the movement of individuals into a population. For example, if a strong wind blows the seeds of a plant from one area into another, the new plant would be said to immigrate into the new area. Immigration can increase a population or help stabilize a declining population. Birth and immigration introduce individuals into a population. Emigration is the movement of individuals out of a population. If resources become scarce within a habitat, some of the individuals might move to areas with greater supplies. Others may even die. Death and emigration remove individuals from a population. Check your reading. List 2 factors that lead to population growth and 2 that lead to population decline. Consider, for example, a flock of seagulls that flies inland during a storm. They stop at a city dump where food is plentiful. These incoming seagulls become part of the seagull population that is already living at the dump. A raccoon population living in the same area has been eating the seagull's eggs, causing the number of seagull births to decrease. If enough seagulls immigrate to the dump, the seagull population would increase, making up for the decrease in births. Immigration would help keep the population stable. The seagull population would also increase if part of the raccoon population moved away. Limiting factors. When a population is growing at a rapid rate, the birth rate is much higher than the death rate. That means that more individuals are being born than are dying during a particular time period. There are plenty of resources available, and the population size is increasing rapidly. Eventually, however, the population will stop growing because a habitat can support only a limited number of organisms. A limiting factor is a factor that prevents the continuing growth of a population in an ecosystem. Abiotic or non-living limiting factors include air, light, and water. Other limiting factors can be living things, such as other organisms in the same population or individuals belonging to different species within the same area. Check your reading. What are 2 limiting factors? Competition can occur between different populations sharing the same habitat. Competition can also occur among individuals of the same population. Suppose, for example, that a population of deer in a forest preserve were to increase, either through births or immigration. Population density at the forest preserve would go up, More and more deer in the area would be competing for the same amount of food. Effects of population density. In the situation described above, the seagull population could decrease as a result of competition for food. Competition is an example of a density-dependent factor. That is, a limiting factor that affects a population when density is high. Disease is another density-dependent factor. The more crowded an area becomes, the easier it is for disease to spread, so more individuals are affected. If population density is low, there is less contact between individuals, which means that disease will spread more slowly. Density-dependent factors have a greater effect on the population as it grows. They can bring a population under control because they apply more pressure to a growing population. There are also density-independent factors. These limiting factors have the same effect on a population, whether it has a high density or a low density. Freezing temperatures could be considered a density-independent factor. A freeze might kill all of the flowering plants in an area, whether or not the population density is high. A natural event such as a wildfire is another example of a density-independent factor. When a wildfire occurs in a forest, it can wipe out an entire ecosystem. Check your reading. How are limiting factors that are density-dependent different from limiting factors that are density-independent? Limiting factors include non-living factors in the environment and natural events such as earthquakes, fires, and storms. During times of drought, there may not be enough food to meet the needs of all the organisms in an area. The quality of the food declines as well. For example, a lack of water may cause a population of trees to produce fewer pieces of fruit, and the fruit itself may be smaller. If there is little food available, a condition called famine arises. If the famine is severe, and if death rates exceed birth rates, then the population size will fall dramatically. Check your reading. How do limiting factors affect populations? Remember, a summary includes only the most important information. Limiting factors affect human populations as well. However, humans have found different ways to help overcome many of these limits. In Section 3.3, you will read about how the human response to limits differs from that of other biological populations. Populations have distinct reproductive survival strategies. Although reproduction of offspring is not necessary for the survival of an individual organism, it is necessary for the survival of a species. Scientists studying populations observe patterns in the reproductive strategies used among species. There are two main strategies that many species use. There are also many species whose strategies fit somewhere in between. Strategies of opportunists. Opportunists are species that reproduce rapidly if their population falls below carrying capacity. They share many characteristics, including a short lifespan and the ability to reproduce large quantities of offspring. Their population size tends to change often, and opportunists live across many areas. Opportunists include algae, dandelions, bacteria, and insects. These species can reproduce and move across an area quickly. In addition, they can adapt quickly to environmental changes. Populations of opportunists often grow rapidly. Strategies of competitors. You might be familiar with the term competitor as meaning an organism that struggles with another to get resources. Scientists who study population growth use the term competitor in another way. Competitors are species with adaptations that allow them to remain at or near their carrying capacity for long periods of time. Competitors have many characteristics that differ from those of opportunists. Species that have a competitive reproductive strategy often live longer and have fewer offspring. Elephants and saguaro cacti are two examples of competitors. The offspring of competitors take longer to develop than those of opportunists. Also, animals with this strategy tend to take care of their young for a longer period of time. Competitors are not distributed across areas as widely as opportunists, but greater numbers of their offspring survive to reproductive age. 8.2. Review. Key concepts. 1. What 4 factors do scientists consider when they measure population change? 2. Give 2 examples of density-dependent factors and 2 examples of density-independent factors. 3. Other than lifespan, how do opportunists and competitors differ? Critical thinking. 4. Analyze. Why would it be a mistake to predict population growth based on birth rate alone? 5. Apply. Give an example of a factor that limits a population near you. Challenge. 6. Synthesize. There has been an oil spill along a waterway famous for its populations of seals, dolphins, and seabirds. 6 months later, all populations show a decline. Explain what factors might have caused such a change, and whether the oil spill was a density-dependent or density-independent factor, or both. Science on the job. Fishing boat captain. Studying the schools. There's a lot more to catching fish than putting a net or a line into the ocean. More and more these days, finding fish means looking at the big and changing picture of fish populations. Once you start fishing, you need to know when to stop fishing, as well as how to protect the other organisms in the ocean environment. Density. If there are too few fish of a particular species in one area, it is best to leave them and look for a place with greater population density. By using the most suitable gear, the captain avoids killing or injuring fish and other animals that shouldn't be part of the catch. Distribution. Some fish live alone, and some live in big groups, called schools. Some, called groundfish, stay on the bottom, while others swim near the surface. One way to see the distribution of fish is with sonar. An image made from sonar shows that a school of hake swims at a depth of about 320 meters, 1,050 feet. Partners in research. Many boat captains set up partnerships with researchers to study fish and to help them to thrive. Sonar equipment is a tool shared by scientists and commercial fishers. Images are made by sending sound waves through the water and receiving the patterns of sound waves that bounce or echo back. Explore. 1. Infer. Boat captains talk to each other about how many fish they catch and where and when they catch them. What are 3 reasons why this would be important? 2. Challenge. Suppose a fishing boat captain overfishes an organism that is a source of food for another organism. Describe what may happen to the other organism. 8.3. Key concept. Human populations have unique responses to change. Before you learned, over time, all populations stop growing. All populations are affected by limiting factors. Reproductive strategies include opportunism and competition. Now you will learn how human populations differ from other populations, how humans adapt to the environment, how human populations affect the environment. Human populations differ from populations of other species. Humans are not the fastest or the largest organisms on Earth. They must get food from other organisms. Humans have a limited sense of smell, and the vision of a human is inferior to that of a hawk. However, the human population now dominates our planet. Why? Humans are able to shape their environment. Humans are also able to determine their own biological reproduction. Because humans can control many factors that limit growth, Earth's carrying capacity for humans has increased. 2 key factors that have increased Earth's carrying capacity for humans are habitat expansion and technology. Habitat expansion. Individuals who study the history of ancient peoples know that populations of humans have spread throughout the world. Discoveries of ancient human tools and skeletons indicate that the first human populations lived on the continent now known as Africa. Over time, human populations have spread over nearly the entire planet. The word habitat refers to a place where an organism can live. Humans have expanded their habitats, and thus the population has grown. Humans can survive in many different environments, by adding air conditioning or heat to regulate indoor temperature. They can design and build shelters that protect them from harsh environments. Technology. Limited resources and environmental conditions, such as climate, do not affect human population growth the way they do the growth of other biological populations. Humans have found ways to fit themselves into almost every climate by altering their clothing, shelter, diet, and means of transportation. Scientific discoveries and the advances of technology, such as improved sanitation and medical care, have increased the standard of living and the life expectancy of many humans. Important goods, such as food and shelter, are manufactured and shipped around the world. Water, which is a limited resource, can be transported through pipes and dams to irrigate fields or reach normally dry areas. Water can also be purified for drinking or treated before it is released back into the environment. Check your reading. How does technology help humans get resources they need for survival? Human populations are growing. As you've read, humans have developed solutions to many limits on growth. These solutions have allowed the human population to grow rapidly. Scientists are studying the history of this growth and trying to predict whether it will continue or change. History of human population growth. Until about 300 years ago, the human population grew slowly. Disease, climate, and the availability of resources limited population size. Most offspring did not survive to adulthood. Even though birth rates were high, death rates were also high. Notice the human population on the graph below. Many historical events have affected its growth. For example, the development of agriculture provided humans with a more stable food source. This, in turn, helped support human population growth. Today, populations across many parts of the world are increasing rapidly. Scientists identify three conditions that allow for rapid growth: the availability of resources, lack of predators, and survival of offspring to reproductive age. As these conditions change, so does the population. Population projections. To help prepare for the future, scientists make predictions called population projections. Population projections forecast how a population will change, based on its present size and age structure. Population projections provide a picture of what the future might look like. Using population projections, government agencies, resource managers, and economists can plan to meet the future needs of a population. In addition to population size and age structure, scientists making population projections consider other factors. These factors include the ages of individuals having children. The average number of offspring produced by an individual also affects projections. In addition, life expectancy and health in a particular population affect population growth. The factors affecting population growth vary from society to society. The human population in the African country Botswana provides an example of how disease and health can affect population growth. In some African countries, death rates due to HIV/AIDS have lowered population projections for the year 2015 by almost 18%. Botswana's population will decline because more than 30% of adults are infected with HIV/AIDS. So many people in Botswana have already died of HIV/AIDS that the average life expectancy has dropped from 63 years of age in the late 1980s to 32 years in 2003. Consider the impact this will have on the population's age structure. Because many people who die from HIV/AIDS are in their reproductive years, the long-term effects on population growth will be significant. Check your reading. What factors do scientists consider when they make population projections? Human population growth affects the environment. You have read that extinction of species is a part of the history of life on Earth. The ways a population uses and disposes of resources have a great impact on local and global environments. As the human population continues to grow and use more resources, it contributes to the decline and extinction of other populations. Some scientists estimate that over 99% of the species that have ever existed on Earth are now extinct. Most of these species vanished long before humans came on the scene. However, some experts are concerned that human activity is causing other species to become extinct at a much higher rate than they would naturally. Human populations put pressure on the environment in many ways, including introduction of new species, pollution, overfishing. Introduction of new species. Travelers have introduced new species to areas both on purpose and by accident. Many species introduced to an area provide benefits, such as food or beauty. Some species, however, cause harm to ecosystems. One example of an introduced species is the zebra mussel. An ocean vessel accidentally released zebra mussels from Europe into the Great Lakes region of the United States. With no natural predators that consume them, the mussels have reproduced quickly, invading all of the Great Lakes, the Mississippi River, and the Hudson River. The mussels compete with native species for food and affect water quality, endangering the ecosystem. Kudzu is another introduced species. In the 1930s, kudzu was used in the southeastern United States to keep soil from being washed away. The plants, which have beautiful purple flowers, were imported from Japan. Starch made from kudzu is also a popular ingredient in some Asian recipes. However, populations of the kudzu vines planted in the United States have grown too far and too fast. Kudzu grows as much as 0.3 meters, about a foot, per day, killing trees and other plants living in the same area. Pollution. While human activities might cause some populations to decline, they can also cause other populations to grow. Sometimes this population growth causes pollution and habitat disturbance. Pollution is the addition of harmful substances to the environment. One example of such an activity is large-scale hog farming. Human demands for pork, combined with a growing human population, have caused the hog farming industry to expand. Between 1987 and 2001, the hog population in North Carolina grew from 2.6 million to 10 million. These 10 million hogs produced more than 50,000 tons of waste each day. Wastes from large populations of hogs affect water supplies, soil, and air quality. Pollution has also affected the Salton Sea in southeastern California. The growing demand for goods and agriculture has led to chemical dumping from industries and pesticide runoff from nearby farms. The rivers that run into the lake carry high levels of harmful chemicals, such as DDT. Local birds that live and feed in this area have weakened shells that cannot support baby birds. Pollution has also caused fish to become deformed. Overfishing. Fish and crustaceans, such as shrimp and lobsters, have long been an important food source for many people. In the 1900s, the techniques and equipment that fishers used allowed them to catch so many fish that fish populations began to decrease. As the human population has continued to grow, so has the demand for fish. However, if fish do not survive long enough in the wild, they do not have the chance to reproduce. Many species have been so overfished that their populations may not recover. Lobster fishing in particular has supported coastal communities in the northeastern United States for generations. But the demand for this food source has caused populations to decline. Areas that fishers trapped for years may now have only a small population of lobsters, and the lobsters fishers are catching may not be as large as those from earlier decades. In order to help lobster populations recover, laws have been enforced to protect their life cycle and reproduction. Today, people who trap lobsters are required to release females with eggs. They are also allowed to keep only mature lobsters. Younger lobsters are returned to the waters to mature and reproduce. Efforts like these help protect the lobster population and secure the jobs of fishers by helping fish populations remain stable. Check your reading. Describe how overfishing would affect resources. 8.3 Review. Key concepts. 1. What factors, other than birth, death, immigration, and emigration, must scientists consider when making projections of human population? 2. Give an example of how Earth's carrying capacity for humans has increased. 3. What are 3 ways that humans affect other populations? Critical thinking. 4. Infer. Consider the effect of HIV/AIDS on Botswana's human population. How might age structure affect Botswana's population growth? 5. Analyze. Do you think it is possible to predict the maximum number of humans that Earth can support? Why or why not? Challenge. 6. Apply. Identify a challenge faced by the human population in your state. Explain how the challenge is related to pollution, introduction of new species, habitat disturbance, or overfishing. Frontiers in Science. Chilling changes. How do organisms survive in this chilly Arctic landscape? Scientists are studying how organisms have adapted to the extreme cold of Earth's northern climates. When life chills out. When faced with the approach of winter, some animals hurry up and some slow down. You may have seen animals getting a move on, migrating to warmer places. Other animals stay alive at home by hibernating or entering a sleep-like state. Hibernation conserves energy until spring by slowing down many body processes. But what happens when an organism's body temperature dips below freezing? Ice formation inside an organism's body is one of the most serious and potentially damaging threats to winter survival. If ice forms inside cells, it can damage the cell's operation and tear holes in the cell membrane, almost certainly leading to death. Some animals and plants take cold weather survival to an extreme. Some frogs and insects can allow their bodies to freeze solid. Some fish have fluids in their bodies that can cool below the freezing point of water and still flow. How do they do it? Scientists studying survivors of extreme cold are not just learning about the ways organisms respond in the wild. Research on cold weather survival helps scientists understand problems faced by human society. Growing crops year-round and using bacteria to clean up pollution in a frozen environment are two examples. There are two main responses that allow living things to live in a sub-freezing world. Avoid ice or tolerate it. Organisms can either avoid the forming of ice or tolerate it. Some animals, like fish called a flounder, produce antifreeze proteins in their bodies. The proteins keep ice from forming in the blood and other fluids. The fluids thicken but still flow. They never turn to ice crystals. Scientists call this response freeze avoidance. Other organisms, including some frogs, many types of insects, and some trees, do allow ice to form inside their bodies. In these organisms, ice forms, but only in the spaces outside the cells. Freeze-tolerant organisms pump fluid out of their cells. Specialized proteins, located outside of the cells, encourage ice to form there, rather than inside the cells. Perhaps the champion of freeze tolerance is a small Arctic insect called the Arctic woolly bear caterpillar. It is only active for a few weeks out of the year, during the brief Arctic summer. It can take this caterpillar up to 15 years to mature into a moth. Temperatures during Arctic winters often fall as low as -70 degrees Celsius. Because the ground is frozen year-round, the caterpillars cannot burrow. Instead, their bodies freeze solid in winter, shutting down nearly all of their cellular activity and expending almost no energy. Scientists have known for centuries that some insects can freeze over the winter, but only recently have they begun to understand exactly how insects' bodies change to allow them to freeze and thaw. Scientist Ken Story has discovered a set of genes that turn on, like a switch, in response to freezing temperatures. These genes are called master control genes because they have the ability to turn other genes in the body on or off. In the case of freeze-tolerant animals, master control genes first turn off nearly all the genes in the body. Arctic woolly bear caterpillars have many thousands of genes. But during the winter, most of those genes are turned off. Keeping genes turned off is an adaptation that helps the caterpillar conserve energy throughout the winter. Is it really extreme? Organisms have adapted to a wide variety of environments. The Arctic woolly bear caterpillar is suited to life near the Arctic Circle. Some organisms must be able to survive in the exact opposite, the extreme heat of deserts. Others thrive in extremely salty conditions. Some plants and bacteria can even live in places with dangerously high levels of poisonous chemicals. An environment doesn't have to be extreme to present challenges to the organisms that live there. Animals in a forest or meadow still need to find food and shelter, avoid predators, and respond to changes. The organisms living in any environment face a unique set of challenges. Unanswered questions. As scientists learn more about how animals survive in extremely cold climates, they also uncover additional questions. Do animals respond to temperature or a lack of light? How do cellular processes that halt during the winter get started up again? Can scientists grow freeze-resistant crops?
Chapter 9. Single-celled organisms and viruses. The big idea. Bacteria and protists have the characteristics of living things, while viruses are not alive. Key concepts. Section 9.1. Single-celled organisms have all the characteristics of living things. Learn about characteristics shared by all living things. Section 9.2. Bacteria are single-celled organisms without nuclei. Learn about the characteristics of bacteria and archaea. Section 9.3. Viruses are not alive, but affect living things. Learn about the structure of viruses and how they affect cells. Section 9.4. Protists are a diverse group of organisms. Learn about protists and how they affect the environment. 9.1. Key concept. Single-celled organisms have all the characteristics of living things. Before, you learned, all living things are made of cells. Organisms respond to their environment. Species change over time. Now you will learn about the various sizes of organisms, about characteristics that are shared by all living things, about needs shared by all organisms. Living things come in many shapes and sizes. You can spot mushrooms in many places while walking through a forest. Scientists have discovered mushrooms that come from the same individual fungus more than 5 kilometers, 3 miles, apart in an Oregon forest. Most of this honey mushroom fungus is below ground, stretching over an area covering more than 1,600 football fields. This mushroom is one of the largest known living things on Earth. Many other living things share the soil in the Oregon forest. Earthworms, insects, and many other organisms that are too small to be seen with the naked eye also live there. For every living thing that is large enough to be seen, there are often countless numbers of smaller living things that share the same living space. The honey mushroom fungus is one example of an organism. You too are an organism, and tiny bacteria living inside your body are also organisms. In fact, any living thing can be called an organism. When you think of living things, you probably begin with those you can observe: plants, animals, and fungi such as mushrooms. However, most living things are too small to observe without a microscope. Even the tiniest organisms are made of cells. Very small organisms are called microorganisms. Many microorganisms are made of just one cell. Check your reading. Compare and contrast the words microorganism and organism. A visitor to a mangrove swamp forest can find an amazing variety of organisms. The mangrove trees themselves are the most obvious organisms. Roots from these trees grow above and below the muddy bottom of the forest. Other organisms live in almost every part of the mangrove tree. A single drop of water from a mangrove swamp may be living space for many microorganisms. The circled photograph on page 282 was taken using a microscope and shows an amoeba that may be found in the water of the swamp. Larger organisms, such as manatees and fish, swim around the roots of mangrove trees. Birds, such as tricolored herons and roseate spoonbills, use the branches. Scientists divide the organisms they identify into groups called kingdoms. This unit will cover all of the kingdoms of life, listed in the table on page 282. You are already familiar with plants and animals. Fungi are another kingdom. Fungi include mushrooms found in a forest. The other three kingdoms are composed of mostly microscopic life. You will learn more about microscopic organisms later in this chapter. Living things share common characteristics. All living things, from the microorganisms living in a mangrove swamp to the giant organisms living in the open ocean, share similar characteristics. Living things are organized, grow, reproduce, and respond to the environment. Organization. Cells are the basic unit of all living things. Cells, like all living things, have an inside and an outside. The boundary separating the inside from the outside of an individual cell is called the cell membrane. Within some cells, another structure called the nucleus is also surrounded by a membrane. In this chapter, you will read about organisms made of a single cell. Some types of single-celled organisms contain a nucleus, and some do not. All single-celled organisms contain everything they need to survive within their one cell. These cells are able to get energy from complex molecules, to move, and to sense their environment. The ability to perform these and other functions is part of their organization. Growth. Living things increase in size. Organisms made of one cell do not grow as large as organisms made of many cells. But all living things need to get energy. All living things also need to obtain materials to build new structures inside cells or replace worn-out cell parts. As a result, individual cells grow larger over time. Reproduction. Living things reproduce, forming other organisms like themselves. Every organism contains genetic material, which is a code contained in a special molecule called DNA. The code contains characteristics of the individual organism. In order to reproduce, an organism must make a copy of this material, which is passed on to its offspring. Some single-celled organisms reproduce by a process called binary fission. In binary fission, material from one cell separates into two cells. The genetic material of the original cell first doubles so that each daughter cell has an exact copy of the DNA of the original cell. You might say that single-celled organisms multiply by dividing. One cell divides into two cells. Two cells divide into 4, 4 into 8, 16, 32, 64, and so on. In some cells, binary fission can take place as often as every 20 minutes. Check your reading. Describe how a single-celled organism is organized, grows, and reproduces. Response. Organisms respond to changes in the environment. Even microscopic organisms respond to conditions such as light, temperature, and touch. The ability to respond allows organisms to find food, avoid being eaten, or perform other tasks necessary to survive. Living things need energy, materials, and living space. Have you ever wondered why you need to eat food, breathe air, and drink water? All living things need energy and materials. For most organisms, water and air are materials necessary for life. Food supplies you with energy. You, like all living things, need energy to move, grow, and develop. All animals have systems for breaking down food into usable forms of energy and materials. Plants have structures that enable them to transform sunlight into usable energy. Some microorganisms transform sunlight, while others need to use other organisms as sources of energy. Most of the activities of living things take place in water. Water is also an ingredient for many of the reactions that take place in cells. In addition, water helps support an organism's body. If you add water to the soil around a wilted plant, you will probably see the plant straighten up as water moves into its cells. Materials in the air include gases such as carbon dioxide and oxygen. Many of the processes that capture and release energy involve these gases. Some organisms, such as those found around hydrothermal vents, use other chemicals to capture and release energy. Viruses are not alive. Sometimes it's not easy to tell the difference between a living and non-living thing. A virus has genetic material enclosed in a protein shell. Viruses have many of the characteristics of living things, including genetic material. However, a virus is not nearly as complex as a cell and is not considered a living thing. For example, animal cells have structures that allow them to get materials or energy from their environment. Virus particles do not grow once they have formed, and they do not take in any energy. Animal cells can make copies of their genetic material and reproduce by dividing in two. Viruses are able to reproduce only by taking over a cell and using that cell to make new viruses. Animal cells also have many more internal structures than viruses. Viruses usually contain nothing more than their genetic material and a protein coat. 9.1 Review. Key concepts. 1. Give examples of organisms that are very large and organisms that are very small. 2. Name 4 characteristics that all living things share. 3. Name 3 things that living things must obtain to survive. Critical thinking. 4. Synthesize. Give examples of how a common animal, such as a dog, is organized, grows, responds, and reproduces. 5. Predict. In a certain lake, would you expect there to be more organisms that are large enough to see, or more organisms that are too small for you to see? Why? Challenge. 6. Design. Try to imagine the different structures that a single-celled organism needs to survive in pond water. Then use your ideas to design your own single-celled organism. 9.2 Key concept. Bacteria are single-celled organisms without nuclei. Before you learned, organisms come in all shapes and sizes. All living things share common characteristics. Living things may be divided into 6 kingdoms. Now you will learn about the simplest living things, about bacteria and archaea, that bacteria may help or harm other organisms. Think about, where are bacteria? Bacteria are the simplest form of life, but that doesn't mean they're not important or numerous. As you look about the room you're sitting in, try to think of places where you might find bacteria. In fact, bacteria are on the walls, in the air, on the floor, and on your skin. It's hard to think of a place where you wouldn't find bacteria. The photograph shows a magnification of bacteria living on a sponge. The bacteria are magnified 580 times. There are hundreds of millions of bacteria on your skin right now. And there are trillions of bacteria that live inside your intestines and help you digest food. Bacteria and archaea are the smallest living things. The names of the organisms belonging in the kingdoms archaea and bacteria are probably unfamiliar. Yet you actually encounter these organisms every day. Bacteria are everywhere, on your skin, in the ground, in puddles and ponds, in the soil, and in the sea. About 300 species of bacteria are living in your mouth right now. Bacteria are the simplest kind of life known on Earth. All bacteria are composed of just one cell without a nucleus. Their genetic material is contained in a single loop within the cell. A bacterium reproduces using binary fission. Bacterial cells are different from the cells of other organisms. A bacterial cell is about 1/10 to 1/20 the size of a typical cell from organisms such as animals, plants, fungi, or protists. These four groups include organisms made up of cells with true nuclei. The nucleus is a structure that is enclosed by a membrane and that holds the genetic material. Despite their small size, bacteria are simple only when compared with more complex cells. Bacteria are also much more complex than viruses because they have many internal structures that viruses do not have. For example, one important feature of most bacteria is a covering called a cell wall, which surrounds and protects the soft cell membrane like a rain jacket. Bacterial cells contain many large molecules and structures that are not found in viruses. Scientists often classify bacteria by their external shapes. Spiral-shaped bacteria occur in single strands. Rod-shaped bacteria may occur singly or in chains. Round-shaped bacteria may occur singly or in pairs, chains, or clusters. Check your reading. Name 2 features that all bacteria share. Archaea and bacteria are found in many environments. 2 types of single-celled organisms do not have nuclei. Bacteria are the most common and can be found in nearly every environment. Archaea are similar in size to bacteria, but share more characteristics with the cells of complex organisms like plants and animals. Archaea. Archaea are single-celled organisms that can survive in the largest range of environments. These environments may be very hot, very cold, or contain so much of a substance such as salt that most living things would be poisoned. As a result, scientists often group archaea according to where they live. Methanogens take their name from methane, the natural gas they produce. These archaea die if they are exposed to oxygen. They may live in the dense mud of swamps and marshes, and in the guts of animals such as cows and termites. Halophiles live in very salty lakes and ponds. Some halophiles die if their water is not salty enough. When a salty pond dries up, so do the halophiles. They can survive drying and begin dividing again when water returns to the pond. Thermophiles are archaea that thrive in extreme heat or cold. They may live in hot environments such as hot springs, near hot vents deep under the sea, or buried many meters deep in the ice. Bacteria. Most single-celled organisms without a nucleus are classified as bacteria. Bacteria are found in almost every environment and perform a variety of tasks. Some bacteria contain chlorophyll. Using sunlight for energy, these bacteria are an important food source in oceans. These bacteria also release oxygen gas, which animals need to breathe. Check your reading. What are some common traits of bacteria and archaea? Bacteria without chlorophyll perform different tasks. Some bacteria break down parts of dead plants and animals to help recycle matter. Some bacteria release chemicals into the environment, providing a food source for other organisms. Scientists often group bacteria by the roles they play in the environment. 3 of the most common roles are listed below. Bacteria that transform energy from sunlight into energy that can be used by cells are called producers. These bacteria are a food source for organisms that cannot make their own food. Decomposers get energy by breaking down materials in dead or decaying organisms. Decomposers help other organisms reuse materials found in decaying matter. Some bacteria live in a very close relationship either inside or on the surface of other organisms. Some of these bacteria may have no effect on their host organisms or host cells. Some bacteria help their hosts. Other bacteria are parasites, organisms that harm their hosts. Bacteria may help or harm other organisms. Some bacteria, such as producers and decomposers, are helpful to other organisms, but other bacteria can be harmful. These bacteria can cause diseases in animals and plants. Helpful bacteria. One shovelful of ordinary soil contains trillions of bacteria, and every fallen leaf or dead animal is covered with bacteria. These bacteria break down the matter in dead bodies and waste materials. These broken down materials are then available for other organisms to build their bodies. Cities use bacteria to break down sewage. Bacteria in sewage treatment plants live on the material dissolved in liquid sewage. These bacteria help make the water clean enough to be released into rivers or oceans. Other bacteria are used to clean up oil spills by decomposing oil suspended on the ocean surface. Bacteria can also change materials that do not come from living things and make them available for other organisms. For example, some bacteria can convert nitrogen gas to nitrogen compounds. This process, called nitrogen fixation, makes nitrogen available to plants in a form that is useful to them. Plants use this nitrogen in making proteins, which are an important part of every cell. Like bacteria, certain types of archaea are helpful to other organisms. Animals that eat plants, such as grass, often depend on archaea. Methanogens help break down the cellulose in cell walls. Termites and cows are two examples of animals that can digest cellulose because of the archaea in their stomachs. Check your reading. Name 2 helpful roles that bacteria can play in the environment. Harmful bacteria. Not all bacteria are helpful to other organisms. Scientists first discovered that bacteria cause some diseases in the late 1800s. Much of the scientific research into harmful bacteria developed because bacteria cause disease in humans. Tuberculosis, cholera, and infant diarrhea are examples of disease caused by bacteria. Bacteria also may cause disease in many other animals and in plants. Bacteria can cause the symptoms of disease in 3 ways. They can invade parts of the body, multiplying in body tissues and dissolving cells. They can poison the body with chemicals they produce and release. They can poison the body with chemicals that are part of the bacteria themselves. One way to fight bacterial disease is with vaccinations. Vaccines help individual organisms prepare to fight diseases they might encounter in the future. Humans, as well as cats and dogs, get vaccinations for bacterial diseases. 9.2 Review. Key concepts. 1. Explain why bacteria are classified as living cells, but viruses are not. 2. Name 2 main groups of bacteria. 3. Describe 3 ways that bacteria affect other organisms. Critical thinking. 4. Visualize. Draw a diagram of a bacterium. Label the parts of the cell. 5. Predict. Where in your neighborhood would you find most of the bacteria that cause decomposition? Challenge. 6. Analyze. Parasitic bacteria do not usually kill their hosts, at least not quickly. For a parasite, why is it better not to kill the host? 9.3 Key concept. Viruses are not alive, but affect all living things. Before you learned, most organisms are made of a single cell. Living things share common characteristics. Viruses are not living things. Now you will learn about the structure of viruses, how viruses use a cell's machinery to reproduce, how viruses affect host cells. Viruses share some characteristics with living things. In the late 1800s, scientists such as Louis Pasteur showed that some small organisms can spoil food and cause disease. Once the cause was found, scientists looked for ways to prevent spoilage and disease. One method of prevention they found was removing these harmful organisms from liquids. Bacteria may be removed from liquids by pouring the liquid through a filter, like a coffee filter or a sieve. To remove bacteria, a filter must have holes smaller than 1/1,000,000 of a meter in diameter. When a filter had removed all of the harmful organisms from a liquid, the liquid no longer caused any illnesses. This method worked when there was only bacteria in the liquid. Sometimes filtering did not prevent disease. Something much smaller than bacteria was in the liquid. Scientists call these disease-causing particles viruses, from the Latin word for slimy liquid or poison. Check your reading. How does the size of viruses compare with the size of bacteria? Scientists have learned much about viruses and can even make images of them with specialized microscopes. Viruses consist of genetic material contained inside a protective protein coat called a capsid. The protein coat may be a simple tube, such as the coat of an Ebola virus, or have many layers, such as the smallpox virus shown on page 26. Viruses may come in many shapes and sizes, but all viruses consist of a capsid and genetic material. Viruses are able to use living cells to get their DNA copied and so reproduce new viruses, a characteristic that makes them similar to living things. Also, the protein coat is similar to a cell's outer membrane, but viruses do not grow, and viruses do not respond to changes in their environment. Therefore, viruses are not living organisms. Viruses multiply inside living cells. Remember that all living things reproduce. Viruses cannot reproduce by themselves, which is one of the ways they are different from living things. However, viruses can use materials within living cells to make copies of themselves. The cells that viruses infect in order to make copies are called host cells. Despite their tiny size, viruses have the ability to cause a lot of damage to cells of other organisms. One of the best studied viruses infects bacteria. It's called a bacteriophage, which comes from the Latin for bacteria eater. Some of the steps that a bacteriophage goes through to multiply are shown in the illustration. 1. Attachment. The virus attaches to the surface of a bacterium. 2. Injection. The virus injects its DNA into the bacterium. 3. Production. Using the same machinery used by the host cell for copying its own DNA, the host cell makes copies of the viral DNA. 4. Assembly. New viruses assemble from the parts that have been created. 5. Release. The cell bursts open, releasing 100 or more new viruses. Viruses have proteins on their surfaces that look like the proteins that the host cell normally needs. The virus attaches itself to special sites on the host that are usually reserved for these proteins. Not every virus makes copies in exactly the same way as the bacteriophage. Some viruses are inside host cells. Others use the host cell as a factory that produces new viruses one at a time. These viruses may not be as harmful to the infected organism because the host cell is not destroyed. Viruses may harm host cells. A host cell does not often benefit from providing living space for a virus. The virus uses the cell's material, energy, and processes. In many cases, after a virus has made many copies of itself, the new viruses burst out of the host cell and destroy it. Harmful viruses cause huge problems. Viruses that cause diseases such as polio, smallpox, diphtheria, or AIDS have had a major impact on human history. About 25 million people died of influenza in an outbreak that occurred just after World War I. In the photograph, nurses worked to ease the symptoms of infected patients. The most infectious patients were enclosed in tents. Others were made as comfortable as possible on beds outside. Since viruses such as influenza can spread quickly, the camp was isolated from the rest of the community. Plant viruses can stunt plant growth and kill plants. When plant viruses invade crop plants, they can cause much economic damage, decreasing food production. Plants, animals, bacteria, and all other living things are capable of being infected by viruses. Today, scientists are discovering ways to use viruses in a positive way. Scientists use viruses to insert certain pieces of genetic material into living cells. For example, The portion of genetic material that allows some marine organisms to produce a chemical that glows can be inserted into tissue samples to help with their identification. 9.3 Review. Key concepts. 1. What are the two parts that every virus has? 2. Why are viruses not considered to be living things? 3. Explain how copies of viruses are produced. Critical thinking. 4. Compare and contrast. What features do viruses and cells have in common? How are they different? 5. Explain. Summarize the steps by which a bacteriophage makes new viruses. Challenge. 6. Synthesize. What characteristics of viruses can make them so dangerous to humans and other living organisms? Extreme science. Tulipomania. The virus and the tulip. The people of Holland around the 1620s were trading fortunes and farmland for one beautiful flower. Tulips had arrived from Turkey by way of Vienna, and interest in the flower spread through Holland like a fever. In a frenzy called tulipomania, collectors paid as much as 5,400 guilders, the price of a city house, for just one tulip bulb, but not any ordinary tulip. Broken flower bulbs. Tulip traders searched for tulips with patterns, stripes, or feathery petals. These plants were called broken bulbs. Within a field of colored tulips, suddenly an odd or patterned flower grew. Once a color break showed up, it stayed with that flower's line into each new generation until the line died off. And died off it did. The patterned petals were caused by a virus inside the plant, and the virus caused the flowers to weaken. The blooms got smaller in each generation. The mystery source. While the trade in tulips rose, growers tried many tricks to produce the crazy patterns. Still, the broken bulbs grew rarely and randomly, or so it seemed. Viruses weren't discovered until 300 years later. Scientists then figured out that a virus had caused the broken bulbs. Back down to the ground. Like any gold rush, tulipomania crashed. People lost fortunes and fought over claims. In 1637, the government stopped all trading. Today, tulips with striped and feathery patterns grow around the world, but the patterns observed are not caused by viruses. Still, every now and then, a strange-looking bulb appears. Instead of prizing it, growers remove it. They don't want a field of virus-infested, weakened plants. Explore. 1. Observe. Look at tulips in a garden catalog and find breeds with patterns. Observe the tulip closely. Draw or paint the modern flower and label its name. 2. Challenge. Viruses can produce sickness, but they have other effects too. Do research on viruses to list some effects of viruses that have value to scientists, doctors, or other people. 9.4 Key concept. Protists are a diverse group of organisms. Before, you learned organisms are grouped into 6 kingdoms. Bacteria are single-celled organisms without a nucleus. Viruses are not living things. Now you will learn about characteristics of protists, about the cell structure of protists, how protists get their energy. Think about, where can protists be found? Protists include the most complex single-celled organisms found on Earth. 50 million years ago, a spiral-shelled protist called a nummulatid was common in some oceans. Even though its shell was the size of a coin, the organism inside was microscopic and single-celled. When the organism died, the shells accumulated on the ocean floor. Over millions of years, the shells were changed into the rock called limestone. This limestone was used to build the Great Pyramids of Egypt. Some of the most monumental structures on Earth would not exist without organisms made of just a single cell. Most protists are single-celled. When Anton von Leeuwenhoek began using one of the world's first microscopes, he looked at pond water, among other things. He described, in his words, many very little animalcules. Some of the organisms he saw probably were animals, microscopic but multicellular animals. However, many of the organisms Leeuwenhoek saw moving through the pond water had only a single cell. Today, more than 300 years later, scientists call these single-celled organisms protists. Protists include all organisms with cells having nuclei and not belonging to the animal, plant, or fungi kingdoms. In other words, protists may be considered a collection of leftover organisms. As a result, protists are the most diverse of all the kingdoms. Most protists are single-celled microscopic organisms that live in water. However, protists also include some organisms with many cells. These many-celled organisms have simpler structures than animals, plants, or fungi. They also have fewer types of cells in their bodies. Check your reading. Why are protists considered the most diverse group of organisms? The group of protists you're probably most familiar with is seaweeds. At first glance, seaweed looks like a plant. On closer inspection, scientists see that it has a simpler structure. Some seaweeds, called kelp, can grow 100 meters long. The name algae applies to both multicellular protists and single-celled protists that use sunlight as an energy source. Both seaweed and diatoms are types of algae. Slime molds are another type of multicellular protist. Given the many different types of organisms grouped together as protists, it is no surprise that protists play many roles in their environments. Algae are producers. They obtain energy from sunlight. Their cells provide food for many other organisms. These protists also produce oxygen, which is beneficial to many other organisms. Both of these roles are similar to those played by plants. Other protists act as parasites and can cause disease in many organisms, including humans. Protists live in any moist environment, including both freshwater and saltwater, and on the forest floor. Some protists move around in the water. Some simply float in place, and some stick to surfaces. The photographs above show a small sample of the large variety of organisms that are called protists. Seaweed is a multicellular protist that floats in the water and can be found washed up on beaches. Slime molds are organisms that attach to surfaces, absorbing nutrients from them. Diatoms are single-celled algae that float in water and are covered by hard shells. Euglena are single-celled organisms that can move like animals, but also get energy from sunlight. Protists obtain their energy in 3 ways. Protists can be classified by their way of getting energy. Some protists capture sunlight and convert it to usable energy. Another group of protists gets its energy from eating other organisms. A third group gets energy by absorbing materials and nutrients from its environment. Some protists, such as the euglena in the upper left photograph, can even switch from one mode of life to another. They swim rapidly through pond water like animals. If they receive enough sunlight, they look green and make their own food like plants. But if they are left in the dark long enough, they absorb nutrients from their environment like fungi. Check your reading. Explain how the organisms in the photographs above get their energy. Algae. Plant-like protists called algae get energy from sunlight. Like plants, they use the sun's energy, water, and carbon dioxide from the air or water. Algae contain chlorophyll, a green pigment that they use to capture the sun's energy. In the process of transforming energy from sunlight, algae release oxygen gas into the air. This important process, which is called photosynthesis, also takes place in plants and some bacteria. Organisms that perform photosynthesis also supply much of the food for other organisms. Diatoms are examples of single-celled algae. Like all algae, a diatom contains a nucleus which holds its genetic material. Diatoms also have chloroplasts, which are the energy-producing centers that contain chlorophyll. Another type of algae, called volvox, are microscopic colonies of nearly identical cells. These cells, arranged in a hollow ball, look like some single-celled algae. Sometimes cells break off from the hollow ball to form new colonies. The new colonies will eventually escape the parent colony. Colonial organisms, such as volvox, are the simplest kind of multicellular organisms. Seaweed is another example of multicellular algae. All organisms that drift in water are called plankton. Plankton include the young of many animals and some adult animals, as well as protists. Plankton that perform photosynthesis are called phytoplankton, plant-like plankton. Phytoplankton include algae and the cyanobacteria you learned about earlier. Phytoplankton live in all the world's oceans and produce most of the oxygen animals breathe. Protozoa. Protists that eat other organisms or decaying parts of other organisms are animal-like protists, or protozoa. They include many forms, all single-celled. Protozoa cannot use sunlight as a source of energy, and they must move around to obtain the energy they need to survive. Certain chemicals in protozoa can recognize when a particle of food is nearby. The food particle is usually another organism or a part of one. The protozoan ingests the food and breaks it down to obtain energy. Some animal-like protists swim rapidly, sweeping bacteria or other protists into a groove that looks like a mouth. One example, called a paramecium, is shown above. A paramecium moves about using thousands of short, wavy strands called cilia. Another group of protozoa swim with one or more long, whip-like structures called flagella. A third group of protozoa has very flexible cells. Organisms such as the amoeba oozes along surfaces. When it encounters prey, the amoeba spreads out and wraps around its food. A number of protists live as parasites, some of which cause disease in animals, including humans. One of the world's most significant human diseases, malaria, is caused by a protist. A mosquito carries the parasite from human to human. When the mosquito bites an infected human, it sucks up some of the parasite in the blood. When that same mosquito bites another human, it passes on some of the parasite. Within a human host, the parasite goes through a complex life cycle, eventually destroying red blood cells. Check your reading. How do protozoa and algae differ in the way they obtain energy? Other protists. Protists that absorb food from their environment can be called fungus-like protists. These protists take in materials from the soil or from other organisms and break materials down in order to obtain energy. They are called decomposers. The term mold refers to many organisms that produce a fuzzy-looking growth. Most of the molds you might be familiar with, like bread mold, are fungi. But three groups of protists are also called molds. These molds have structures that are too simple to be called fungi, and they are single-celled for a portion of their lives. One example of a fungus-like protist is water mold, which forms a fuzzy growth on food. This food may be decaying animal or plant tissue or living organisms. Water molds live mainly in fresh water. Slime molds live on decaying plants on the forest floor. One kind of slime mold consists of microscopic single cells that ooze around, eating bacteria. When their food is scarce, however, many of the cells group together to produce a multicellular colony. The colony eventually produces a reproductive structure to release spores. Wind can carry spores about, and they sprout where they land. A walk in a moist forest might give you a chance to see a third kind of mold. This organism looks like a fine net, like lace, several centimeters across, on rotting log. These slime molds are not multicellular, but instead one giant cell with many nuclei. They are the plasmodial slime molds. Check your reading. How do fungus-like protists get energy? 9.4. Review. Key concepts. 1. What are two characteristics of protists? 2. What feature do all protists have in common? 3. What are the 3 different types of protists? Critical thinking. 4. Provide examples. Some protists are dependent on other organisms and cannot live independently. Other protists are independent and have organisms that depend on them. Give an example of each. Challenge. 5. Hypothesize. Scientists are considering reclassifying protists into many kingdoms. How might they decide how many kingdoms to use and how to place organisms in these kingdoms?
Chapter 10. Introduction to Multicellular Organisms. The Big Idea. Multicellular organisms live in and get energy from a variety of environments. Key concepts. Section 10.1. Multicellular organisms meet their needs in different ways. Learn about specialized cells, tissues, and organs. Section 10.2. Plants are producers. Learn how plants get energy and respond to the environment. Section 10.3. Animals are consumers. Learn how animals get energy and how they interact with the environment. Section 10.4. Most fungi are decomposers. Learn about fungi and how they get energy. 10.1. Key concept. Multicellular organisms meet their needs in different ways. Before you learned, organisms get energy and materials from the environment. All organisms are organized, grow, respond, and reproduce. Differences in genetic material lead to diversity. Now you will learn about the functions of cells in multicellular organisms, how multicellular organisms are adapted to different environments, about sexual reproduction. Think about, why is teamwork important? For any team to be successful, it is important for people to work well together. Each person has a different role. For example, the team in this restaurant includes people to greet diners and seat them, people to buy and cook the food, and people to take food orders and serve the food. By dividing different jobs among different people, a restaurant can serve more customers at the same time. What would happen in a large restaurant if the diners were seated, cooked for, and served by the same person? Multicellular organisms have cells that are specialized. In single-celled organisms, all the functions of life are performed by one cell. These functions include getting energy and materials, removing wastes, and responding to changes in the environment. In multicellular organisms, however, different jobs are done by different cells. The cells are specialized. A blood cell carries oxygen. A nerve cell sends or receives a signal. Just as the different jobs of running a restaurant are divided among different people, in multicellular organisms, different functions are divided among different cells. In this chapter, you will read about plants, animals, and fungi. These 3 kingdoms. The cells in multicellular organisms are organized in ways that enable them to survive and reproduce. Levels of organization. For any multicellular organism to survive, different cells must work together. The right type of cell must be in the right place to do the work that needs to be done. Organization starts with a cell. Cells in multicellular organisms are specialized for a specific function. In animals, skin cells provide protection, nerve cells carry signals, and muscle cells produce movement. Cells of the same type are organized into tissue, a group of cells that work together. For example, what you think of as muscle is muscle tissue, made up of many muscle cells. A structure that is made up of different tissues is called an organ. Organs have particular functions. The heart is an organ that functions as a pump. It has muscle tissue, which pumps the blood, and nerve tissue, which signals when to pump. Different organs that work together and have a common function are called an organ system. A heart and blood vessels are different organs that are both part of a circulatory system. These organs work together to deliver blood to all parts of the body. Together, cells, tissues, organs, and organ systems form an organism. Organ systems and the organism. In almost all multicellular organisms, different organ systems take care of specific needs. Here are a few examples of organ systems found in many animals. Nervous system enables a response to changing conditions. Muscular system produces movement and supplies heat. Respiratory system takes in oxygen and releases carbon dioxide. Circulatory system delivers oxygen and removes carbon dioxide. Digestive system breaks down food into a usable form. Organ systems allow multicellular organisms to obtain large amounts of energy, process large amounts of materials, respond to changes in the environment, and reproduce. Check your reading. How are the functions of organ systems related to the needs of an organism? Give an example. Different organ systems work together. For example, the respiratory system works with the circulatory system to deliver oxygen and remove carbon dioxide. When an animal such as a turtle breathes in, oxygen is brought into the lungs. Blood from the circulatory system takes up the oxygen, and the heart pumps the oxygen-rich blood out to the cells of the body. As oxygen is delivered, waste carbon dioxide is picked up. The blood is pumped back to the lungs. The carbon dioxide is released when the turtle breathes out. More oxygen is picked up when the turtle breathes in. Multicellular organisms are adapted to live in different environments. All organisms have characteristics that allow them to survive in their environment. An adaptation is any inherited characteristic that increases the chance of an organism surviving and producing offspring that also reproduce. An adaptation may have to do with the way an organism gets its energy or processes materials. An adaptation may relate to the shape or structure of an organism's body. An adaptation can even be a form of behavior. Check your reading. The text above mentions different types of adaptations. Name 3. When most multicellular organisms reproduce, the offspring are not exact copies of the parents. There are differences. If a particular difference gives an organism an advantage over other members in its group, then that difference is referred to as an adaptation. Over time, the organism and its offspring do better and reproduce more. You're probably familiar with the furry animal called a fox. Different species of fox have different adaptations that enable them to survive in different environments. Here are 3 examples. Fennec. The fennec is a desert fox. Its large ears are an adaptation that helps the fox keep cool in the hot desert. As blood flows through the vessels in each ear, heat is released. Another adaptation is the color of its fur, which blends in with the desert sand. Arctic fox. The Arctic fox lives in the cold north. Its small ears, legs, and nose are adaptations that reduce the loss of heat from its body. Its bluish-gray summer fur is replaced by a thick coat of white fur as winter approaches. Its winter coat keeps the fox warm and enables it to blend in with the snow. Red fox. The red fox is found in grasslands and woodlands. Its ears aren't as large as those of the fennec, or as small as those of the Arctic fox. Its body fur is reddish-brown, tipped in white and black, coloring that helps it blend into its environment. The diversity of life on Earth is due to the wide range of adaptations that have occurred in different species. An elephant has a trunk for grasping and sensing. A female kangaroo carries its young in a pouch. The largest flower in the world, the rafflesia flower, is almost a meter wide, blooms for just a few days, and smells like rotting meat. Adaptations are the result of differences that can occur in genetic material. The way multicellular organisms reproduce allows for a mixing of genetic material. You will read about that next. Sexual reproduction leads to diversity. Most multicellular organisms reproduce sexually. In sexual reproduction, the genetic material of two parents comes together, and the resulting offspring have genetic material from both. Sexual reproduction leads to diversity because the DNA in the offspring is different from the DNA in the parents. Two different cellular processes are involved in sexual reproduction. The first is meiosis, a special form of cell division that produces sperm cells in a male and egg cells in a female. Each sperm or egg cell contains only one copy of DNA, the genetic material. Most cells contain two copies of DNA. The second process in sexual reproduction is fertilization. Fertilization occurs when the sperm cell from the male parent combines with the egg cell from the female parent. A fertilized egg is a single cell with DNA from both parents. Once the egg is fertilized, it divides. One cell becomes two cells become four, and so on. As the cells divide, they start to specialize, and different tissues and organs form. Differences in genetic material and in the environment produce differences in offspring. Whether a tulip flower is red or yellow depends on the genetic material in its cells. How well the tulip grows depends on conditions in the environment as well as genetic materials. Most reproduction that occurs in multicellular organisms is sexual reproduction. However, many multicellular organisms can reproduce by asexual reproduction. With asexual reproduction, a single parent produces offspring. Budding is a form of asexual reproduction. In budding, a second organism grows off or buds from another. Organisms that reproduce asexually can reproduce more often. Asexual reproduction limits genetic diversity within a group because offspring have the same genetic material as the parent. Check your reading. How do offspring produced by sexual reproduction compare with offspring produced by asexual reproduction? With sexual reproduction, there is an opportunity for new combinations of characteristics to occur in the offspring. Perhaps these organisms process food more efficiently or reproduce more quickly. Or perhaps they have adaptations that allow them to survive a change in their environment. In the next 3 sections, you will read how plants, animals, and fungi have adapted to similar environments in very different ways. 10.1 Review. Key concepts. 1. How do specialized cells relate to the different levels of organization in a multicellular organism? 2. What is an adaptation? Give an example. 3. What 2 cellular processes are involved in sexual reproduction? Critical thinking. 4. Compare and contrast. How does the genetic diversity of the offspring differ in sexual reproduction versus asexual reproduction? Predict. If fertilization occurred without meiosis, how many copies of DNA would be in the cells of the offspring? Challenge. 6. Synthesize. Do you consider the different levels of organization in a multicellular organism an adaptation? Explain your reasoning. 10.2 Key concept. Plants are producers. Before you learned, multicellular organisms have tissues, organs, and systems. Organisms have adaptations that can make them suited to their environment. Sexual reproduction leads to genetic diversity. Now you will learn how plants obtain energy, how plants store energy, how plants respond to their environment. Plants capture energy from the sun. If you stand outside on a warm, sunny day, you may see and feel energy from the sun. Without the sun's energy, Earth would be a cold, dark planet. The sun's heat and light provide the energy almost all organisms need to live. However, energy from the sun cannot drive cell processes directly. Light energy must be changed into chemical energy. Chemical energy is the form of energy all organisms use to carry out the functions of life. Plants are an important part of the energy story because plants capture energy from the sun and convert it to chemical energy. Producing sugars. Plants capture energy from sunlight and convert it to chemical energy through the process of photosynthesis. The plant takes in water and carbon dioxide from the environment and uses these simple materials to produce sugar, an energy-rich compound that contains carbon. Oxygen is also produced. Plants are referred to as producers because they produce energy-rich carbon compounds using the sun's energy. The cells, tissues, and organ systems in a plant work together to supply the materials needed for photosynthesis. Most photosynthesis takes place in the leaves. The leaves take in carbon dioxide from the air, and the stems support the leaves and hold them up toward the sun. The roots of the plant anchor it in the soil and supply water. The sugars produced are used by the plant for energy and as materials for growth. Check your reading. What is the product of photosynthesis? Another name for a plant is autotroph. Autotroph means self-feeder. Plants do not require food from other organisms. Plants will grow if they have energy from the sun, carbon dioxide from the air, and water and nutrients from the soil. Storing and releasing energy. Plants are not the only organisms that capture energy through photosynthesis. Algae and certain bacteria and protists also use photosynthesis. Plants are different from single-celled producers, however. Plants are multicellular organisms with parts of their bodies specialized for storing energy-rich material. Single-cell producers can store very little energy. Only part of the energy captured by a plant is used as fuel for cellular processes. Some of the sugar produced is used as building material, enabling the plant to grow. The remaining sugar is stored. Often the sugars are stored as starches. Starch is an energy-rich compound made of many sugars. Starches can store a lot of chemical energy. When a plant needs energy, the starches are broken back down into sugars, and energy is released. Cellular respiration is the process by which a cell uses oxygen to break down sugars to release the energy they hold. Some plants, such as carrots and beets, store starch in their roots. Other plants, including celery and rhubarb, have stems adapted for storing starch. A potato is a swollen underground stem called a tuber. Tubers have buds, the eyes of the potato, that can sprout into new plants. The starch stored in the tuber helps the new sprouts survive. Check your reading. What is the original source of a plant's stored energy? Plants are adapted to different environments. Almost everywhere you look on land, you'll see plants. Leaves, stems, and roots are adaptations that enable plants, as producers, to live on land. Not all plants, however, look the same. Just as there are many different types of land environments, there are many different types of plants that have adapted to these environments. Grasses are an example of plants that grow in several environments. Many grasses have deep roots, produce seeds quickly, and can grow in areas with a wide range of temperatures and different amounts of precipitation. Grasses can survive drought, fires, freezing temperatures, and grazing. As long as the roots of the plant survive, the grasses will grow again. Grasses are found in the Arctic tundra, as well as in temperate and tropical climates. Now compare trees to grasses. If the leaves and stems of a tree die away because of fire or drought, often the plant will not survive. Because of their size, trees require a large amount of water for photosynthesis. A coniferous tree, like the pine, does well in colder climates. It has needle-shaped leaves that stay green throughout the year, feeding the plant continually. A deciduous tree, like the maple, loses its leaves when temperatures turn cold. The maple needs a long growing season and plenty of water for new leaves to grow. Plants have reproductive adaptations. It may surprise you to learn that flowering plants living on cold, snowy mountaintops have something in common with desert plants. When rain falls in the desert, wildflower seeds sprout very quickly. Within a few weeks, the plants grow, flower, and produce new seeds that will be ready to sprout with the next rainy season. The same thing happens in the mountains, where the snow may thaw for only a few weeks every summer. Seeds sprout, flowers grow, and new seeds are produced, all before the snow returns. You will read more about plant reproduction in Chapter 3. Some plants have adaptations that protect them. Plants in the mustard family give off odors that keep many plant-eating insects away. Other plants, such as poison ivy and poison oak, produce harmful chemicals. The nicotine in a tobacco plant is a poison that helps to keep the plant from being eaten. Check your reading. Name 2 different types of adaptations plants have. Some adaptations plants have relate to very specific needs. For example, the Venus flytrap is a plant that grows in areas where the soil lacks certain materials. The leaves of the Venus flytrap fold in the middle and have long teeth all around the edges. When an insect lands on an open leaf, the 2 sides of the leaf fold together. The teeth form a trap that prevents the captured insect from escaping. Fluids given off by the leaf digest the insect's body, providing materials the plant can't get from the soil. Plants respond to their environment. During a hot afternoon, parts of the flower known as the Mexican bird of paradise close. As the sun goes down, the flower reopens. The plant is responding to a stimulus, in this case, sunlight. A stimulus is something that produces a response from an organism. Plants, like all organisms, respond to stimuli in their environment. This ability helps them to survive and grow. Gravity. Gravity is the force that keeps you bound to Earth and gives you a sense of up and down. All plants respond to gravity. They also have a sense of up and down. Roots grow down and stems grow up. Suppose you place a young seedling on its side so that its roots and stems stretch out to the side. In a very short time, the tip of the root will begin to turn down and the tip of the stem will turn up. Touch. Many plants also respond to touch as a stimulus. Peas, morning glories, tropical vines, and other climbing plants have special stems called tendrils. Tendrils respond to the touch of a nearby object. As the tendrils grow, they wrap around the object. The twining of tendrils around a fence or another plant helps raise the plant into the sunlight. Light. Light is a powerful stimulus for plants. You can see that stems and leaves grow toward light by placing an indoor plant near a window. After several days, the tips of the stems start to bend toward the window. What happens if you turn the plant around so that those stems reach into the room? The stems will bend as they continue to grow, turning back toward the light. Plants respond to light with the help of a hormone. A hormone is a chemical substance produced in one part of an organism and travels to a different part where it produces a reaction. Hormones act as chemical messengers. They allow an organism to respond to changes in its body or to changes in the environment around it. Auxin, a plant hormone that stimulates cell growth, is produced at the tip of a plant stem. Auxin moves away from light. As a result, the cells on the darker side of a plant stem contain more auxin than those on the lighter side. Higher levels of auxin in plant cells on the darker side cause those cells to grow longer. The longer cells cause the plant stem to bend, moving the tip of the stem toward the light. Plants respond to seasonal changes. Most regions of the world go through seasonal changes every year. For example, during the summer in North America, temperatures rise and the days get longer. As winter approaches, temperatures go down and the days become shorter. These types of seasonal changes have an effect on plants. For plants, a shorter period of daylight will affect the amount of sunlight available for photosynthesis. Shorter days cause many plants to go into a state of dormancy. When plants are dormant, they temporarily stop growing and so require less energy. In temperate climates, the approach of winter causes the leaves of deciduous trees to die and drop to the ground. The trees enter a state of dormancy, during which their growth is slowed. Other plants, such as wild cornflowers, do not survive the change. New cornflowers will grow the following season from seeds left behind. Check your reading. What stimulus causes a deciduous plant to respond by dropping its leaves? For many plants, reproduction is also affected by seasonal changes. For some plants, the amount of daylight is a factor. A few plants, such as rice and ragweed, produce flowers only in autumn or winter, when days are short. They are short-day plants. Long-day plants flower in late spring and summer, when days are long. Lettuce, spinach, and irises are long-day plants. You will read more about plants in Chapter 3. 10.2 Review. Key concepts. 1. What process makes a plant a producer, and what does a plant produce? 2. Name 3 stimuli that plants respond to, and give examples of how a plant responds. 3. How do seasonal changes affect plants? Give an example. Critical thinking. 4. Give examples. Give 3 examples of ways that plants are adapted to their environments. How do these adaptations benefit the plant? Challenge. 5. Apply. Some experiments suggest that the hormone auxin is involved in the twining of tendrils. Use what you know about auxin to explain how it might cause tendrils to twine around anything they touch. Draw a diagram. 10.3 Key concept. Animals are consumers. Before you learned, plants are producers. Plants have adaptations for capturing and storing energy. Plants respond to different stimuli. Now you will learn how animals obtain energy, how animals process food, about different ways animals respond to their environment. Think about: what can you tell from teeth? Many animals have teeth. Teeth bite, grind, crush, and chew. A fox's sharp biting teeth capture small animals that it hunts on the run. A horse's teeth are flat and strong for breaking down the grasses it eats. Run your tongue over your own teeth. How many different shapes do you notice? What can the shape of teeth suggest about the food an animal eats? Animals obtain energy and materials from food. You probably see non-human animals every day, whether you live in a rural area or a large city. If the animals are wild animals, not somebody's pet, then chances are that what you see these animals doing is moving about in search of food. Animals are consumers. A consumer is an organism that needs to get energy from another organism. Unlike plants, animals must consume food to get the energy and materials they need to survive. Animals are heterotrophs. A heterotroph is an organism that feeds on or consumes other organisms. By definition, animals are, quite simply, multicellular organisms that have adaptations that allow them to take in and process food. Animals need food and have many different ways of getting it. For some animals, feeding is a relatively simple process. An adult coral simply filters food from the water as it moves through the coral's body. Most animals, however, must search for food. Grazing animals, such as horses, move along from one patch of grass to another. Other animals must capture food. Most bats use sound and hearing to detect the motion of insects flying at night. Its wings make the bat able to move through the air quickly and silently. What animals eat? Just about any type of living or once-living material is a source of food for some animal. Animals can be grouped by the type of food they eat. Herbivores feed on plants or algae. Carnivores feed on other animals. Omnivores feed on both plants and animals. Another group are those animals that feed on the remains of once-living animals. Many insects do, as do some larger animals, such as vultures. Other animals, such as worms, act as decomposers. Check your reading. Describe how herbivores, carnivores, and omnivores get their energy. Different species of animals have adapted in different ways to take advantage of all of the energy-rich material in the environment. To get energy and materials from food, all animals must first break the food down. That is, they must digest it. Processing food. Energy is stored in complex carbon compounds in food. For the cells in an animal to make use of the energy and materials stored in this food, the large complex compounds must be broken back down into simpler compounds. Check your reading. How must food be changed so an animal gets energy? Digestion is the process that breaks food down into pieces that are small enough to be absorbed by cells. A few animals, such as sponges, are able to take food particles directly into their cells. Most animals, however, take the food into an area of their body where the materials are broken down. Cells absorb the materials they need. Animals such as jellyfish have a single opening in their bodies where food is brought into a central cavity or gut. The unused materials are released through the same opening. A digestive system uses both physical and chemical activity to break down food. Many animals have a tube-like digestive system. Food is brought in at one end of the animal, the mouth, and waste is released at the other end. As food moves through the system, it is continually broken down, releasing necessary materials called nutrients to the cells. Releasing and storing energy. Animals obtain energy from sugars and other carbon compounds the same way plants do, through the process of cellular respiration. As you read in section 2.2, cellular respiration is a process in which energy is released when sugars are broken down inside a cell. The process requires both oxygen and water. Check your reading. What is the function of cellular respiration? Many animals take in water in the same way they take in food, through the digestive system. Oxygen, however, is often taken in through a respiratory system. In many animals, the respiratory system delivers oxygen to the blood, and the blood carries oxygen to the cells. Animals have different structures for obtaining oxygen. Many insects take in oxygen through spiracles, tiny openings in their bodies. Fish have gills, structures that allow them to pick up oxygen dissolved in the water. Other air-breathing animals take in oxygen through organs called lungs. Most animals do not feed continuously, so they need to be able to store materials from food in their tissues or organs. Many animals, including humans, take in large amounts of food at one time. This gives an animal time to do other activities, such as caring for young or looking for more food. Animals interact with the environment and with other organisms. Animals, as consumers, must obtain food as well as water from their environment. An animal's body has many adaptations that allow it to process food. These can include digestive, respiratory, and circulatory systems. Also important are the systems that allow animals to interact with their environment to obtain food. In many animals, muscle and skeletal systems provide movement and support. A nervous system allows the animal to sense and respond to stimuli. Animals respond to many different types of stimuli. They respond to sights, sounds, odors, light, or a change in temperature. They respond to hunger and thirst. They also respond to other animals. Any observable response to a stimulus is described as a behavior. A bird's drinking water from a puddle is a behavior. A lion's chasing an antelope is a behavior, just as the antelope's running to escape the lion is a behavior. Check your reading. What is a behavior, and how does it relate to a stimulus? Some behaviors are inherited, which means they are present at birth. For example, a spider can weave a web without being shown. Other behaviors are learned. For example, the young lion in the photograph learns that a porcupine is not a good source of food. All behaviors fall into one of three general categories: individual behaviors, interactions between animals of the same species, interactions between animals of different species. Individual behaviors often involve meeting basic needs. Animals must find food, water, and shelter. They sleep. They groom themselves. Animals also respond to changes in their environment. A lizard may warm itself in the morning sunlight and then move into the shade when the sun is high in the sky. Interactions that occur between animals of the same species are often described as social behaviors. Basic social behaviors include those between parents and offspring, and behaviors for attracting a mate. Within a group, animals of the same species may cooperate by working together. Wolves hunt in packs, and bees maintain a hive. Behaviors among animals of the same species can also be competitive. Animals often compete for a mate or territory. Check your reading. What are some ways that animals of the same species cooperate and compete? Interactions that occur between animals of different species often involve the search for food. A predator is an animal that hunts other animals for food. Predators have behaviors that allow them to search for and capture other organisms. A cheetah first stalks an antelope, then chases it down, moving as fast as 110 kilometers per hour. An animal that is hunted by another animal as a source of food is the prey. Behaviors of prey animals often allow them to escape a predator. An antelope may not be able to outrun a cheetah, but antelopes move in herds. This provides protection for the group, since a cheetah will kill only one animal. Other animals, such as the pufferfish and porcupine, have defensive behaviors and structures. Animals of different species can also interact in cooperative ways. Tick birds remove ticks from the skin of an impala. This behavior provides food for the bird and provides relief for the impala. Sometimes animals take advantage of the behavior of other animals. Many animals eat the remains of prey left over after a predator has finished feeding. Animals respond to seasonal changes. Animals, like plants, are affected by seasonal changes in their environment. Certain types of food may not be available all year round. A region might go through periods of drought. Some animals do not do well in extreme heat or cold. Unlike plants, animals can respond to seasonal changes by changing their location. Migration is the movement of animals to a different region in response to changes in the environment. Each spring, millions of monarch butterflies begin to fly north from Mexico and parts of Southern California. They first migrate to the southern United States, where the females lay eggs on milkweed plants. The new generation will travel further north and then lay eggs too. Summer monarchs live as butterflies for only 3 to 5 weeks. After 3 or 4 generations, the last generation reaches the northern United States and Canada. Monarchs cannot survive the winter temperatures of the north, so the last generation of butterflies makes the long journey back to Mexico and California in the fall. These butterflies will live 8 or 9 months. The fall migration route is shown on the map. Not all animals migrate in response to seasonal changes. Many non-migratory animals do change their behaviors, however. For example, when winter cold reduces the food supply, some animals hibernate. Hibernation is a sleep-like state that lasts for an extended time period. The body systems of a hibernating animal slow down, so the animal needs less energy to survive. Many animals, including frogs, turtles, fish, and some types of insects, hibernate. You will learn more about different types of animals in chapters 4 and 5. 10.3 Review. Key concepts. 1. In what way are animals consumers? 2. Name 3 body systems that relate to how an animal gets its energy. 3. What is a behavior? Critical thinking. 4. Give examples. Identify 3 categories of animal behavior and give an example of each. 5. Analyze. How is migration similar to hibernation? How is it different? Challenge. 6. Analyze. Scientists often look at feeding patterns as a flow of energy through the living parts of the environment. Describe the flow of energy as it relates to plants, herbivores, and carnivores. Science on the job. Wildlife photographer. An animal's world. Have you ever watched a TV documentary or flipped through the pages of a magazine and wondered, how did the photographer ever get that picture? For a wildlife photographer, understanding animal behavior is essential. Cover and protection. Photographers use camera traps and blinds. A blind, made of branches, is built upwind of an animal gathering place. Steve Winter has built camera traps where a hidden camera emits a beam of light when an animal steps into it. Specialized gear. Certain environments, such as snow and cold, swamp and mud, or seawater, present challenges to a person with a camera. Underwater photographers need to use scuba gear to swim with animals like the Caribbean reef shark. Behavior. To photograph an endangered species, like the jaguar, a photographer must learn animal behavior. Steve Winter and a team of scientists used dogs with keen scent tracking to find jaguars who are active mostly at night. They learned that jaguars have a favorite scratching tree. Perfect photo opportunity. Explore. 1. Observe. With or without a camera, find a spot where you are likely to find wildlife. Sit as still as possible and wait. What animals do you observe? What do they do? 2. Challenge. Interview a photographer about digital photography and ask how technology is changing photography. 10.4. Key concept. Most fungi are decomposers. Before you learned, all organisms interact with the environment. Plants transform sunlight into chemical energy. Animals get energy by eating other organisms. Now you will learn how fungi get energy and materials, about different types of fungi, how fungi interact with other organisms. Fungi absorb materials from the environment. Plants are producers. They capture energy from the sun and build complex carbon compounds. Animals are consumers. They take in complex carbon compounds and use them for energy and materials. Fungi, at least most fungi, are decomposers. Fungi break down or decompose the complex carbon compounds that are part of living matter. They absorb nutrients and leave behind simpler compounds. Fungi are heterotrophs. They get their energy from living or once living matter. They, along with bacteria, decompose the bodies of dead plants and animals. They also decompose materials left behind by organisms, such as fallen leaves, shed skin, and animal droppings. Characteristics of fungi. Except for yeasts, most fungi are multicellular. The cells of a fungus have a nucleus and a thick cell wall, which provides support. Fungi are different from plants and animals in their organization. Plants and animals have specialized cells, which are usually organized into tissues and organs. Multicellular fungi don't have tissues or organs. Instead, a typical fungus is made up of a reproductive body and network of cells that form thread-like structures, called hyphae. A mass of hyphae, like the one shown in the diagram below, is called a mycelium. The hyphae are just one cell thick. This means the cells in the mycelium are close to the soil or whatever substance the fungus is living in. The cells release chemicals that digest the materials around them, and then absorb the nutrients they need. As hyphae grow, openings can form between the older cells and the new ones. This allows nutrients to flow back to the older cells, resulting in what seems like one huge cell with many nuclei. Reproduction. Fungi reproduce with spores, which can be produced either asexually or sexually. A spore is a single reproductive cell that is capable of growing into a new organism. The mushrooms that you buy at the store are the spore-producing structures of certain types of fungi. These spore-producing structures are reproductive bodies of mushrooms. A single mushroom can produce 1 billion spores. Spores are released into the air and spread by the wind. Because they're so small and light, the wind can carry spores long distances. Scientists have found spores 160 kilometers above Earth's surface. Some spores have a tough outer covering that keeps the reproductive cell from drying out. Such spores can survive for many years. If the parent fungus dies, the spores may remain and grow when conditions are right. Fungi reproduce in other ways. For example, a multicellular fungi can reproduce asexually when hyphae break off and form a new mycelium. Yeasts, which are single-celled fungi, reproduce asexually by simple cell division or by budding. Yeasts can also produce spores. Fungi include mushrooms, molds, and yeasts. A convenient and simple way to study fungi is to look at their forms. They are mushrooms, molds, and yeasts. You are probably familiar with all of them. The mushrooms on your pizza are a fungus. So is the mold that grows if you leave a piece of pizza too long in the refrigerator. The crust of the pizza itself rises because of the activity of yeast. Mushrooms. What we call a mushroom is only a small part of a fungus. A single mushroom you buy in the store could have grown from a mycelium that fills an area 30 meters across. When you see a patch of mushrooms, they are probably all part of a single fungus. For humans, some mushrooms are edible and some are poisonous. A toadstool is a poisonous mushroom. The cap of a mushroom is where the spores are produced. Both the cap and the stalk it grows on are filled with hyphae. Check your reading. What is produced in a mushroom cap? Molds. What we call mold, that fuzzy growth we sometimes see on food, is the spore-producing part of another form of fungus. The hyphae of the mold grow into the food, digesting it as they grow. Not all food molds are bad. Different species of the fungus penicillium are used in the production of brie, camembert, and blue cheeses. Some species of the aspergillus fungus are used to make soy sauce. One interesting application of a mold is the use of the fungus trichoderma. This mold grows in soil. The digestive chemicals it produces are used to give blue jeans a stonewashed look. Many molds cause disease. Fungal molds cause athlete's foot. Molds also affect plants. They are the cause of Dutch elm disease and the powdery white mildews that grow on plants. Compounds made from molds are also used to treat disease. Penicillin is an antibiotic that comes from the penicillium fungus. It is used to fight bacterial diseases, such as pneumonia. Molds reproduce with spores, which are typically carried by moving air. The hat thrower fungus, Philobolus, however, has an interesting adaptation for spreading spores. Philobolus grows in animal droppings. It has a spore-containing cap, its hat, that grows on top of a stalk. The stalk responds to light as a stimulus and bends toward the sun. As the stalk bends, water pressure builds up, causing the spore cap to shoot off like a tiny cannonball. A spore cap can be thrown up to 2 meters away. If the spore cap lands in grass, then cows and other grazing animals will eat the caps as they graze. A new cycle begins, with more Philobolus being dispersed in the animal's droppings. Yeasts. Yeasts are single-celled fungi. Some species of fungi exist in both yeast form and as multicellular hyphae. Yeasts grow in many moist environments, including the sap of plants and animal tissues. They also grow on moist surfaces, including shower curtains. Certain yeasts grow naturally on human skin. If the yeast begins to reproduce too rapidly, it can cause disease. Yeasts are used in many food products. The activity of yeast cells breaking down sugars is what makes bread rise. The genetic material of the yeast Saccharomyces cerevisiae has been carefully studied by scientists. The study of this organism has helped scientists understand how genetic material controls the activities of a cell. Fungi can be helpful or harmful to other organisms. Fungi have a close relationship to the environment and all living things in the environment. Fungi, along with bacteria, function as the main decomposers on Earth. The digestive chemicals that fungi release break down the complex compounds that come from living matter, dead matter, or the waste an organism leaves behind. A fungus absorbs what it needs to live and leaves behind simpler compounds and nutrients. These are then picked up again by plants as producers to start the cycle over again. Fungi also live in the sea, recycling materials for ocean-living organisms. Check your reading. What beneficial role do fungi play in the environment? The thread-like hyphae of a fungus can grow into and decompose the material produced by another organism. This means that fungi can be helpful, for example, by releasing the nutrients in a dead tree back into the soil. Or fungi can be harmful, for example, attacking the tissues of a plant, such as the Dutch elm. Most plants interact with fungi in a way that is helpful. The hyphae surround the plant roots, providing nutrients for the plant. The plant provides food for the fungus. Some fungi live together with single-celled algae, a network referred to as a lichen. The hyphae form almost a sandwich around the algae, which produce sugars and other nutrients the fungus needs. Lichens can live just about anywhere. Lichens are found in the Arctic and in the desert. They can even grow on bare rock. The hyphae can break the rock down slowly and capture the particles of newly formed soil. This eventually prepares the ground for new plant growth. On the harmful side, many fungi produce toxins, harmful chemicals. In 1845, a fungus infected Ireland's potato crop, causing the population of Ireland to drop from 8 million to about 4 million. Many people died from disease. Others died from starvation because of the loss of the important food crop. And hundreds of thousands of Irish left Ireland, many emigrating to the United States. Today, several fungal diseases are spreading through the world's banana crops. Check your reading. Name some ways fungi can be harmful to organisms. The toxic quality of a fungus can be put to good use, as in the case of the antibiotic penicillin. The photographs show what happens when a bacterium comes in contact with penicillin. The antibiotic prevents the bacterial cells from making new cell walls when they divide. This causes the cells to break open and the bacteria to die. 10.4 Review. Key concepts. 1. Describe the structure of a fungus. 2. How do fungi reproduce? 3. Describe two relationships between fungi and other organisms. Critical thinking. 4. Analyze. Scientists used to classify fungi as plants. Today, scientists say that fungi are more like animals than plants. How are fungi like plants? How are they like animals? 5. Predict. What might change in an environment where there were no fungi? Challenge. 6. Connect. Think of at least one way your life is affected by fungi in each of their three main forms: mushrooms, molds, and yeast. Are these effects beneficial or harmful to you? Timelines in science. Discoveries in biodiversity. Scientists have discovered new species in the treetops of tropical forests and in the crannies of coral reefs. The quest to catalog the types and numbers of living things, the biodiversity of Earth, began in the late 1600s. A wave of naturalists set sail from Europe to the Americas and to Africa to find specimens of living things. In the late 19th century, biologists reached agreement on a system for naming and classifying each new species. The mid-20th century brought an understanding of DNA and how it could be used to compare one species with another. Now, new organisms could be pinpointed with precision. To this day, millions of undiscovered species lie deep in the unexplored ocean, in tropical forests, and even in heavily trafficked U.S. cities. A large concentration of Earth's known species now live in named and protected biodiversity hotspots. 1670. Marion illustrates from life. In her day, it was typical to work from preserved specimens, but Maria Marion draws from life. Shown below is her illustration of the Legu lizard she observes in South America. In 1670, she publishes a richly illustrated book of insects, the first to describe the process of metamorphosis. 1775. A Catalogue of Living Things. In 1775, Carl Linnaeus completed a book called Systema Naturae. His book outlines a system to organize and name plants and animals. The naming system gave scientists a precise and consistent method for sharing discoveries. 1859. Naturalists in the Amazon. Henry Walter Bates travels from England to the Amazon rainforest in South America with Alfred Russel Wallace. Bates sails home in 1859, bringing over 14,000 specimens, mostly insects. About 8,000 of Bates' finds are new discoveries. Wallace loses his collection in a shipwreck. 1889. Naming discoveries. At the height of discovering new species, conflicts arise over who gets to name living things. In 1889, a conference settles the matter. The first person to publish a description of an organism has the right to claim the discovery and to name it. Technology. Improvements in navigation. Since the age of exploration in the 1500s and 1600s, European shipbuilding and sailing had boomed. Vast improvements had been made, especially in charting and mapping. Travel from Europe to Africa, Asia, and the Americas became an important part of business and science. Still, ships often lost their way or wrecked in raging storms. The invention by John Harrison in 1765 of the marine chronometer, a clock that could work at sea, changed navigation forever. Nobody expected a landlocked clockmaker to solve the puzzle, but the sea clock provided an accurate way to record sightings of stars and planets, and thus plot longitude at sea. The ocean remained a dangerous passage, but now, if tossed off course, a captain could still steer clear. Technology. Living things too small to see. Before the 1600s, scientists were unable to see microorganisms. Anton von Leeuwenhoek, a drapery maker who made microscopes in his spare time, was one of the first to observe these tiny organisms. He made the first observation of bacteria and viewed lake water through a microscope like the one shown to the far right. Others used microscopes to draw detailed close-ups, such as an insect's compound eye shown here. 1970. The bacteria kingdom divides. Dr. Carl Woese discovers that bacteria-like organisms living in places with extremely high temperatures have genetic material very different from bacteria. Woese suggests a new kingdom or domain called Archaebacteria, which is later shortened to Archaea. 1974. Fogging the rainforest. Terry Irwin spouts plumes of insecticide into trees and discovers thousands of new insect species in the rainforest canopy. Irwin uses an organism insecticide that does not harm other parts of the ecosystem. Irwin spreads ground sheets, or big funnels, around a tree, sprays, and after an hour or so, catches the fallen insects, which look like jewels. 1990. Diver, scuba pioneer, heads research. Dr. Sylvia Earle is the first woman to serve as chief scientist at the National Oceanic and Atmospheric Administration, NOAA. Earle uses scuba diving and discovers many new fish and marine species, especially new seaweeds in the Gulf of Mexico. Here, she shows a sample to a student in a submersible vehicle. 2002 to present. New plant genera. Scientists who study plants and insects in tropical rainforests discover so many new species that they are sometimes backlogged in naming them. In the plant kingdom, it has been estimated that for every 1,000 species discovered, one new genus will be found. In 2002, scientists found a new genus of ginger in the wild in Singapore. More and more genera are identified in the laboratory using DNA. Technology. Submersibles. To explore great depths, scientists use manned submersibles or robots. Scientists use the submersible Alvin to discover giant worms around thermal vents. This was at a depth where people thought that life could not exist. Underwater submersibles were also used to discover life in cold springs and to collect bacteria to be used for biomedical research. In addition, unmanned submersibles, such as robots, search for giant squids and other life deep in the ocean. Application. Biodiversity hotspots. In 1988, British ecologist Norman Mike Myers creates a list of 18 areas that he calls biodiversity hotspots. In 2000, Myers and others increased the number of hotspots to 25. How does an area get to be called a hotspot? It has to have a large number of species that exist only in that location, and it has to be a region in great danger of habitat loss. Hotspots cover only 1.5% of the Earth's surface, yet they contain 44% of all species of higher plants and 35% of all land vertebrates. Scientists place most of their effort in discovering new species in these hotspots. Into the future. Although scientists have explored most of the continents, little is known about what life is like in the more remote areas. Deep-sea exploration, for example, is only just beginning, and big surprises surface with each expedition. Technology will continue to delve deeper toward the floor of deep oceans with underwater robotics, manned submersibles, and better mapping and imaging systems. Science will increasingly rely on organizing the growing data on biodiversity. Currently, scientists who study one area of life mostly share data with others in the same field. For example, scientists studying plants share their data with other botanists. An effort to create global databases to share information about all species is beginning to bring together this data. Databases that catalog the genes of living things are being created. The better we know the genetic profile of various species, the better we can identify and compare them. In addition, the attention to the health of biodiversity hotspots and ecosystems everywhere may help stop extinction, the dying off of a whole species. Extinction decreases the diversity of living things, and scientists have recognized that Earth's biodiversity plays a big role in keeping ecosystems healthy. Activities. Writing about science. Documentary. Research one hotspot and report on the diverse species living there. Note any legal steps or other efforts to conserve biodiversity in that area. Reliving history. Devise an animal classification system based on a criterion such as habitat, diet, or behavior. What are the strengths and weaknesses of your system?
Chapter 11. Plants. The Big Idea. Plants are a diverse group of organisms that live in many land environments. Key concepts. Section 11.1. Plants are adapted to living on land. Learn about plant characteristics and structures. Section 11.2. Most mosses and ferns live in moist environments. Learn about how mosses and ferns live and reproduce. Section 11.3. Seeds and pollen are reproductive adaptations. Learn about seeds, pollen, and gymnosperms. Section 11.4. Many plants reproduce with flowers and fruit. Learn about flowers, fruit, and angiosperms. 11.1. Key concept. Plants are adapted to living on land. Before you learned All organisms have certain basic needs and characteristics. The bodies of multicellular organisms are organized. Plants are producers. Now you will learn about plant diversity, about common characteristics of plants, how the bodies of plants are organized. Plants are a diverse group of organisms. Plants are nearly everywhere. Walk through a forest and you're surrounded by trees, ferns, and moss. Drive along a country road and you pass fields planted with crops like cotton or wheat. Even a busy city has tree-lined sidewalks, grass-covered lawns, and weeds growing in vacant lots or poking through cracks in the pavement. Earth is home to an amazing variety of plant life. Plants come in all shapes and sizes, from tiny flowers no bigger than the head of a pin to giant trees taller than a 12-story building. Plants are found in all types of environments, from the icy Arctic to the steamy tropics. Plants share common characteristics. Scientists estimate that at least 260,000 different species of plants live on Earth today. The photographs on this page show 3 examples of plants that are very different from one another. Orchids are flowering plants that mostly grow in tropical rainforests. To get the sunlight they need, many orchids grow not in the soil, but on the trunks of trees. Horsetails are plants that produce tiny grains of a very hard substance called silica. Sometimes called scouring rushes, these plants were once used to scrub clean or scour dishes and pots. Bristlecone pine trees live on high mountain slopes in North America, where there is little soil and often high winds. These trees grow very slowly and can live for several thousand years. You can see from these 3 examples that plant species show great diversity. Despite how different an orchid is from a horsetail and a bristlecone pine, all 3 plants share certain characteristics. These are the characteristics that define a plant as a plant. Plants are multicellular organisms. A plant cell has a nucleus and is surrounded by a cell wall. Plants are producers. They capture energy from the sun. Plant life cycles are divided into 2 stages or generations. Check your reading. What characteristics are shared by all plants? Plant parts have special functions. You could say that a plant lives in 2 worlds. The roots anchor a plant in the ground. Above ground, reaching toward the sun, are stems and leaves. Together, stems and leaves make up a shoot system. These 2 systems work together to get a plant what it needs to survive. A plant's root system can be as extensive as the stems and leaves that you see above ground. Roots absorb water and nutrients from the soil. These materials are transported to the leaves through the stems. The leaves use the materials, along with carbon dioxide from the air, to make sugars and carbohydrates. The stems then deliver these energy-rich compounds back to the rest of the plant. Check your reading. What are 2 plant systems, and what are their functions? Transporting water and other materials. Stems serve as the pathway for transporting water, nutrients, and energy-rich compounds from one part of a plant to another. In most plants, the materials move through a vascular system that is made up of long tube-like cells. These tissues are bundled together and run from the roots to the leaves. A vascular bundle from the stem of a buttercup plant is shown. Transport is carried out by 2 types of tissues. Xylem is a tissue that carries water and dissolved nutrients up from the roots. Phloem is a tissue that transports energy-rich materials down from the leaves. Xylem cells and phloem cells are long and hollow, like pipes. The xylem cells are a little larger than the phloem cells. Both tissues include long fibers that help support the plant body, as well as cells that can store extra carbohydrates for energy. Making sugars. Plants produce sugars through the process of photosynthesis. Photosynthesis is a series of chemical reactions that capture light energy from the sun and convert it into chemical energy stored in sugar molecules. The starting materials needed are carbon dioxide, water, and light. The end products are sugars and oxygen. The chemical reaction for photosynthesis can be written like this. Carbon dioxide plus water plus sunlight yields sugars plus oxygen. Photosynthesis takes place in chloroplasts, structures that contain chlorophyll. Chlorophyll absorbs the sunlight that the chloroplasts need to produce the reaction. Most chloroplasts in a plant are located in leaf cells. As you can see from the illustration on page 89, the structure of the leaf is specialized for capturing light energy and producing sugar. Check your reading. What is photosynthesis, and why is it important? The upper surface of the leaf, which is turned toward the sun, has layers of cells filled with chloroplasts. Vascular tissue located toward the center of the leaf brings in water and nutrients and carries away sugars and carbohydrates. Tiny openings at the bottom of the leaf, called stomata, lead to a network of tiny spaces where gases are stored. The carbon dioxide gas needed for photosynthesis comes in through the stomata, and oxygen gas moves out. This process is called gas exchange. Controlling gas exchange and water loss. For photosynthesis to occur, a plant must maintain the balance of carbon dioxide and water in its body. Carbon dioxide gas from the air surrounding a plant enters through the stomata in its leaves. Open stomata allow carbon dioxide and oxygen to move into and out of the leaf. These openings also allow water to evaporate. The movement of water vapor out of a plant and into the air is called transpiration. Both sunlight and wind cause water in leaves to evaporate and transpire. For photosynthesis to occur, a plant needs to have enough carbon dioxide come in without too much water evaporating and moving out. Plants have different ways of maintaining this balance. The surfaces of leaves and stems are covered by a waxy protective layer called a cuticle. The cuticle keeps water from evaporating. Also, when the air is dry, the stomata can close. This can help prevent water loss. Check your reading. What are 2 ways plants have to keep from losing too much water? The stomata are an adaptation that allows a plant to adjust to daily changes in its environment. Plants can respond to hot, dry weather by keeping their stomata closed. Stomata can be opened during the night, when evaporation is less likely to occur. Most plants have stomata. Some species of plants have special adaptations for survival in a particular environment. For example, a cactus plant has adaptations that allow it to survive in a desert. The spines of a cactus are actually modified leaves. A plant with regular leaves would lose too much water through transpiration. In cacti, most photosynthesis occurs in the thick, fleshy stem, where the cactus also stores water and carbon dioxide gas. Plants grow throughout their lifetimes. Plants grow for as long as they live. This is true for plants that live for only one season, such as sunflowers, and for plants that can live for many years, such as trees. Plants grow bigger when cells at the tips of their roots and stems divide and multiply more rapidly than other plant cells do. A plant's roots and stems can grow longer and thicker, and can branch or divide. However, only stems grow leaves. Leaves grow from buds produced by growth tissue in a plant's stems. The bud of an oak tree is shown on page 91. Plant stems are structures with more than one function. You have read that a plant's stem includes its vascular system, which allows the plant to transport materials between its leaves and roots. Long, stiff fibers in the tissues of the vascular system provide support and give the plant shape. Plant stems can also store the sugars produced by photosynthesis. Many plants, including broccoli, celery, and carrots, convert sugars into starches, and then store this energy-rich material in their stems or roots. Check your reading. What are 3 functions of plant stems? Plants with soft stems. The soft stems and leaves of many weeds, wildflowers, garden flowers, and vegetables die when the environment they live in becomes too cold or too dry. This type of plant survives by using the carbohydrates stored in its roots. Then, when the environment provides it with enough warmth, water, and sunlight, the plant will grow new, soft green stems and leaves. Plants with woody stems. Some plants, such as trees and shrubs, have tough, thick stems that do not die each year. These stems keep growing longer and thicker. As the stems grow, they develop a type of tough xylem tissue that is not found in soft stems. This tough xylem tissue is called wood. The growing tissues in woody stems are located near the outer surface of the stem, right under the bark. This means that, for a tree like one of the oaks in the photograph above, the center of the trunk is the oldest part of the plant. 11.1. Review. Key concepts. 1. What characteristics do all plants have in common? 2. How does the structure of a leaf relate to its function? 3. What tissues move materials throughout a plant? Critical thinking. 4. Summarize. Describe how the structure of a leaf allows a plant to control the materials involved in photosynthesis. 5. Analyze. Do you think the stems of soft-stemmed plants have chloroplasts? How about woody-stemmed plants? Explain your reasoning. Hint. Think about the color of each. Challenge. 6. Evaluate. Scientists who study the natural world say that there is unity in diversity. How does this idea apply to plants? (11.2) Key concept. Most mosses and ferns live in moist environments. Before, you learned all plants share common characteristics. The body of a plant has specialized parts. Plants grow throughout their lifetimes. Now you will learn about the first plants, about reproduction in non-vascular plants, such as mosses, about reproduction in vascular plants, such as ferns. Plant species adapted to life on land. Evidence indicates that life first appeared on Earth about 3.8 billion years ago. Tiny single-celled and multicellular organisms lived in watery environments such as warm, shallow seas, deep ocean vents, and ponds. Fossil evidence suggests that plant life did not appear on land until about 475 million years ago. The ancestors of the first plants were among the first organisms to move onto land. What did these plant-like organisms look like? Scientists think they looked much like the green algae you can find growing in watery ditches or shallow ponds today. Both green algae and plants are autotrophs, or producers. Their cells contain chloroplasts that enable them to convert the sun's light energy into the chemical energy stored in sugars. The first plants. Suppose that hundreds of millions of years ago, the area now occupied by your school was a shallow pond full of tiny floating organisms that could photosynthesize. The sun overhead provided energy. The pond water was full of dissolved nutrients. The organisms thrived and reproduced, and over time the pond became crowded. Some were pushed to the very edges of the water. Then, after a period of dry weather, the pond shrank. Some organisms at the edge were no longer in the water. The ones that were able to survive were now living on land. Scientists think that something like this took place in millions of watery environments over millions of years. Those few organisms that were stranded and were able to survive became ancestors to the first plants. Life on land is very different from life in water. The first plants needed to be able to get both nutrients and water from the land. There is no surrounding water to provide support for the body, or to keep body tissues from drying out. However, for organisms that survived, life on land had many advantages. There is plenty of carbon dioxide in the air and plenty of direct sunlight. Check your reading. Why is having plenty of sunlight and water an advantage for a plant? Your answer should mention photosynthesis. Mosses and ferns. Among the first plants to live on Earth were the ancestors of the mosses and ferns you see today. Both probably evolved from species of algae that lived in the sea and fresh water. Mosses are simpler in structure than ferns. Mosses and two closely related groups of plants known as liverworts and hornworts are descended from the first plants to spread onto the bare rock and soil of Earth. Ferns and their relatives appeared later. Mosses are non-vascular plants. Moss plants have adaptations for life on land. For example, each moss cell, like all plant cells, is surrounded by a thick wall that provides it with support. Moss cells also have special storage areas for water and nutrients. Mosses do not grow very large, but they have simple structures that function like roots, stems, and leaves. These adaptations help moss plants survive on land, while algae survive only in water. If you look closely at a clump of moss, you will see that it is actually made up of many tiny dark green plants. Mosses belong to a group called non-vascular plants. Non-vascular plants do not have vascular tissue. Water and nutrients simply move through non-vascular plants' bodies, cell by cell. A plant can get enough water this way as long as its body is no more than a few cells thick. Check your reading. What limits the size of moss plants? Water also plays a part in the reproductive cycle of a moss plant. In the first part of the cycle, the moss grows and maintains itself, producing the male and female structures needed for sexual reproduction. If conditions are right and there is enough water, the plant enters the spore-producing stage, the second part of the cycle. Mosses reproduce with spores. Mosses, ferns, and fungi all reproduce with spores. Spores are an important adaptation that allowed the ancestors of these organisms